A very good morning to one and all present here. I welcome you all to the virtual second international conference on clinical and applied microbiology on December 14th and 15th, 2023, organized by BioLEAGUES, Sinha College of Pharmacy, our academic partner, and United Innovators, our research partner, and the Leaders Magazine, our media partner. BioLex is a not-for-profit professional association which prominently promotes research and development. We at BioLex have brought a revolution in the field of worldwide conferences. The conferences organized by BioLex bring together the professional research and leaders who have explored all venues in the field of life sciences, medicine, and technology. BioLex conducts events worldwide which help in enhancing the skill set of the people from diverse industries and forms a common platform for eminent personalities, physicians, researchers, doctors, and academicians, professional business figures, and much more. BioLeague Conference encourages better comprehension about improvements and progresses over the world. We work with our motto of creating a better tomorrow by organizing conferences and creating a network which will help grow a better tomorrow with the help of advanced technology and achievement. The second International Conference on Clinical and Applied Microbiology, second ICCAM, is organized by BioLeaks to be held on 14th and 15th December 2023 virtually with the unique theme of sustainable future through microbial innovations. With the aim of connecting top applied research and early stage innovation from universities, labs, and startups with industry end users and prospectors. This unique event has taken the initiative to gather the world-class experts both from academic and corporate industry in a common platform to share the research findings to the world and enlighten other esteemed delegates on the latest trends in the field of microbiology. The conference seeks to fill knowledge gaps and roadblocks related to microbiology. It gives me immense pleasure to introduce our honorable dignitaries and keynote speakers with much pride. Now, I would like to introduce our founder and CEO of BioLeaks, Mr. Rudra Banu Satpathy, and our conference organizing secretary, Dr. Shinshan Das. Now, I would like to introduce our keynote speakers of the day. Professor Dr. Jugsharan Singh, Professor, Department of Microbiology, University of Delhi, India. Dr. O.S. Aisha, Vice Principal and Head, PG and Research Department of Microbiology, Muhammad Sadak College of Arts and Science, India. Professor Dr. Renu Bharadwaj, Consultant Microbiology, Section Editor, International Journal of Antimicrobial Agents, Editorial Board Member, BMC Infectious Disease and Indian Journal of Medical Microbiology, India. Dr. Narayana Patil, Associate Professor, School of Bioengineering, Sciences and Research, MIT, Art Design and Technology University, India. Dr. Rajashekar Reddy Ponnuru, Principal and Head, St. Peter's Institute of Pharmaceutical Sciences, India. Now, I would like to welcome and introduce the session chairs of the day, Dr. Y. Katyayani, Assistant Professor, Microbiology, Guntur Medical College, India. Dr. Chinchan Das, Founder, Benevolencia Foundation, Government Registered, India. I would like to welcome all the participants and we are looking forward to your active participation today. Now, I would like to thank each and everyone who have gathered here. Now, I kindly request the founder and CEO of BioLeaks, Mr. Rudra Banu Satpathy, to welcome the gathering by his welcome address. Sir, please.
Very good morning, one and all, and thank you very much, Niveta, for this introduction and invitation. It's really a great morning here, and I wish everyone my all best wishes and regards, as well as welcome to second international conference on clinical and applied microbiology. As we all know, as a responsible organization, today at this knowledge zenith era, it's highly important that continuous medical education as well as continuous professional education has to be practiced in a regular routine. So we have been facilitating this type of academic and educational programs all across the globe by connecting universities in India as well as all across the world so that we unite and congregate and share our knowledge and experience. I express my hearty gratitude to all the revered keynote speakers, esteemed dignitaries, revered organizing secretaries, office bearers, organizing committee members, and all of you for this wonderful and valuable time. Moreover, I express my hearty gratitude to all 100 plus participants, those who have joined here, and I wish my all the best towards all 72 plus presentations what we have, and my gratitude once again to all 9 revered keynote speakers. So India has been a potential market towards microbiological sectors. If we talk about medical microbiology, we talk about industrial microbiology, we talk about agricultural microbiology or environmental microbiology as well. We are keen in producing microbiologists, those who are working all across the globe towards sustainable development, ecological security, soil and water pollutions as well. The most important sectors we have been highlighting is towards agricultural microbiology. So we are connecting with experts and professionals across the country so that agricultural productivity could be developed through biofertilizers, pests and disease management, as well as eco-friendly agricultural practices could be done. The third most important vertical is pharmaceutical microbiology, on which we are connecting with the experts so that we can work towards the capacity building of pharmaceutical institutions, colleges, and universities, and train them through our programs of scopes and research in microbiological industrial sectors, pharmaceutical sectors after their education as well. We are also connecting with very sensitive professionals like oncology and cardiology, diabetology, where we deal with microbiology in terms of diagnostics, pathology, infectious disease management, patient care, and treatment as well. The very important and the most prior aspect on which we need to address today is to create a global collaborative environment on which we have been working. We are connecting with institutions. We urge healthcare institutions, colleges, and biotechnology universities, those who are focusing on microbiology as well, to come forward and collaborate with us so that we can join hands with overseas universities. Because in upcoming next 4 years, India is going to be one of the largest academic destination that will create a huge impact of higher education. From the day of beginning of your research, we have been prioritizing to provide you all possible assistance in terms of technology, in terms of community management, in terms of networking, as well as in helping you out to publish and your research and go for the patent filing also. We have all end-to-end possible services that helps you to go global and think beyond your educational campuses. Thank you very much. I wish you all the best.
Thank you, sir. Thank you so much, sir. Now we'll move on to the keynote lectures we have been waiting for. Now I request Professor Dr. Juksharan Singh to render the lecture. Sir, please.
Hello.
Good morning, sir.
Can you see my
Yes, sir, we can see your
screen?
Yes, sir, we can.
Okay. And shall I start?
Yes, sir, please.
All right. Well, good morning. Good morning to everyone. See, I am very happy to be here to be sharing some of our thoughts, some of my thoughts with the global audience regarding the antimicrobial resistance. I have just been listening to the CEO and Rudra, and I'm very happy that they are organizing or they can or they are in fact raising a platform where scientific research and scientific findings can be discussed, and some of these recommendations then, which are very, very important, can be communicated to the relevant agencies. Well, starting my lecture, see, this lecture is on developing a point of care test for detection of antimicrobial resistance. Antimicrobial resistance, as you know, by this time, it's a buzzword almost everywhere, and not for any wrong reasons. This means the development of the resistance to the antimicrobial agents or to the antibiotics, to say, and as a result of which these microbes, whether these are bacteria or whether these are viruses or parasites, they cannot be killed by the antimicrobial agents. So that's what is AMR. You must have heard it in with other terms like drug resistance or antibiotic resistance. But currently, the most relevant term is the AMR. Let's see. And it is a point of care test which we have been developing in our laboratory for the detection of this kind of resistance in bacteria, so that a relevant antibiotic can be used for the treatment of the infection. Let's see. See, by the year 2050, the AMR is projected to emerge as a formidable challenge. In fact, this has come from a very detailed report of Jim O'Neill in a study which was conducted by the British government. And of course, not only that, which galvanized the report, which galvanized the whole situation. Besides that, scientists all over the globe have been emphasizing or they have been reiterating about this challenge. The challenge is, as I said, AMR, the antimicrobial resistance. That means the development of the resistance by the microbes. Now, how are we going to this challenge is going to be really a big challenge. And if I'm not wrong, whatever has been projected about AMR is no less than a catastrophe. That means because of this AMR, by 2050, we are going to run into a big problem. Or rather, I won't be wrong if I say that the medical science or humanity, even if I say humanity, is going to run into a big trouble if if this challenge is not taken up. The reason being that with the development of these AMR or antimicrobial resistance microbes and no new antibiotics coming up, the medical science will not be able to perform some very sophisticated surgeries like heart surgery or joint surgeries or anything else. It may not be possible for the medical science to carry out all these things. Well, this is all what has been projected. See, and the resistant microbes will be responsible for the causation of the infections, the magnitude or the number of which will be much more than the things like cancer or other lifestyle, or you can say other non-infectious diseases. So this is what has been projected. Now, how are we going to, if we do not take, measures to avert this challenge or avert this catastrophe, we are going to run into a real problem. So how this challenge, how are we going to meet this challenge? It's a big challenge. Well, I have just listed a couple of things just to orient yourself toward this lecture. One of them is, one of the very important thing would be saving the current inventory of the antibiotics. That will be one thing. Developing novel or newer antibiotics. Well, they are not easy to come by. We know we have almost run into a molecule deficit. Rather, we are not getting any new molecules as far as antibiotics are concerned. Of course, if some novel breakthrough comes up and we find out, we come across a very newer niche from where we can get sources of the antibiotics like microbes from the seabed or something like this, which has not been explored earlier. And if we are able to get some novel antibiotics, that's it, which won't be resistant, to which the microbes won't be resistant, that's a different matter. But for the time being, we don't have anything. Developing novel antimicrobial agents than antibiotics. Fine. Well, a lot of research has been is going on to develop antimicrobial agents which are not antibiotics. Like, for example, just to illustrate this, peptides. Well, we have been working on peptides for a very long period of time. But of course, we have not come across any peptide which is anything near to the antibiotics. So we are keeping our fingers crossed. Developing novel diagnostics for the infectious diseases, which means we can diagnose the infectious diseases early, we can quickly control them, prescribe antimicrobial agents so that they do not continue to procrastinate for a longer period of time, and they do not act as a nidus for, you can say, spread of the infectious diseases. So diagnostics has a very, very important role to play in infectious diseases, we know, but at the same time, if we develop novel or new diagnostics, there's a lot of role in averting the AMR. See, it has. Then another one is One Health approach. Well, I don't know how many of you have heard about this word, but this is One Health means the problem of the AMR cannot be solved simply by medical personnel or scientists or clinicians. Other two components of the environment, that means the veterinarians who are equally using a large number of antibiotics for treatment of the ailment of the these animals, they should also join hands. It's not that they should. Similarly, the environmentalists and the agriculturists who are also using antimicrobial agents, they are also into the loop. That means all the three large components of the whole, you can say, earth. That means the medical scientists and all the paraphernalia associated with it, the veterinarians, all the paraphernalia associated with this, and the environmentalist and the agriculturist. Okay, they all put together should come, because all of them, they are into the loop for the development of the antimicrobial resistance, and everyone has to play their role in a concerted manner so that we can avert this challenge, or we can meet this challenge or avert this catastrophe. So this is what is called as a One Health approach, which of course is again a sort of a lot of collaboration between scientists coming from the three worlds. As I said, the environmental or agriculturalists plus the medical plus the veterinarians, and tackling the AMR situation. Well, that is a altogether different ball game. But anyway, so this is some of these methods. Well, there are several other things which may be which are being done or can be done to avert this catastrophe or to meet this formidable challenge of the AMR. Now let's come back to the topic I'm going to talk. This topic basically falls into the first objective, that means saving the current inventory of the antibiotics. Or you may also put this subject as a development of the newer diagnostics for infectious diseases. as I said, they are also going to play a very, very important role in the in meeting our goals for the challenge of the or the, you can say, control of the drug resistant microbes. So let's see what is the type of the work we have been doing. Just to give you a small idea, what is a point of care test? A test. So we are going to develop that kind of a test for detection of the antimicrobial resistance in the microbes, so that quickly they can be detected and quickly they can be treated. So point of care test means a simple, and I'm just going to give you a list, because this is something one should know. A simple and a rapid test, less than 30 minutes. The less than 30 minutes is an international standard which has been put by some of the agencies which are dealing with AMR. They want that anything beyond 30 minutes may not meet the requirements, or may not be a real, you can say, use. But anyway, this is debatable, but less than 30 minutes is the current international thing, or 1 hour. Americans, some of the American agencies like BARDA, they have put one minute, sorry, 1 hour or 60 minutes, whereas British standards, some of these agencies which are funding agencies like Longitude Prize, etc., they have put 30 minutes. That means you should be able to detect antimicrobial resistance is less than 30 minutes. Okay. Then require minimum, and these kind of tests, they require minimum manipulations. Results can be interpreted unequivocally, does not require much expertise, give a actionable results. Okay, preferably see near the bedside or the near the patient on the spot. This test can be done, and preferably in a format which is a very simple sample in, result out. That means you put the sample in, let's say blood or urine, and quickly the results come out. That should be the format, and it should be cost effective, scalable. Well, this kind of a test for a detection of the antimicrobial resistance or sensitivity, it is a formidable challenge itself to develop this kind of a test, so that we can detect resistant microbes quickly and then treat them so that they do not spread. And developing that kind of a test is a, as I said, is a real challenge. Now let's see, just to give you an example. Okay, see, these are the 3 very well-known tests. You must have seen this pregnancy test. People can do at home. This, you can see the things like AccuCheck and a variety of others, medical, I mean, simple aids which can detect sugar at home, or even this, see, test which gives you, which was very popular during the COVID pandemic, that you can detect your temperature and a variety of other parameters. These are all point of care tests. People can do themselves, the patient can do himself in a very, very, the results are not interpretable quickly without any ambiguity. Okay, but for each of these tests, you can see the chemistry, the mechanics, the electronics, all these things, the scalability and other things which are put behind these kind of tests is really very complex. These are very, although very simple, I would say, parameters for which I have projected this, but developing a similar kind of a test for AMR is very difficult, very difficult. Why is that? Why is that? See, the main reason for that is for detection of the antimicrobial resistant, or to detect antimicrobial or the resistance of the microbes to antibiotics, whatever tests are currently available, or see, we have to grow the microbes. And minimum the microbes will take at least 18 hours, 24 hours, or let's come to still lower, 18 hours or 12 hours. That is how, or maybe 10 hours, you have to minimum grow them, and that is how you detect the resistant of the, or the sensitivity of the microbes to the antibiotics, and then prescribe the relevant antibiotics to the patients. This is what happens all over the globe. If you look, see, this is a ward here. The sample, if you want to know what kind of infection he has and what is the antibiotic to be prescribed, the sample will go to the lab, it will come back. Okay. But here it will similarly, if it is OPD, again the sample will come back to the centralized laboratory and come back. Again, it will take time and before you can prescribe any antibiotics. So that means by the time the antibiotic, the report of the sensitivity or resistance of the microbe comes, you have to prescribe the antibiotics. This patient or any person here who is suffering from an infectious disease, you cannot leave him without antibiotics. You have to give some kind of an antibiotics. It's a different matter that once the results of the antimicrobial resistance comes back, you might have to change your antibiotics. That's a different matter. And that's how it is done. But you have to prescribe an antibiotic empirically. The clinician based or the treating patient person the doctor has to prescribe an antibiotics for this purpose, because these results will come only after, let's say, 18 hours, or let's say only after 12 hours. If I go by the latest automated antimicrobial testing laboratories or instruments, even it will not be less than 12 hours or 18 hours or so. But you cannot leave the patient during that period to antibiotic. And the problem is during that. When the antibiotic is prescribed here, the antibiotic would be prescribed a broad spectrum antibiotics. See, patient with suspected infection, you can see here, patient is the ward on the OPD. Doctors prescribed antibiotics empirically. Okay. And then prescribes generally a broad spectrum antibiotic. Even for a simple infection, it could be a broad spectrum antibiotics. And that is how this green color and then this yellow color arrow you can see, and then this is the usual thing, the usual practice. And these are on the sideway panel, you can see a variety of the tests, which are some of these Vitec2, Phenex, Microscan. These are the latest equipment which are used, and these are the classical above the antibiotic sensitivity by disk method or by the e-test, which will, again, as I said, will minimum take not less than 18 hours or so, and some antibiotics has to be prescribed. And generally this antibiotic prescribed in the absence of the sensitivity or resistance data, in the absence of that, the antibiotic prescribed is a broad spectrum antibiotic. And while it may not be needed, as a result of which we are losing our broad spectrum, those antibiotics which are real a gold for us to treat infections. We are using them for no reason, as a result of which the microbes get exposed to these newer antibiotics, and they develop a resistance to them. So that means if we can develop a test, as I said, in less than 30 minutes, if you can develop a test in less than 30 minutes, the most relevant antibiotics will be prescribed to the patient, as a result of which we will be able to save our current antibiotics. See, so that is how a test, which is a quick test, which may be a point of care test or any test which is very quick for development, for the detection of the antimicrobial resistance, would add to our efforts to control not control, just to, you can say, control, or you can say avert, the emergence of antibiotic resistance, or AMR, in bacteria. See, so that means, see, if we have to go for this kind of a test, one thing is very clear. that you cannot do culturing. All the cultures are out because 30 minutes or 1 hour or 2 hours, 3 hours, if you want this test to be done in this much time, all the culturing things are out. See here. Remember, so sensitivity or research to the different antibiotics. If you have to find out this, the sensitivity or resistance, remember the idea is to develop a POC. All prerequisite of POC has to be met. Remember the results of the sensitivity of the bacterium or resistance of the bacterium to antibiotics should be available in less than 30 minutes or let's say 1 hour. So all culture methods are out. Something innovative must be done, which means so what is that innovation? What is the current international effort to do that? It is a people are or scientists are developing a combination of short culture in presence of antibiotic and imaging. Short culture, I mean, it could be less than 30 minutes, but will that work? We really don't know. 1 hour, 5 hours. Yes, people have seen that up to 5 hours. If you take antibiotics and take the infecting microbe and incubate them for 5 hours, there will be some changes in this microbe, and which will tell whether it is resistant or whether it is sensitive. Okay, but what are those changes? Hardly you see any metabolic changes which are very, you can say, very foolproof. Some morphological changes can be seen. Yes. So, but then do you think the morphological changes in the antibiotic we will be in a position to identify whether the microbe is resistant or whether it is sensitive? Yes. The scientists say yes, it is possible, and but that has to be detected with the help of bacterial imaging. So that is what is the current, you can say, topmost approaches. Well, there are several other approaches people are working, even in this format. That means when there is a combination of short culture and bacterial imaging, variety of other people are doing, scientists are doing a variety of modifications, permutations, and combinations to come to this, to come to how to get the sensitivity in or resistance in less than, not less than 30 minutes, 5 hours, or even 6 hours or so. Let's see how do we do it. See, one of the method which is currently by my reading or by my, you can say, knowledge goes, is almost is the one of the front runners. One of the front runners is in a Swedish laboratory where they are taking these antibiotics. Can you see this picture? You will see this picture where this kind of a Tarasky plate or this kind of a micro titer plate is used, where there is a well here. You can see this well, a smaller well, which where the bacterial which is to be tested is put in agarose, in the presence of the agarose. These wells contain agarose where the bacterium is put, and there is a diffusion chamber along with that of the antibiotics. That means the microbes get exposed to this antibiotic for a very short period of time. Here they get exposed to this antibiotic for a very short period of time, and this is done to a large panel of antibiotics, and they are exposed for 1 hour, 2 hours, 3 hours. The least is the better. And then you can see here, from here we can see for a short period of time what is the morphology of these bacteria, and then image these bacteria, imaging hundreds and thousands of these bacterium, and then using the analytical tools you can come to the conclusion whether the microbe is sensitive or it is resistant. That means something you would be able to do in current projection is by this company or the university who are doing a collaborative research, is that they have been able to do this kind of detect changes in the microbes in less than 5 hours. That means within 5 hours they will be able to tell whether the microbe is resistant or it is sensitive, and as a result of which you can prescribe the appropriate antibiotic. That means you don't have to prescribe an antibiotics which is empirically, but then even if this test is done in 5 hours, this test or sensitivity is available in 5 hours, do you think 5 hours is enough? No, probably at this stage it is no, because the person must be prescribed antibiotics immediately. Okay, not for 5 hours or 6 hours, because if you prescribe an antibiotic after 5 hours or 6 hours of looking at these results, it may be too late. So those problems are there, but this is currently one of the largest, you can say, front runners technology, which is being used by a variety of scientists all over the globe. And this is just to illustrate. Now let's come back to what we have been doing in our laboratory. As I said, a variety of different kind of approaches can be used. Our approach is this. See, it is a resistance gene array. Our approach is resistance gene array based rapid detection of AMR and an algorithm driven therapy. This is our group at the University of Delhi South Campus. This is the prize. This is the money which we got from the UK charity, that is the Longitude, some money funding for the development of these tests. Of course, we have got funding from other Indian agencies also. The idea is, what is the technical feasibility and plan? The idea is that you have a sample. Sample means any clinical sample, which is blood, urine, or CSF, whatever. Then you do immediately this goes into the you can say into the point of care instrument. This drop of blood goes into this, where it is stripped off and the DNA is taken off. This DNA is melted, denatured. It is labeled, of course, with fluorescent dyes. Or see, there are options here also, and we are developing what kind of those options are there. And then finally it is put into a format. This is the further, you can say, the detailed picture where the analysis is done. Here, this you can see here, these are the array, the resistance gene array, and this is already there. The DNA or the melted DNA or the fragments of the DNA which are labeled, they will come and they will attach complementarily with the array, as a result of which those will be highlighted up. This information will go, and then we will know what kind of resistant microbes are there. Based on this kind of a profiling, we would know that what antibiotics are to be used. Now, these antibiotics which are suggested, they are based on the WHO or the recent recommendations, that you use only those antibiotics which you feel can be they are not the broad spectrum one first, and then only if the clinician feels or the doctor feels that it is they will not work, then and only then the suggested treatment is the next one. That means most of the time we are trying to see, you will be able to prescribe antibiotics, antibiotic for an infection which is quite targeted, but at the same time may not be broad spectrum, and you will be able to not be exposing your broad spectrum antibiotics to every small infection, thereby developing the microbes resistance to that. So that is our approach. This is what we have been doing. This is what I call it. As I said, this is a resistance array. Here is the resistance array. This is the resistance array of the antibiotic, of the resistance genes, antibiotic resistance resistant genes. So resistance gene array based rapid detection of AMR and an algorithm driven therapy, because it will be based on a variety of information. At the same time, the WHO recommendations of how the antibiotics they are to be used, so that we can conserve our broad spectrum antibiotics. So what we have been doing is, you can see these are variety of antibiotic resistance gene databases which are available. See, these are variety of them. We have selected some of them. The card we have selected, which is basically a McMaster University. This is found to be one of the best databases, updated very regularly for the resistance gene array. This is a CBMR. This is our own database of the beta lactamases. See here, this is this database we have made. So we have used all this data to identify the resistance gene array. See, it's a big problem. It's a big, you can say, a job to do all this. What kind of genes to be selected? All genes have to be selected. And how to make these arrays, how many of them. So these things are in progress, but we have made a lot of progress in this. So these, see, everything fine. My lecture is being heard. Hello.
Yes, sir, we can hear you, sir. Please go
Okay,
ahead.
okay. So see, these are the work components: resistance gene array development, selection, in situ DNA isolation, melting, shearing, labeling of the DNA, resistance gene array, microarray design, algorithm driven selection of the antibiotics, and integration of the components 1 to 4 using a microfluidics point of care format. These are the five components, and we have got different number of students and different teams, we can say, who are working on each one of these things, and these which are to be integrated. There are postdocs involved, there are doctoral students involved who are selecting these things one by one, and we are, you can say, into the process of making this equipment. How is this going to be? How it would look like? It will be something like this. Finally, the equipment would. This is the first, you can say, after, you can say, this is a design for. Rather, I would say, after a lot of reiteration, we have come to this kind of a design. There is a upper panel, and then there is a which will primarily contain all the DNA thing, sharing, labeling, etc. And then this is a central module. This central module is the one which will contain array. The DNA fragments will come, will hybridize here, and the signals are picked up. And then this is the third component where the option for the antibiotics for the treatment would be displayed. Option 1 will be those antibiotics trying to use the most antibiotics which have been used, but at the same time would be effective to the microbe. And option 2 or 3, they are used only in case which will be broad spectrum antibiotics, and will be used only in case of the first one doesn't work. So this is a kind of a handheld device which we are thinking it will come, and this is how it will look, something like this. As I said, this is the module number 1, where this is the sample inlet. How the sample will be taken in. It will be DNA, will be taken out, sheared, melted, everything. Waste goes into this thing, all the liquid waste, liquid reservoir, buffers, etc. There are reservoirs for this, all this thing. And then from here, the labeling will also take place. And then finally, the DNA of the resistant microbe comes into this array, where you can see this array is there. These things are the resistance gene array. Hybridization takes place, and then the signals of which kind of resistance genes are present will be picked up and then collated into a treatment option 1 and 2, which, as I said, is again based on a variety of parameters, so that we can conserve our antibiotics. The moment you put this thing, it will take this is that place, this is the module which is taking a lot of time. Preferably it should be half an hour, but not possible at this stage. It will be only a couple of hours which we have been able to think of, and then this is what we are developing. And this module is removable. See, the moment you do process one sample, you click this module, this will open up, remove that cassette of this array, put the new one, so that it is the thing is ready for the equipment is ready for the new for the next sample. And then, of course, there will be a washing and other things of these things of these components during this period. So this is how we have been going step by step. You can see it's a huge job which we have been doing for the last 5 years, and let's see how we are going to progress. Slowly and slowly we are progressing, and people, as I said, they are using a variety of other methods. MALDI-TOF is being used, a variety of other quick methods are being used, but imaging is one of the front runners in this area, where we can, because time is the time amount of you can say time, in how much time the results of the AMR would be available is the real factor here. But it's a formidable a challenge which people are trying to meet all over the globe, to develop this kind of a quick or a point of care diagnostic for detection of antimicrobial sensitivity or resistance, in our bid to contribute to the aversion, or to avert this catastrophe of this projected catastrophe of the antimicrobial resistance throughout the globe. See, there are almost all national agencies, other international agencies like WHO, CDC, and even the General Assembly of the United Nations, they have formed various committees to look into this problem. All national governments are supposed to make committees and take this kind of measures, a variety of measures to control under the One Health program to control AMR. But as I said, it's a big challenge for the scientists, and if we are not able to meet this challenge, the projected things are really very bad. I'm slightly not using the words humanity or catastrophe, but these are the words which have been clearly used in the literature, so that we can avert this kind of a catastrophe. See, these are some of all my students who are working. Some postdocs are also there. We have been working on genomics of Yersinia in my lab for almost 30 years, and this is how developing from this, because this is a highly resistant organism on antibiotics. So slowly we have been getting into AMR for the last 7 to 8 years, and we have more channeled our energy more toward this side. And then I would like to thank all the national agencies from where I don't think there is any national agency from where I have not our group has not taken funds for the development of this work. And for finally, I would like to thank my department, where all this work has been done or is being done. This Department of Microbiology at University of Delhi at South Campus in New Delhi. And I take this opportunity to thank you all for listening very patiently to this lecture. Thank you very much.
Thank you, sir. Thank you so much for your wonderful keynote session.
My pleasure.
Yes, sir.
So if there are any questions, I would be very happy to answer these.
Yes, sir. Participants, if you have any question, you can raise your hand or else you can put it in chat box.
So see, I'm going to stop sharing, okay?
Okay, sir.
All right. Okay.
Okay.
All right.
Thank you, sir. I think no one has any question. We'll move on to the next keynote session now. Now, I request Dr. O.S. Aisha to render the lecture. Ma'am.
Can you hear me, ma'am?
Yes, ma'am, we can hear you.
Yes. Good morning, everyone present over here. I'm
Good
Dr.
morning.
Aisha. I'm Dr. Aisha, Vice Principal and Head, PG and Research Department of Microbiology, Moments of the College of Arts and Science, Sholinganallur, Chennai. Madam, can you see the screen?
Yes, ma'am, we can see your screen,
Yeah,
but
can
we
you
can't
start
see your
now?
slide, ma'am.
Slide
We
is not
can,
visible. Okay,
yes.
ma'am. There is some technical error. I'll let you.
Okay, ma'am, sure. Yes, ma'am, we can see your slide now.
Yes, ma'am. I'm Dr. Aisha, Vice Principal and Head, PG and Research Department of Microbiology, Moments of the College of Arts and Science, Sholinganallur, Chennai. I am very happy to share my views regarding the bacteria which are being used in immunotherapy. It's a group of cancer is a group of diseases involving abnormal cell growth with the potential to invade or spread to other parts of the body and disrupt the body's regular function, as you all already know. According to World Health Organization, cancer has been accounted for almost 1 in 6 deaths in 2020. It is one of the second leading cause of death in the world. And the reasons are many, but there is the complexity of cancer cells, like the following, followed by the following reasons. So first one, let us say about the complexity of cancer cells. Complexity means they are able to mutate and they are able to adapt to all the environment, and it leads to a diverse population with varying characteristics such as gene expression, metabolism, and metastatic potential. Can you hear me, ma'am?
Yes, ma'am, we can hear you, but your slides are not moving, ma'am.
Can you see, ma'am, now?
Yes, ma'am, we can see the first slide number 4.
Slide number 2 is
4.
visible or not?
Yeah, ma'am. It is visible. Yes.
Yes, the first slide. Okay. So today I'm going to tell you about the cancer and what are the bacteria to be used up in the immunotherapy. So nowadays many cancer tumors are being produced all over the world, and it's a group of diseases involving abnormal cell growth with the potential to invade or spread to other parts of the body and disrupt the body's regular function. According to World Health Organization, cancer has been accounted for almost 1 in 6 deaths in 2020. It is one of the second leading cause of death in the world. So can you see the slide, ma'am? Hello?
Hello, ma'am.
Can
Yes,
you see
ma'am.
it, ma'am? Yes, ma'am.
Yeah, ma'am, we can see your
If
slide.
there is anything, if you couldn't see, you just tell me and let me stop.
Okay, ma'am.
Okay.
Now you are in slide
So,
2, right, ma'am?
Yeah, slightly.
Okay, ma'am, fine, we can see.
Okay. So now there are so many reasons for these cancerous cells not to be destroyed by drugs as well as other therapeutic agents. So first reason may be the complexity of cancer cells. And the complexity means they are able to mutate and they are able to adapt, leading to a diverse population with varying characteristics such as even in the gene expression, metabolism, and metastatic potential. And these are posing many problems to be destroyed. So they are able to adapt to what are the environments they are having. They are flourishing well with the environment. And second reason may be tumor micro environment. So this is one of the most important reasons. By this, all the cancerous cells will flourish well. So this environment is being created inside the body, and these cancer cells. Hello. Yes, the comprising tumor cells, immune cells, endothelial cells, fibroblasts, cytokines, chemokines. Okay, all these things will provide a very good environment for the growth of cancer cells. Then the next reason may be the immune system regulation. How the immune system is being regulated, and moreover, these cells cannot be able to expose to the immune system. So that's the reason the cancer cells could not be destroyed. So the visibility of the immune system. So if the immune is once the antigen is entering the body, what should happen in our body? So the same antigen must be exposed to the immune system. Then only the immune system can be able to act on it. But at this case, when the cancer cells are exposed to the immune system, the immune system will fail itself to know about the cancer cells, whether they have been injured or not. So that's an important thing. That's the important thing for the failure and the treatment. The next one is immune evasion tactics. So here, cancer cells can deactivate the immune cells by expressing certain surface proteins. So they can express some surface proteins and they alter normal cells to impede the immune response. So examples may be so here, this is how they are preventing the T cells. Main role in the immune system, as far as immune system is concerned, the main role is being produced only by the
Ma'am,
T cells. Yes, ma'am.
sorry for the interruption, ma'am. Again, your slides are not moving. It's still in slide 2 only, ma'am.
Okay, ma'am. Now it's moving.
No, ma'am, it's not moving. It's still in slide 2 only.
Yes, yes, yes. Slightly. Slightly.
Yes,
Can I go,
ma'am.
ma'am? Yes,
Yes,
please.
ma'am.
So
Now,
now I'm explaining what are the reasons for the cancerous cells not to be destroyed. So one of the main reasons is complexity of cancer cells. Cancer cells are more complex in nature. They are able to mutate and adapt to a particular environment with varying characteristics, such as gene expression, metabolism, metastatic potential, and these are presenting a very difficult task for the destroyment of the cells. And next thing is tumor microenvironment. So it comprises all tumor cells, immune cells, endothelial cells, fibroblasts, cytokines, and chemokines. And these things are posing a very good environment for the growth of tumor cells. At the same time, the tumor cells may not be, next thing is the tumor cells may not be exposed to the immune system. That is one of the reasons we are not able to destroy the cancerous cells. So next thing is immune system regulation. For everybody there is an immune system. When an antigen enters the body, our immune system will try to eliminate the foreign antigen. So here, what is happening in cancerous atmosphere, that is the cancerous environment, these immune cells may not be exposed to the immune system. So that's why the immune system fails to recruit the innate as well as adaptive immunity towards the cancerous growth. And that's why these cancerous cells will spread to the other parts of the body. So next reason is immune evasion tactics. So cancerous cells are able to deactivate the cells by expressing some surface proteins. So these surface proteins will safeguard themselves in exposing to the immune system. Slides are changing, ma'am?
No, ma'am, it's still in slide number 3 only, ma'am.
Yes, slide 3. I do not know what is the reason, but here it is changing. Let me find out.
Maybe it's network error, I think,
And
ma'am.
maybe. Sorry for the interruption, ma'am.
It's okay, ma'am, fine. Yes, ma'am, now we can see. The slides are changing now.
Yes, ma'am. Thank you so much, ma'am. Sorry for the interruption.
Sure,
So
ma'am, fine.
next reason may be immune system regulation. So the visibility of the cells may be reduced to the immune system. So that's why it's not being exposed to the immune system. Last reason is maybe immune evasion tactics. Okay, this I told you in the previous slide itself. And the immune system will itself will be deactivated by the cancerous cells. That's why these cells are not exposed to the. And then the cancerous cells, they are able to manipulate the pathway also. So sting pathway means where the T cells and other cells are able to operate for producing an immunity. So these pathways are desensitized by the cancerous cells, and it is able to help the cancerous cells to grow very well. So the next reason, heterogeneity of tumors. So we have got lot of tumors. The tumors may be different types. Between different tumors, we can see a lot of there's a possibility to develop well among themselves. So this would also affect the treatment strategies of the cancer. So now let me tell you about immune system role in cancer defense. How this immune system is playing a role in defending a cancer. So this immune system will detect and target cells that would become cancerous. So we can find out the cancer cells by means of this immune system. For example, tumor infiltrating. This is one of the example I am highlighting. TILs, that is tumor infiltrating lymphocytes. These cells, the presence of these cells indicates that the immune system is responding to the tumor or not. So now, the important topic that is immunotherapy in cancer. A lot of therapies are available. Even then, the cancer is not coming to an end. So that's why the researchers are working in various fields of this area. So it's a biological therapy. This immunotherapy is a biological therapy. The biological therapy is one of the most important therapy by which the side effects could be reduced. So it works with the body's natural defenses. And see, now the cancer treatment is becoming very difficult task to be eliminated. So our immune system must mount an immune response against the cancer, otherwise it cannot be destroyed. So this is here in this biological therapy, the substances made by the body or in the laboratory, either to stimulate or enhance, suppress or desensitize the immune system and help the body to fight it. So what is the main role of this immunotherapy or the biological agent produced inside the body? It enhances the immune system's ability to combat cancer by training it to recognize and eliminate the cancer cell. Here, the recognition of cancer cell it itself becomes a difficult task. So the recognition of the cancer cells by the immune system is necessary. So that's being that's being given by the body what is it, the biological therapy. So it stimulates the production of immune cells specialized in targeting cancerous growth. So when the immune cells are produced against cancer cell, definitely we can stop the growth. So that's why biological therapy is going to play a major role in treating the cancer. So this biological therapy can be used separate or in combination with chemotherapy. You know, you all might be knowing about chemotherapy, radiation therapy, and other cancer treatments. These are the existing treatments available for the cancer. If it is in the previous stage, like stage 1 and stage 2 are curable, but whereas if it is in metastatic, then it is very difficult to cure the cancer. So that's why these biological therapies can be combined with chemotherapy, radiation, and other cancer treatments. So, what are the types of therapy available? Immune checkpoint inhibitors. Immune checkpoint, what is the role of immune checkpoints in this cancer? Actually, these immune checkpoints will play a major role in exposing the immunity. So here we can make use of immune checkpoint inhibitor. So here they are the drugs, okay, which can prevent the cancerous cell to interact with the T cells. T cells are going to play a major role in elimination of these cancerous cells. So here the inhibit, ma'am, hello, excuse me, ma'am. Can you hear me? Ma'am, my slides are changing. Hello.
Yes, ma'am. Yes, ma'am.
Can I move on?
Yes, ma'am.
Yes. So these are some types of, the biological therapy. So here we can have the immune checkpoint inhibitor. So immune checkpoint inhibitors are playing a major role. Otherwise, the cancer cell will be able to bind to T cells. So in that case, what will happen? Cancer cells will be able to grow very well in that environment. So we need to check. We need to give a checkpoint on this point. So what is the checkpoint we may give? We may give the drugs that are the checkpoint inhibitors. So they will prevent the binding of cancer cells to the T cell. So now the T cells will be able to act very well and they will be allowing the immune system to respond to the cancer cell. Second one is T cell transfer therapy. So this T cell transfer therapy is also called adaptive cell therapy, adaptive immunotherapy, or immune cell therapy. So this is going to play a major role. It boosts the natural ability of a T cells to fight against cancer. The same thing only. What I told you in the previous point is also repeated over here. Main role is only played by T cell. So our aim is to expose the T cell and our aim is to not to interrupt the T cell. So these then the T cells will be able to fight against the cancer by selecting and modifying the most active immune cells in the lab. And these are some of the examples given here. Next, monoclonal antibody. Monoclonal antibodies are also called therapeutic antibodies. These therapeutic antibodies are also playing a major role. And this is the lab-created immune system protein and that bind to specific targets on the cancer cell. Example is also given here. And next thing is vaccine, treatment with vaccine. Our cancer vaccines are also available. They are also able to boost up the immune system response to cancer cells. And these are some of the examples, BCG and Sipuleucel is also given. Next thing is immune system modulator. Immune system modulators are also playing a major role. They enhance the body's immune response against cancer using various like cytokines, immunomodulatory drugs, and other things. So now see the treatment is mainly based on the type of cancer available. So these are some of the cancer like Hodgkin lymphoma, brain cancer, lung cancer, head and neck cancer, breast cancer, bladder cancer, cervical cancer, kidney, melanoma, colorectal cancer, leukemia, esophageal cancer. So these are the various cancerous growth available, but the treatment options are only limited. Anyhow, if it is combined with the biological therapy, it would be a great remedy for the cancer. So, and now let me talk about the gut bacteria that boost therapy or response to cancer immunotherapy. So, there is a strong relationship between this gut bacteria and the brain axis. So, here I have given some research, okay, I have referred some research papers, and the research suggests that gut bacteria population make up the potential disturbances links to cancer occurrence and development. So there is a direct link between the gut population and the cancerous growth. So that's why the Human Microbiome Project, HMP, took on the project of sequencing human genome microbiota. So human genome microbiota is playing also one of the important role. Microbiota that normally inhabit the skin, mouth, nose, and digestive tract and vagina. So these microbiota have been analyzed. It is being estimated that 500 to 1,000 species of bacteria are found in human gut. The majority of the species are Bacillota, Bacteria iota. So archaea and fungi are also prevalent in this human gut region. So this slide is showing the intestinal microflora. So how much amount of intestinal microflora are present in the gut region? Actually, the total number is also given 10 raised to 14 microorganisms. So that is more than 500 species are found in the intestinal microflora. That is starting from this intestine, the gut, the small intestine, and okay. So everything is given. Lactobacilli is also predominating, and Streptococcus, Lactobacilli, other species are also able to predominate in this region. In the stomach region, the count may be 10 raised to 2 to 10 raised to 3. In duodenum region, it may be more than 10 raised to 4 to 5. In jejunum, ileum, it may be 10 raised to 3 to 10 raised to 7. Colon and appendix, 10 raised to 9 to 10 raised to 12. So these are the flora of the intestine, the gut. So here, what is given in this slide? The slide is indicating that a healthy gut has a power to prevent cancer. So we need to have a balanced diet, and also we need to maintain the bacteria in the gut region. So that's why many cancer growth can be stopped by having a balanced diet. So here, there is a bidirectional communication between the gut microbiota and the brain. So, as I told you earlier, there is a bidirectional communication between the gut microbiota and the brain. So, we must be specific about the gut microbiota. Why? Because it encompasses the central nervous system, the neuroendocrine system, the neuroimmune system, the hypothalamic pituitary adrenal axis, the autonomic nervous system, entering nervous system, the vagus nerve, the gut. So, the gut microbiota is playing a major role in maintaining all these things. So here the functions of gut microbiota is also given here. Interacting with the intestinal cells, food digestion, inhibiting harmful bacteria, immune system development regulation, pathogen protection, energy allocation, strengthening gut barriers. So these are some of the roles played by gut microbiome. So we should be very specific about this gut microbiome. And nowadays we are eating lot of junk foods that will alter the gut microbiome also. So that is the main point I wanted to insist on this aspect. So now, see, I'm a microbiologist, so let me concentrate on this bacteria-based immunotherapy. You know, bacteria may also be used in this immunotherapy, and directly or indirectly, it is going to help the other treatment options. So it's a promising and emerging field in the cancer treatment. William B. Coley is a scientist, okay? He produced a trailblazer in bacteria-based cancer immunotherapy in 19th century itself. And he used Collins toxins. Actually, the toxins have been separated from Streptococcus pyogenes injection in conjunction with Serratia marcin. So both these things have been used upon sarcomas and other malignancies in tumor patients. And he was able to see the tumor ablation. The tumor has been reduced by using this infiltrate. So see, there is a triangle relationship between the bacteria, tumor, and the immune system. So when this relationship is good, then we can, when this relationship is being good, we can reduce the cancerous growth. So the mechanism, how it is going to work, I'm going to tell you now. Bacteria, this is acting as a natural immune antigen. Actually, this bacteria is going to colonize the tumors, and they will multiply in the tumors. They have tumor-lysing effects, and they will also stimulate the immune system to kill tumors by activating immune cells. So here also I want to insist a point that we have to activate the immune system by any means. So here, as far as the bacteria is concerned, it is going to activate the immune system, and the immune system will act on the cancer cell. Oh, so, and it could also be used in combination therapy like photodynamic PDT, that is photothermal therapy, radiation therapy, chemotherapy, and so immune checkpoint inhibitors or oncolytic viruses. Okay, so with all these things, we can make use of this bacteria also. So now, the role of bacteria in certain cancer treatment is also given. Activation of immune cells. So from the beginning, I'm insisting only one point, that the immune system must be activated. Okay. So that is also played by this particular bacteria. And next thing is immunoadjuvants. Okay. These adjuvants can enhance the overall immune response against cancer and limiting immune checkpoint inhibitor toxicity. So this will modulate the effects of checkpoint inhibitors and improving the safety. And next thing is natural immune antigen. So these antigens will stimulate the immune system to attack tumors. Target immune checkpoints. So it removes the tumor-induced suppression. So when tumors are produced, there will be suppression of immune system that will be removed by this target immune checkpoint. And the enzyme production by cancer cells. So here, sometimes the cancer cells will also be able to produce many enzymes. Okay. So if it is being affected, we can stop the growth of cancer cells. Next thing is bacteria within tumor cells. The immune system ability to detect the bacteria inside the tumor cells can be leveraged to provoke an immune reaction against the tumor cell. So next thing is tumor microenvironment. So this tumor microenvironment, as I told you in the first slide itself, this will affect the presence of blood vessels, T cells, and myeloid cells, which in turn influences the tumor growth and immune suppression. So what I want to tell you is, when we have a thorough idea on these aspects, we are able to stop the growth of cancer at the earlier stage itself. So by these ways, the bacteria is going to help the immune system to provoke an immune response against the cancer cell. So next, we are going to see what are the types are available under this heading. Live unnamed bacteria. Here, some of the bacteria can be able to colonize or proliferate inside the tumor tissues. So, inside interacting with the host to exert an anti-tumor effect. So, they are able to colonize the cancerous cell. So, some examples are given here: Salmonella species, E. coli, and Acinetobacter multifilia, Uropathogenic E. coli, Helicobacter hepaticus, Clostridium, and Butyrium vera. So how this bacteria is going to work in the cancerous cells, I'm going to tell you now. So bacterial administration. So when you are administering a bacteria through intravenous or subcutaneous or intramural routes, it is being, after the injection, what will happen? It is being spread out inside the tumors and other host tissue, normal host tissues also. So when the bacteria present in the normal tissues declines, how it is being declined? Because of the presence of oxygen levels, okay, and immune clearance. Okay, so now next the bacteria is migrate to the tumor hypoxic core. When the oxygen level is reduced in the normal cell, it moves on to the tumor, the tumor area, which is being propelled by the chemotactic response. Then now the bacteria will be able to grow very well, and the multiplication of the bacteria will be there at this case. So now the immune system, the bacteria will triggers the immune system, which will fight against the cancer. So what are the bacterial? These are the bacterial components being used up in the cancer. The surface structure of bacteria can also activate the immune system. For example, nucleic acids, DNA, RNA, cell wall components, peptidoglycan, lipotecoic acid, membrane components like monophospholate lipid A, LPS, lipopolysaccharides, proteins like flagellin, polysaccharides, beta-glucan, peptides like muramyl peptides, bacterial structures like GOS, BGS, small molecules like bacterial cyclic dinucleotides would be at the best bacterial components fighting against the cancer. Next, flagellin is also one of the important. Flagellin consists of flagellin. It activates toll-like receptor 5 mediated innate immune response. Next, flagellin is also one of the thing which is able to recognize the receptor, specifically with the germ-line encoded pattern, includes T helper cell responses and suppress threats. So this is how this is able to stop the tumor growth or the cancer growth. So next, Salmonella tracheomeриum is also one of the bacterium which expresses a flagellin. This is a flagellated bacterium, so it expresses a flagellin B, and this can also prevent the cancer. Monophosphorylate lipid A, this also eradicates the toxic pro-inflammatory effects and maintains sufficient immunogenicity. Next, beta-glucan and miramyl peptides, these are some of the examples given here. It regulates the receptors to stimulate the immune cells. And LPS is also given. What I told you in the previous slide is expanded here. It increases the effectiveness of adaptive CD8 cell, T cell transferency activations, and weaken the immune suppressive immunity. And bacterial growth, the empty envelopes of gram-negative bacteria. This is also going to play a major role towards the cancerous cell. It is more immunostimulatory than LPS. Compared to the LPS lipopolysaccharide, this is more immunostimulatory, and it induces the immune cells to produce long-term anti-tumor memory. See, when you take immune system, the immunity is basically depending upon only the memory cells. The memory cells should memorize the antigen, then only they are able to respond to the antigen. So here it is the case, the bacterial growth is giving the memory effect of the particular immune system. So the cells may be aware of the immune cells like T cells, may be aware of the cancerous growth, and it can proceed afterwards. So bacterial cyclic dinucleotides, okay, they amplify the immune effects in tumor regions by triggering STING pathway. So I gave you in the first slide itself, I told you about the STING pathway. STING pathway, through this pathway only the immune system is going to fight against the cancer. So when the cancerous cell is able to modify this pathway, there is a problem starting. So this modification can be modified by this bacterial cyclic dinucleotide. And other bacterial derivatives, metabolites can modulate the immune system response. We have various cellular and molecular pathways, example cytokines, monophosphorylate lipid, bacterial toxin, the transforming growth factor. These are some of the factors which can modulate the immune response. And so monophosphorylate lipid, this is the first bacterial immune adjuvant approved by human use, for human use. This is the first bacterial immune adjuvant approved for human use. Next thing is bacterial toxin. It activates anti-tumor adaptive immune response. Next thing is transforming growth factor, beta-1. This promotes the secretion of interferon gamma. It enables B16F10 tumor growth. And the example is also given here. So this tumor is being stopped by transforming growth factor, beta-1. Bacterial membrane vesicles, BME is also playing a major role. It increases the antigen absorption and penetration. It activates the innate immunity. So, innate immunity is one of the immunity which is most important in treating the cancer cell or stopping the cancer cell. And so the next thing is pathological antigens. Okay, contain BME. So that is the previous thing I told you. So bacterial membrane vesicles, BME. So BME is also playing a major role. The example is also given here. Pseudomonas aerogenes are derived BME induced innate and adaptive immune response. So here the innate and adaptive immune response may be induced by means of these targets. Next, when we move on to bacterial vaccine, a lot of bacterial vaccines are available. The use of bacterial antigens to elicit an immune response against cancer-specific antigen. So here, the bacterial antigens are able to mount an immune response. So the mode of the route may be the oral, intravenous, or intratumoral, or intraperitoneal, or subcutaneous, or intradermal. So by these routes, we can inject these bacterial vaccines. So some of the examples of bacterial vaccines are given here. BCG vaccine, Bacillus calmette-guerin, Listeria-based vaccines are available, Salmonella-based vaccines are also available, Corynebacterium diphtheria toxin-based vaccines are also available. So this is BCG is a significant player in the field of bacteria-based immunotherapy. So this is an example given here. The case of bladder cancer is taken, and the example is also given here. So if the cancer is non-muscle invasive, just try to remember this point. If it is a non-muscle invasive carcinoma, and if it is not in the metastatic stage, we can go ahead with this treatment. So here the pre-treatment with surgery. So here intraurethral resection of bladder tumor. So here in this case, to remove the visible cancer from the bladder, we are making use of this pre-treatment surgery. And next thing is preparation for BCG treatment. How we are preparing this BCG? This BCG is a liquid drug from weakened microbacterium related to TB causing bacteria. And here we should see that if there is any abnormal reactions when it is being injected inside the host immune system. So that is we always we should be aware of all these things because we are administering a foreign agent. So we are in need of reducing a cancer. So at that time we must be taking care of not to cause any side effects of the administering bacteria. So that's why here the point is insisted. We have to ensure the patient is suitable for intravesical immunotherapy. So when the patient is suitable, we can go for administration of this BCG vaccine. This will be inserted through a catheter into the bladder, and it will be deposited. It will be directly. It will enter the bladder through the catheter, and so it can stimulate the immune system to fight against the cancer. So now let us move on to engineered bacteria. So a lot of genetic engineering techniques have been emerged. They are the emerging techniques nowadays we are finding out in the field. So a lot of genetically engineered bacteria are also available. So genetically engineered bacteria would also play a major role in minimizing the toxicity and improve the tumor targeting properties. And they will also show the immune modulation effects and safety. Examples are given here. Escherichia coli, NaCl 1917, Listeria monocytogenes, Clostridium tetani, Lactobacillus aerosolus. So these are some of the engineered bacteria available. So this is an example for transferrin modified bacteria. The previous slide I told you, for this example has been given. Bioengineered cells with the transferrin modified nanothin polyelectrolyte shell to optimize targeting and minimize host immunogenicity. Actually, this is a modified bacteria which is going to play a role. The modification, why we are doing modification? The modification weakens the host immune response to the bacteria. Always you just try to remember when a bacteria is also entering the body, our body will think that this is an antigen and the body will try to mount an immune response. In that case, we have to weaken the bacteria. So this is how the bacteria will be modified. When you weaken the bacteria will become will be modified and it will not produce any adverse side effects. So this is the step to be carried out, which is promoting the treatment of cancer. And it enhances the precision and effectiveness of the bacterial treatment within the complex tumor microenvironment. And what are the advantages here? It gives the specificity and efficacy for potential application in complex tumor environment. So when we combine these things along with the other and chaotic vasculature trapping. So here the bacteria exploit the disorganized blood vessels of tumors to become trapped. So the tumors will be rich in vessels and other things. Okay. So these bacteria exploit the disorganized blood vessels of tumors to become trapped. And next thing is attraction to the tumor microenvironment. So bacteria are drawn to the specific conditions within TME where they replicate preferentially, taking advantage of the environment. So here in this region, the bacteria are able to multiply flourishingly in this area. And regulation of specific and non-specific immune cells. So the immune cells will be modulated at this level and targeted immune components altering their behavior to support anti-tumor activity. All these things are being done to prevent the growth of the tumor. or prevent the growth of the tumor causing cell. And the activated immune cells now at last can be able to act against the cancerous cell. So the same point is also repeated over here, and this is the toxicity. The bacteria have the potential to reduce the toxicity associated with immune checkpoint inhibitor. So when we are moving with this bacteria along with other cancer immunotherapy, the other toxicity will be reduced and the immune system will fight against it. So now we'll see the potential risk and challenges of these things. Hypoxia and tumors. I told you already about the oxygen concentration in the tumor. So the tumor regions have an oxygen concentration of 7 to 28 mmHg compared to 40 to 60 mm in normal tissue. And immunosuppressive environment. It leads to an immunosuppressive tumor microenvironment which restricts the anti-tumor immunity. And next thing is malignancy indicator. This hypoxia is a major feature of malignant tumors. So for biosafety, what is the biosafety concerns? Bacterial toxicity. There is a need to evaluate the bacterial toxicity. Because of bacterial toxicity, it may lead to some other unwanted conditions. So we need to look into this. And individual variability is also most important because the immune system is different for individuals, the different individuals based on the immunity. The persons below poverty lines may work in a different way. The persons will have the balanced diet will work in a different way. So adverse immune responses, like understanding the potential negative effect on the immune system. We have to see a positive effect on the immune system in this case. So positive response only will stimulate the immune system, otherwise it will fail to stimulate the immune system. Then non-uniform production standard, this is also most important, ensuring consistency and quality in the production of bacterial immunity. Consistency is one of the most important criteria which should be bear in mind to fight against the cancerous cell. Additional challenges are also there. Tumor interstitial pressure. This is when the treatment delivery, drug delivery is also one of the most important criteria which we have to look in. And the treatment delivery is also most important, so that should be an additional challenge for us. And nutrition deprivation, it also affects both tumor and normal cells. And inadequate blood perfusion, okay, it leads to poor drug delivery and hypoxia. So all these things could be favorable. Immature lymphatic system. So here the lymphatic system is immature also. It will be very difficult for the drug delivery. And infection associated toxicity, like harmful effects from bacteria-based immunotherapy. So when the bacteria is giving unwanted side effects, this will also affect the toxicity. And the unpredictable behaviors in vivo. So what are the unpredictable behaviors in vivo? The bacteria, we cannot think that the bacteria would move in a positive way. It may also move in a negative way. So this is an unpredictable behavior of the bacteria. Adverse effects, side effects similar to flu symptoms. Sometimes they may also cause side effects, so like flu-like symptoms. And resistance to treatment. See, increasing drug resistance, that is one of the most important thing we have to look at. Since resistance is one of the thing which is preventing most of the diseases to be cured. So nowadays people might have have a look in the corona. When people have been exposed to corona, most of the members, okay, they would have died due to corona. So that is only due to the resistant nature of the virus and the resistant nature of the drugs extended by the organism. So yeah, this resistance to treatment is also should be bear in mind. And tumor microenvironment, this is one of the unique feature of the which can fuel cancer progress and treatment resistance. So what are the advantages of this bacteria and immunotherapy? Bacteria exhibit a high targeting and colonization ability in tumor microenvironment. The multiplication period is very less for the bacteria, so it is able to multiply maybe within even in 20 minutes. Okay. So this multiplication capacity is also being one of the major added advantage of the bacteria. Their structure and metabolism can also activate the immune system for anti-tumor effects. So enhanced immune modification effect. Engineered bacteria can produce therapeutic payloads like cytotoxic agents, cytokine. When these agents are more increased, what will happen? In turn, it will induce the immune system to provoke the action against the cancer. Modification can enhance their immune modulation effect, and the safety will also be improved by engineered bacteria, and the drug delivery is also good for the anticancer. So altogether, they improve the efficacy, safety, and delivery of the cancer treatment. And the combination therapy, when we see the combination therapy, engineered bacteria can be used in combination with other treatments. I told you in the previous slide itself, it can be used along with the radiation therapy, or it can be used along with chemotherapy, or the other therapies available. And so, future directions, what are the future directions in this field? So, further engineering bacteria, see, we have to enhance our understanding and techniques for modifying bacteria for immunotherapy. This when we are modifying the bacteria, it will be a great help to medical field by which we can destroy the tumors, improving safety and efficacy. We have to improve the safety measures, and also we have to improve the efficacy of the particular drug. Personalized treatment. So, individual's microbiome, based on the individual's microbiota and the cancer type, we need to move on. So, these are the future directions where the growing scientists like you can be maybe aware of all these things, and it paves the way to find a new drug for new drug for cancer. Actually, it is a promising field, but the further research is also a mandatory thing for as far as this immunotherapy is concerned. So maybe the younger generation can find out a new thing and can modify many things, can improve the efficacy of the drug, and it will go good with the cancer. It will go good with the cancer therapy. So, happy researching. Now I would like to extend my sincere thanks to the BioLEAGUES team, and I extend my sincere thanks to my management, Mohammed Sadaq Trust, and my college, Mohammed Sadaq College of Arts and Science, and our dean, respected dean, Dr. R. Meghanathan sir, our respected principal, Dr. H. Abhirami madam, for the constant encouragement given towards in all excellency. And I would also like to thank my department colleagues, microbiology department colleagues. Without them, I would not be able to provide all these things to the audience. And I would like to thank the entire faculty members of MS Mohammed Sadaq College of Arts and Science. And I would like to thank all other heads of the departments as well the technicians for providing me this platform to present this topic to the audience. Thank you very much once again. Thank you.
Thank you, ma'am. Thank you so much for your wonderful keynote session. Participants, if you have any doubt, you can raise your hand or put it in chat box. Okay, ma'am. I think no one have any doubt. We'll move on to the next keynote.
Thank you so much, ma'am. Thank you so much for the opportunity given. Thank you, ma'am.
Thank you, ma'am.
Thank you.
Now I request Professor Dr. Renu Bharadwaj to enter the lecture. Ma'am?
Yes, I'm here.
Yes, ma'am. Thank you.
Just give me permission to start my video.
Yes, ma'am. Sure.
No, it's not starting. Okay, you given me permission to share screen here. Okay, good morning, everybody. I am here to share an interesting aspect of clinical microbiology, that is leptospirosis, which is an important emerging zoonosis. I'm Dr. Renu Bharadwaj. I'm currently a consultant microbiologist, and I was professor and head of microbiology, BJ Government Medical College, Pune, and I have retired from that post. Now, what do we mean by zoonosis? A zoonotic disease is a disease or an infection that can be transmitted from vertebrate animals to humans or from humans to vertebrate animals. So it can go both ways. WHO is currently recognizing the importance of these zoonotic diseases and addressing them through a One Health approach. So it's not only leptospirosis, which is an important zoonosis. There are various other bacteria and parasites which can cause infections between humans and animals and humans. So these are being addressed through a One Health approach. Now, what is this One Health approach? One Health recognizes that the health of people is closely connected to the health of animals and our shared environment. Because we live along with animals in the same environment, so our health is definitely related to each other. One Health has become important in recent years due to human population expanding into new geographic areas. We are invading into territories which were originally animal territories. There's a change in climate and deforestation. We've seen even leopards coming into areas which are now occupied by human beings. Movements of people, and animal products has increased for international travel and trade. So we are sending food from one continent to another continent. So if it is not properly decontaminated, probably bacteria and other things could travel with that food from one continent to another continent.
I think there was some sort of a network issue for ma'am. Please wait for 2 or 3 minutes while we have it sorted. Ma'am, is the network issue sorted? Ma'am, if the network is fine, we can
You can
proceed.
carry on. Okay.
Yes, yes, please do.
Okay, I'll just start with the historical background of the disease. The disease was first described in 1812 among Napoleon's troops at the siege of Cairo. Then it was described by Adolf Weil, and it was named as Weil's disease. It is still often known as Weil's disease, leptospirosis, and is named after him. Then it was described in 1915 in Japan, and the organism got its name as Spirochaeta icterohemorrhagica. In India, it was first described by Tucker in 1907. Then there was almost a period of oblivion when most people didn't know about leptospirosis, and the disease was not commonly reported from this country. Then later on, it started getting described much later in different settings. Now, there are two types of settings in which this disease is described. One is the epidemic rural setting. The rural setting is where we commonly come in contact with animals, where people are staying with animals in their houses, where people are working in rice fields. Now, why rice fields? Because in the rice fields, the rats at night often travel through the rice fields and they pass urine in the rice fields. When the people work in the rice fields during the day, they are exposed to this rat urine. So these are two scenarios in the endemic setting where we often get leptospirosis. In the epidemic setting, wherever they are flooding, so this happens more often in the urban area. Whenever they are flooding, specifically after the major Bombay floods we had, we found major outbreaks of leptospirosis in Bombay because the drains all overflowed. The drains had all the discharge of animals in the drains, and these discharges came out in the water. People who were wading through these drains then were exposed to all these animal discharges, and through small cuts and abrasions, the leptospirosis entered into the skin. And we had major outbreaks of leptospirosis during this period. So these are the epidemic prone settings in which you get leptospirosis. Subsequently, leptospirosis has been reported from almost all parts of India. If you see this map, most of it has been the coastal parts of India. So it is coastal belt which is more commonly reported to get leptospirosis. Now, what is leptospirosis caused by? Leptospirosis is caused by an organism known as Leptospira interrogans. It is a thin spiral organism with closely set coils. So it's a spirochete basically. It is differentiated from other spirochetes because it's got hooked ends. It is tightly coiled. It is 6 to 12 microns in length and 0.1 micron in diameter. Each cell has got about 18 or more coils and the ends are pointed and to a distinctive hook. If you look at the classification of spirochetes, it's very different from the classification of all other organisms which you've seen, where there's a genus and a species and it ends at that. It belongs to the order Spirochaetes, family Spirochaetaceae, genus Leptospira. Now, in this, there are two major species, Enterogans, Biflexa. In the Enterogans group, you have all the pathogenic strains, which are now called as serovars. In the Biflexa, you have all the saprophytic strains, which are present in nature and usually do not cause infection. So there are 26 serogroups which are reported so far and more than 200 serovars. These serovars are divided on the basis of the serological or antigenic classification. Now we are shifting to the genotypic type of classification. There are 16 genotypes. Now this is not a commonly used type of classification. Maybe it will in the future. But because each genotype has got pathogenic and saprophytic serovars. So people still prefer to use the serological classification, that is Enterogans and Biflexa, which have got pathogenic and saprophytic serovars. Now, looking at the morphology of the organism is best demonstrated under the dark field microscope rather than a stain. In the dark field microscope, you get a very typical tumbling type of motility. Once you've seen the organism, you will never mistake that motility which you see. It is a tumbling type of motility, and even in the dark field, you will see the ends. You can appreciate the end at the end of the organism. The other stains which can be used to demonstrate the organism is the monochrome stain, the Fontana stain, more commonly used than the Gram stain, and the transmission electron microscope. Now, this is the Fontana stain, which is a more commonly used stain for staining the organism because the organism is so thin, it cannot be appreciated under the light microscope unless you increase the width of the organism. So when you impregnate it with silver, you will get a little thicker organism, and then you can appreciate it as a dark colored structure in a yellowish background. So you get a dark brown structure under a yellowish background, much thinner than the Treponema pallidum, which can be easily seen. So it's a slender spiral organism. You cannot make any difference between the pathogenic and the saprophytic serovar on the morphology itself, and there's no difference in the staining characters in the. So you cannot from a smear make out whether this is a pathogenic or a saprophytic serovar. Now on a monochrome stain, you can see the ends also properly. So this is just being stained by a single stain. You can see the ends being stained and you can see the hooks. Here you can appreciate the hooks properly. All the serovars look just like this and they take up this monochrome stain much better. The Gram stain was not be taken up very well by this organism. Most other bacteria take up the Gram stain. It's very difficult to stain this organism by Gram stain. In fact, we could only stain one particular serovar or Bacteria hemorrhagica by this particular Gram stain. In that case, it takes up the Gram negative stain, but very difficult to stain by the Gram stain. On the electron microscopy, you can really appreciate the hook properly. You see the end of the spirochete, you can appreciate the hook. In some of the serovars, the hook is not so clearly appreciable, but in this particular one, you can appreciate the hook very nicely. Now, how do you grow this particular Leptospira? It is difficult to grow. It requires either albumin or serum before it can grow. It grows at 30 degrees centigrade, not in the incubator. It takes approximately 3 weeks to grow. So it's no use for diagnosis because after 3 weeks, if you are going to tell a patient that you had leptospirosis 3 weeks back, the doctor is not interested and the patient is not interested. So culture rarely takes any role in diagnosis of leptospirosis. But the medium which is required to grow it is either rabbit serum, Fletcher's medium, Korthos medium, and Noguchi's medium. These are medium which have been used. Actually, the simplest medium to use for growing it is normal. You can take tap water, add rabbit serum to it, and the organism will grow. The most widely used medium, because it is difficult to get rabbit serum, is using all components of the required for growth is what is required called as Eringhausen, Macklinghouse, Johnson and Harris medium, or the EMJH medium, which is basically based on oleic acid, albumin medium. Now, it can grow in solid medium. This is just for academic growth because it really does not play any role in diagnosis of the organism. The colonies of the organism are subsurface and you have to see these colonies under a microscope. You cannot see the colonies on the surface of the medium and they look like this, like balls, cotton wool balls, colonies of leptospirosis. Now, if you try to identify it by biochemical reactions, all serovars are oxidase positive. This is the oxidase test and the tube test. All serovars are catalase positive. It grows at 13 degrees centigrade and grows in the presence of 8 azagarine is the only thing which can be used to differentiate the pathogenic and the saprophytic serovars. Otherwise, you cannot differentiate any of the serovars from one serovar from the other by any of the biochemical reactions. So it cannot be used to differentiate it. Sugar fermentation, no sugars are fermented. So again, no use to identify any of the serovars. Egg yolk reaction, they are all lipase positive. So it's a letter showing a lipolytic action on egg yolk. Now looking at the antibiotic susceptibility testing, this is important because all microbes are becoming drug resistant. You see, everybody is talking of antimicrobial resistant as far as microbes are concerned, whether it is gram-negative bacteria, whether it is gram-positive bacteria. Fortunately for us, all spirochetes are still susceptible to most of the commonly used antibiotics, which is the beta-lactam antibiotics, macrolides, tetracyclines, fluoroquinolones, and streptomycins. So we've actually tested them by MICs and MBCs. So there's a slight increase in MIC to penicillin, but no clear resistance was observed in any of the isolates that we ever had. So drug of choice remains penicillin G. People are still a little hesitant to use penicillin G because of anaphylaxis to penicillin. So the drugs which are generally used are doxycycline and ampicillin. Now, how do you get leptospirosis? Either by direct exposure to the infected animals or exposure to an environment contaminated with the urine, blood, or tissue of an infected animal. That is why, but butchers who are dealing with actually blood of infected animals, people who are walking in water which is infected with the urine of infected animals is where you get it. Entries through inflamed or broken skin, maybe even small cuts, the small cuts you have in your skin, broken skin on your of your heels would be enough to let it go through. Intact mucous membrane also can sometimes allow the mucous listing to go through, and conjunctiva. So waterborne infection, even through water, if you drink water from the water fountains with directly into your mouth, the pressure of the water fountains can get the leptospira to enter into your mouth. Infection can also rarely occur through inhalation. Human-to-human transmission is rarely been reported. Invasion of mother to placenta from the mother has been occasionally documented. Now, seroprevalence in animals. Almost all animals have been shown to have leptospirosis. So it's not only rodents. Our original concept was Leptospira icterohemorrhagica generally got through rodents. In fact, when we did a survey in and around Pune, we found it more commonly present in pigs than goats, cattle, dogs, sheep, rodents. So wherever we looked for it, it was there. But rodents were not the only animals which we got it from. Now, once it is entered into the animals or the humans, it enters through the lymphatics, goes into the bloodstream, and then spreads to the tissues. In the tissues, it causes hemorrhages and coagulation defects because of damage to the blood vessels, specifically the small blood vessels, leading to extravasation of cells and hemorrhage. Now, virulence factor of the leptospirosis is the lipopolysaccharide and the outer membrane proteins, hemolysins, sphingomyelinases, and phospholysins. These virulence factors may directly activate hemostasis pathway or alternately induce an autoimmune response. So either it can directly act on the human body or it can act on the human body through an antigen-antibody pathway. So immune complexes generally aggravate severity of lesion and leptospirosis. Leptospirosis always does a biphasic disease. In the first phase of the disease, you will have fever, headache, muscle pain, nausea, conjunctival suffusion, redness of the eyes, skin, mucosal rash. Here, leptospira will be seen in the blood. This is the septicemic phase. The next phase of the disease, or what we call as the immune phase, where this is the classical Weil's disease, you will get jaundice, you might get kidney failure, symptoms of hepatic renal vascular dysfunction. Here, the clinical course is very rapid and the patient is not treated on time and the patient may even succumb to the illness. Complications is multi-organ failure and this has a very high mortality. Leptospira here are seen in the urine. So the comments, what is the clinical presentation really of leptospirosis? It is the greatest mimicker of diseases. It can present to you with something like mild influenza-like disease to multi-organ failure. Now, as our original concept of Weil's disease was, it was jaundice with renal failure. Now, when we really investigated in our OPD, we found that the anecteric or the one without jaundice were more common than the jaundice one with jaundice or the icteric or the Weil's disease form of it. In fact, 62% of our patients present with anecteric or without jaundice or the self-limiting or the mild form of jaundice. So often we may be missing out a patient or leptospirosis if we don't look for it. The anecteric form of leptospirosis can present to you with flu-like illness, can present to you with pneumonia, can present to you with meningitis, can present to you with cardiac involvement with conduction defects, can present as arthritis, thrombocytopenic purpura, so it may even mimic dengue, renal failure, hemorrhages in the eyes, and uveitis. If you look at the demographic of leptospirosis, more common around the monsoon season. So you will get it maximum around July to October, or in the south where the monsoon season is different, you will get it during the monsoon season. So it's more common around the monsoon season where flooding is more common. More common in young males probably because they're more common outside in the wet soil when it's raining. Now, how do you go about a diagnosis of leptospirosis in patients? Because you see the clinical presentation is not very self-explanatory. You will have to suspect it and then go confirm a diagnosis. So diagnosis can be done by bacteriological diagnosis, serological diagnosis, or molecular diagnosis. Diagnosis has to be early and precise for you to really have your patient respond dramatically to antibiotics. So though most of the simple antibiotics respond well, you have to give them well in time before the immune phase responds. Because once the antigen-antibody reaction has taken place, the patient will not respond. So no specific symptoms are seen in the early phase. So you have to suspect it early and then use a combination of clinical, biochemical, and epidemiological parameters to suspect leptospirosis. What are the clinical parameters which you will think of? Any patient reporting with fever, headache, body ache, and any one of the following. Jaundice, oliguria means less urine, cough, hemoptysis, and breathlessness, hemorrhagic tendency. Anyway, there's a bleeding tendency, including the eyes, signs of meningeal irritation. Biochemical parameters which you will be looking for is any three of the following. Urine, protein, blood, and pus in the urine. ESR more than 50 millimeters, neutrophilia, platelets less than 1 lakh per millimeter, urea more than 40 milligrams %, bilirubin more than 2 milligrams %, serum amylase more than 200. Now, these are all compiled into what have been described as the FAINS criteria. Now, these are available on the internet. You need not have a look at this. These were initially described only on the basis of clinical and epidemiological criteria. Later on, a laboratory parameter was added to it. So there's a part A, there's a part B, and there's a part C. And this total scoring has to be 26. And each of these have been scored. The scoring has to be 26 for you to call it leptospirosis. So this is a scoring system which is also available. And this is fairly accurate. So if you do not have facilities available for diagnosis, the FAINS criteria can be used for diagnosis. Now, what is the sample you would like to collect for diagnosis? Most commonly collected sample is urine, a midstream sample between the 10th to the 30th day of illness. And now, since the bacteria they will die in an acidic urine is always acidic, you would like to alkalinize the urine. So there is a time between the urine being transmitted to the laboratory. The bacteria may die. So a drop of alkali is added to soda bicarbonate added to the urine before transmitting it to the laboratory. Blood in EGTA can also be collected for microscope. For culture, if you want to do it, you have to take 2 tubes for culture media, and for which only 2 drops of blood have to be added. So normally when we do blood for culture for other bacteria, we always want to collect lots of blood so that we can isolate the bacteria. Here we want to dilute it. So we just add 2 drops of blood to 2 tubes of EMGH medium. And then you look for growth by confirming by dark field microscope. DFMA refers to dark field microscope. So bacterial diagnosis is usually by dark field microscope. Now, to be able to comment on a dark field microscopy requires experience. Unless you've seen lot of dark field microscopy, you will not be able to appreciate the motility of leptospirosis. So often fibrils can mimic a leptospira, and you might be able to give a false positive report. So a lot of interpretive, there may be a problem in interpretation. Isolation and culture media is got a very low sensitivity. It takes a very long time. I told you it takes 3 weeks. You cannot wait 3 weeks to treat a patient because it will be too late to treat that particular patient. So culture plays very little role in diagnosis. So basically the backbone of diagnosis for leptospirosis is serology. Now there are 2 types of serological tests, the genus specific test, leptospira, and the serovar specific tests. The genus-specific tests are the ones which are predominantly used is the ELISA, or the enzyme-linked immunosorbent assay. You have the microscopic agglutination test, you have an indirect fluorescent antibody test, you have the indirect hemagglutination test, you have a microcapsule agglutination test, you have a latex test, and you have a microscopic agglutination test, which is the gold standard. So as far as serological tests are concerned, the gold standard for diagnosis of serology is the MAT, or the microscopic agglutination test, which can also be serovar specific for you to find out which serovar is the infecting serovar in this particular patient. Now, these are just some of the slides of patients we have seen. This is a fluorescent antibody test, which we have actually taken serum of the patient, taken a slide on which we have put a culture of leptospira, and we have put in a fluorescent antibody, tagged it fluorescent antibody, and looked under the fluorescent microscope. These are leptospira, indicating that the patient's serum had antibodies to leptospira. This was a rapid test, a slide agglutination test, where you've taken culture of leptospira, added the patient's serum, just rotated it on a on a slide rotator, and we have seen clumping, which indicated we gave us the result in minutes to tell us that there was clumping, that the patient had leptospirosis. So this gave us a rapid test. At that time, there were a lot of epidemics of leptospirosis occurring, and many rapid tests came in the market. This was one of them, the lepto dry test, dry dot, where you just had to add a dropout serum to this, and you got this clumping to indicate that this was leptospirosis. Then there was another lepto dipstick test, where you have to get both these things positive for you to call it leptospirosis. Any one this thing like this was a false positive. So this detects IgM antibodies. Now, you know, even IgM antibodies take some time to develop. They do not develop on day one. So they take about 4 to 5 days to develop. So in the first 3 to 4 days, you may not get a lepto dipstick positive, and the results come in about 3 hours. But this is suitable for the peripheral level, where we may not have ELISA readers and we may not have other things also available. Currently, in most cities, the test which is used is ELISA IgM test, again, which may not be positive in the first 3-4 days. The antigen used in the ELISA IgM is a genus-specific antigen, so you will not know which serovar is infecting this particular patient, which is actually not important as far as treatment point of view, but which is important for epidemiological point of view to find out which is the animal which is infected in this particular area, which needs to be treated also. So you can it differentiates between current and past infection. So it is not that the patient may have had leptospirosis in the past and ELISA would still be positive. Though when we went about it and we monitored it over the years, the ELISA IgM positive stays positive for at least 6 to 9 months. So even an infection which has occurred in the last 6 months would still give you an ELISA IgM positive. Now this is a problematic specifically in rice growing areas. Areas like Sindhudurg get an outbreak of leptospirosis every monsoon season. Every monsoon season, there's an outbreak of leptospirosis, and sometimes you have to differentiate between the current infection and the past infection. Now, this is the gold standard microscopic agglutination test. This is again done under the dark ground microscopy, where you look for clumps of the leptospira forming, where you saw the single ones when they look, the way they look like under the dark ground microscopy. When you add the patient's serum, they form large clumps like this. So this can be done for various serovars, so you can find out which serovar it is and which animal is infected with it. So this is the gold standard as far as the diagnosis of leptospirosis is concerned. We have our national laboratory at Andaman, and this is the test which they will be doing. We send a referral there. Now, molecular tests are also available against leptospirosis. You can do a polymerase chain reaction, the PCR test, which is expensive, little complicated unless you have a setup which is already doing PCR for leptospirosis and you have nucleic acid probes available. For those who are interested in doing the PCR, these are the genus-specific primers which you will pick up the 23S RNA and the pathogenic leptospira targeting the 16S RNA. The saprophytic primer is targeting the ISSR. Now, this is showing you a PCR targeting the 331 base pair. You can see the 331 base pair. This is coming for all the serovars. Again, this will not differentiate which serovar it is. It will just give you a rapid diagnosis, which will be positive even before the serology comes positive. So the advantage of molecular is it will come positive even before the IgM test is positive. You also have real-time kits which have come up now for PCR. You have those which are Tacman-based and CyberGreen chemistry-based, which target for the LELIP32, and these are also those who have the smart cycler can also use this, or various other real-time PCR kits are now available in the market. Now, what is there available to you for prophylaxis and therapy, rodent control measures, and hygiene? Specifically, those who are working in farms and fields now must make sure they wear protective clothing, high gumboots. Most of our ladies who go into the farms are often not wearing slippers, so they are exposed to all these discharges of animals. And those specifically who are working with high-risk population must try and wear gumboots, so they are not exposed to animals. Vaccination of animals is available. Vaccination for humans is still not available, where we can vaccinate those humans who are working in high-risk population. In fact, in areas like Sindhudurg, where they have this every year they have these outbreaks. Currently, the public health system is distributing doxycycline to everybody just before the onset of the leptospira season, because we do not have a vaccine available to us, which is effective. The treatment which we have available for leptospirosis is penicillin G, as I told you earlier also. Penicillin is the most effective. If the patient is really serious, the drug of choice is penicillin, then doxycycline and ampicillin. The need of the hour is to develop a user-friendly, specific and sensitive test for the diagnosis of leptospirosis to accurately estimate the disease burden globally and devise better methods for prevention of the disease. We do not yet have a good vaccine either for the animals or for humans, and to search for a broadly reactive antigen in the leptospira, which covers all the serovars, which can be used for vaccine and which we can use for humans and animals, because the incidence of the disease is increasing day by day as we come more and more in contact with animals. So leptospirosis is common wherever knowledgeable physicians, epidemiologists, and skilled laboratory personnel exist. So if you look for it, leptospirosis is there. If you do not look for it, you will just label the disease as PU or pyrexia of unknown origin and leave it like that, and you will not have a diagnosis with you. So look for it is all the message of my today's talk is. Thank you. Any questions? Anything you'd like to share from your side also, I would like to know.
Thank you, ma'am. Thank you so much for your wonderful session. Yes, ma'am. Participants asked the question in chat box. I'll read the questions. What is the incidence of leptospirosis in India? With which regularity does it occur in India?
The incidence of leptospirosis varies from region to region. See, in Chennai, it is very, very high. They have almost an incidence of 40% in all the fevers. So if you look at the normal fevers which they get specifically in the monsoons, 40% of the fevers are due to leptospirosis. In Maharashtra, we used to have an incidence of about 10 to 12%. In the north, say if you go to Chandigarh in the dry plains, it is very low. It will be about 4 to 5%. So it depends on which area you are. So some areas it is less. And if you are looking at a rural community, again, the incidence will be higher.
Okay,
in
ma'am.
urban community, it will be lower. But then again, I've seen people from high, well-to-do societies suddenly land up with leptospirosis where they couldn't remember when they came in contact with animals.
Okay, ma'am. Thank you, ma'am. The next question. What is the gold standard rapid test that you recommend for leptospirosis?
There is no gold standard rapid test just now. That's what I want you to work on and develop. The only gold standard test we have is a microscopic agglutination test, which is not rapid by any standards. What most laboratories are using just now is IgM ELISA because that is the simplest to do. But that also will not be positive in the first 5 to 7 days. We do not have an antigen test in the market just now. They came in during those epidemic phases, but they've all gone off the market. There's not a single antigen test in the market just now.
Okay, ma'am. Thank you, ma'am. The next question. What are the virulence factors of pathogenic leptospira?
The virulence factor essentially is the outer membrane protein or the lipopolysaccharide. But other enzymes like the sphingomyelinase is also a virulence factor. Basically, it is an antigen-antibody reaction I told you, which causes coagulation defects.
Thank you, ma'am. Thank you so much. Participants, I think your questions are answered. Again, one more question added, ma'am. Does saprophytic leptospira pass through rat cycle anytime?
It does, but it doesn't cause infection usually. It can pass through.
Okay, okay. That's it, ma'am. Thank you, ma'am. Thank you so much for your wonderful keynote session.
Thank you.
Thank you.
वो है গেছে কি শুরু হয়েছে BioLEAGUES কোথায় লিংকে ঢুকে দেখলাম তখন শুরু হয়েছে
Now we'll move on to the next keynote presentation of the day. I request Dr. Nayana Patel to render the lecture.
Hello everyone.
Yes,
Can you
ma'am.
let me know if you can hear me? Am I audible?
Yes, ma'am, you are audible. We can hear you.
Can you allow me to share my screen?
Yes, ma'am. One sec. Renuma,
Yeah.
could you
Yeah,
please
I'm
stop
just trying to
sharing
get
your
off the
slide?
screen. I'm trying to do that. How to do it?
Okay, okay, ma'am.
How do I get off it? This.
No, that into my. Yes, ma'am. Thank you, ma'am. Nayana, ma'am.
Yes. Give me a second.
Okay, sure.
Are you able to see my screen?
Yes, ma'am, we can see your slide.
And can you let me know if you
Could
can
you please share in full screen mode, ma'am?
Yes. Can you see
Sure,
the
ma'am.
slides
Yes, ma'am.
changing?
Yes. Yeah, ma'am, it's changing. Thank you.
Good afternoon, everyone. Uh, I'm Dr. Naina Patil. I'm Associate Professor in MIT School of Bioengineering, Sciences and Research from ADT University, Pune. Uh, Thank you, first of all, to the organizers for giving me this opportunity. The earlier talks which we were discussing and which we were hearing were mostly related to the biomedical aspects. Today, I'm going to touch upon one more aspect of microbiology, which is environmental bioremediation. Uh, Here I'll first introduce you to the problems associated with plastic biopolymers and the synthetic polymers, and then I would slowly take you towards the next aspect of my presentation, which is how microbes are being utilized, and lastly I would like to give you a glimpse of what we at bioengineering do with respect to plastic bioremediation using synthetic biology and genetically engineered organism. I hope my talk will serve the purpose of this conference. So my topic today, what I'm going to present is how microbes, or how we can harness the powers of microbes for plastic bioremediation. Let me take you to how this is first of all a crisis. Plastic pollution, of course, it's a well-known and a well-acknowledged environmental crisis. Why it is a crisis? Basically, the plastic, why it is our favorite, it is because of the durability, because of the flexibility to use it, but the same durability leads to a very longer persistent of this plastic into the environment. That is the main reason why a useful plastic has become such a big problem. Now, this plastic what we use is of course beneficial at our end, but when it turns into waste, that is the major chunk of the pollution. And this plastic waste is not because plastic can just stay in the environment, but because we are not able to balance it. The volume what we make, or what the plastic production is, and how we manage it is completely imbalanced. If we today also, if we can balance that, the production and the management, plastic waste can be brought under control. Now, if we look at today's situation in 2023, and the current statistics shows that 43% of the plastic what is a waste or which has been mismanaged is going to enter one of the ecospheres. Either it goes into the air, water, or soil, and finally it is going to come back to us through all these routes. So we already know plastic is a problem. We have seen so many different conferences dealing with this, but why I have selected this topic, why we are discussing this topic today, because this year in July 2023, it was the first day which is considered as a global plastic overshoot day. What is this overshoot day? This is the date where the amount of plastic which we produced has gone beyond our managing capacity. That is why this year this plastic topic has become one of the hot topics, and that is why I have selected this topic for my presentation. Now, why have we reached this overshoot? There are multiple reasons. Let me give you a statistical analysis or some figures which will show you how huge the problem is. As you know, plastic is very popular, but how much? What is the scale at which we are producing it? So globally, or at a global level, we produce around 400 million metric tons of plastic every year, and about 10 million tons they finally end up into oceans. Now, why ocean is our favorite place to dump plastic? Because it is far away from us. That is what we assume. But this plastic which ends up in oceans or in water bodies finally convert into microplastics, and they enter into our water bodies, finally coming into drinking water. And a recent survey has shown that this plastic is around 63% tap water in India has this microplastic into it. That is how we are affected by the plastic which we throw into oceans. China, even if it is the largest producing house of plastic, around 32% of the plastic is produced by China. But if we look at the management, United States and India come ahead of China, so that the recycling or the waste management, if you look at the worst three countries who are not able to manage the waste, we are one of them, which is unfortunate. Now, when I say we belong to the top three where we are not able to manage plastic, that is not just because of plastic production, but the number of people we have in our country. But where do we stand with respect to plastic? India basically contributes to around 18% of the global plastic, which is around 275 million metric tons. And this plastic which we produce is of course utilized in several different sectors, and 9.46 million tons of this plastic remains uncollected in India. It is thrown in and around cities, outside the cities, outskirts of the cities. So these are not properly disposed of. And the 99% of the plastic waste which is collected or which is just processed finally ends up into a landfill. Now, landfill, there is a limitation to landfills, how much plastic can be dumped into it. And as I said, plastic is going to remain for a very long time. Just dumping it or putting it into a pit hole is not going to solve the problem. Now, why this plastic stays in the environment for a very long period? Why it is a pollutant basically? For that, I'll have to take you to a bit of chemistry and different types of plastics. So if we look at different types of plastic, we see plastic every day. So all the plastic belongs to at least one of this category. So basically we have PET, which is polyethylene and terephthalate kind of plastics, or we have polyethylene, we have polypropylene kind of plastics, which can be low density, high density, and PS plastics. So basically, if you see the PET plastic, it is bottles, water bottles, what we see lying around. It's a very common sight to have a water bottle with everyone right now. And the PE type of plastics are basically high density and low density plastics. The high density is mostly for the containers, milk containers, or whatever plastic bottles you get in which food is packaged, that is usually high density. But the low density PE is the polythene bags. And PVC, which is one of the category of plastic, is usually used in pipes and more hard kind of coverings. While the PP is mostly into the disposable containers, the food containers which we use, and PS is mostly the foam, the packaging foam. Now, if you look at the distribution, how is the demand and how much is utilized, the main product which is accumulating and which has been a problem is the PE and PP kind of plastic. If you look at the PET bottles, it is hardly 7%, while PE and PP contributes to more than 50%. Now, why this is a problem? Why we are dealing with this plastic waste? Basically, if we go to the chemistry of plastic, we have two types of backbones in plastic. One is a heteroatomic backbone and one is just the carbon backbone. So the PUR or the polyurethane and the PET bottles or the PET kind of plastic has this kind of structure wherein you have different molecules making the backbone. And because there are different molecules, there are so many branchings, it is easy to chop it. You have sites where we can cut this backbone and we can get rid of it. But when we talk about the carbon backbone, we just have a long stretch of carbon molecules. As you can see, there are not a lot of points where we can cut it. The carbon backbone are quite sturdy, and the PE, PP, PS, and the PVC plastic, most all of them have the backbone. And this lack of hydrolyzable groups or bonds is the reason why the PE and PS are the most troublesome plastics. Now, whatever plastic is produced, as I said, India is the one who is not able to manage the plastic. It is on the second rank. But if you look at the percentage of plastic which is recycled by every country, uh, in India, around 60% of the plastic is recycled. as compared to others, this is a better percentage globally. So what do we do with this plastic? We recycle it. How do we recycle? As I said, most of it, around 58% of it, is dumped into landfills. The second option is to incinerate, burn this plastic. And the last one is recycle. If you look at the percentage, hardly 18% of it is recycled. Most of it goes into landfills or they are incinerated. Now this 18% of the recycling is repurposing, basically reusing the plastic, while the closed loop recycling and the upscaling is done using the thermolysis, basically heat treatment and chemical pyrolysis. So when you do heat degradation or chemical degradation, naturally we are going to lead to oxidation hydrolysis kind of reaction, and this is going to need conditions which are very specific, very harsh for the environment. This technique use special equipments which are going to be expensive, and of course it leads to production of byproducts which are secondary pollutants or release of toxins. The other way is allowing it to degrade naturally, but as I showed you in the earlier slide, the backbone of this PE and PS make them resistant to degradation, and that is why they are extremely slow. Now what we have as an option now, we have bioremediation. That is what I'm trying to bring you towards. So bioremediation, how it is better than natural degradation. We have a better speed, we have better control, and we can have a bioremediation process which is going to always give you beneficial effects, not something which will have a secondary pollutant kind of impact. So there are different enzymes. Bioremediation, as everyone already knows, is about using microbes, living cells, plants, fungi, or enzymes which can be used to clean up a pollutant. Now, as we are talking about plastic, the most favorite biomolecule which is used for plastic is enzymes. Now, as I said, we have different types of plastic. So naturally, we have different sets of enzymes which can be used to degrade this plastic. For example, the PET plastic, it is going to be based on the PETase or the PET digesting enzymes and the MHETase enzyme. These two enzymes are well studied, well reported, and extensively used to deal the PET kind of plastic. While this other two, which is PP and PS kind of plastic, there are still struggles going around in this region, in this area. We have not yet completely deciphered the actual pathways which are responsible for PE and PS biodegradation. But we have few candidates, few key candidates which can be used for the PE and PS kind of plastic, which is lacase, magnesium peroxides, dehydrogenase, and oxygenase. While the PS, as they are used as foam forms, a different set of enzymes such as deoxygenases, isomerases, aldolases are the favorite ones when we talk about the PS plastics. Now, where do we use this PETase and MHETase? So basically, we have the PET backbone, which is first digested by PETase to give you two products, which is the ET and MET, and the MET further can be digested by using a second set of enzyme, METase, into TPA and EG. These two byproducts are easily taken up by the bacteria, and they integrate it into the TCA cycle and finally end up as a biomass. So the two players which we have for PET is the PTase and the METase. As I said, these are very popular standardized kind of enzymes, and now we have a cocktail of these enzymes which are easily expressed inside the bacteria. The bacteria are able to secrete these enzymes in combinations or as individual enzymes. And when you treat the PET plastic with these enzymes, sequentially PETase and MHE, sorry, hello, should I continue?
Yes, ma'am. That was just some background noise, I think. Please
Sorry.
continue.
Yeah. So as I was saying, we have different bacteria which can express these enzymes. as a dual enzyme together, as a coenzyme, which can be coupled to one another, or as individual enzymes which can chop these backbones in the PET plastic and finally give you EPA and EG, which are the easiest forms of these polymers, which can be assimilated or accumulated inside the bacteria. Now, when we talk about the PE and PS, this is the set of enzymes, and this is the reaction which is followed. First of all, the PE and PS kind of plastics are fragmented. The small polymers are made out of it. These oligopolymers can then be treated by oxidases, alkalases, lactase, esterases, and which can go into the bacteria and assimilate this products as a biomass. This is assimilation is mainly carried out by metabolic pathways such as the citric cycle or the TCA or fat hydrolysis kind of pathways. So these enzymes, when you add artificially, are going to give you biofragments which can be easily converted into biomass. Now, these enzymes, the set of enzymes which I was talking about, can be isolated from all of these organisms. These are the top five um, organisms which are the source of our enzymes. It can be bacteria. We can have different consortiums, combinations of bacteria. Basically, consortiums work better as compared to individual bacteria. Similarly, a combination of fungi and bacteria has also been a popular choice, wherein certain fungal enzymes first make the plastic prone to the digestion using bacterial enzymes in the next set. Also, gastropods, several snails are used which can feed on your foam packaging material, plastics, PET bottles. Larvae are also fed this kind of plastic. So we have the black soldier fly larva, which is very popular choice in degrading polyurethane and the foam polystyrene kind of plastics. While certain arthropods, certain insects are also been employed, wherein their gut has different bacteria or enzymes which can chop this plastic into a simpler forms. Now, as we are talking about microbes today, that is our theme. So I will focus more on the microbial part. The microbes are, even if they're tiny microscopic things, they can provide solution to mega problems such as a plastic pollution. How do they do it? They can colonize on the plastic. They can erode the surface using extracellular enzymes. The colonies itself can hamper the structure of the plastic, as well as there are certain combinations of microorganisms who can, through give and take, can degrade the surfaces and finally break down the major complexes into the plastics. This is a list, and this is a ever growing list, a list of organisms which are the top candidates or the most explored organisms in degradation of different forms of plastics. Now, this is already well known, well standardized method of plastic degradation literature. There is a plethora of literature available. So today, what I'm going to tell you new about this plastic, that is how we deal with plastic in our institute. Our set of research students, some of our project students, they are working on this plastic problem, and we are trying to find out solutions for the plastics which are used regularly. But one niche area which is untouched, that is a novel or maybe a source of plastic which is lying around us but has not been given that much emphasis. That is the sanitary napkins which are used. The sanitary napkin is 90% plastic, and it is never seen as a source of plastic. So today I would like the participants to please acknowledge this fact that plastic is not just the bottles and the plastic bags which are lying around, but this source of plastic which is overlooked or understated is the plastic based sanitary napkins which are used. Now, why it is such a big problem? So if you see here, um, in India, we have millions around. 300 plus million girls and women who experience menstruation, and 36% of them use the plastic-based sanitary napkins. Now, according to the Menstrual Hygiene Alliances in India, around 12 billion of these napkins are generated every year. So, if we look at some of the numbers, so if you see a person can use up to 7 to 8 pads per cycle, which leads to, and if we count the number of people or the women who are using it, that ranges about 121 million women per year, producing or leading to a waste of 1 million pads per month, which adds up to 12 billion pads per year. Now, as I was saying, this source of plastic has never been emphasized upon. I would really appreciate if at least one of the participants take up this as a challenge and try to solve the problem, because hardly it has been paid attention to. The main reason is sanitary waste is considered as a taboo. No one wants to talk about it or avoid talking about it. It makes people uncomfortable. It should not be the case, at least in a scientific community. When we try to solve plastic problem, we cannot ignore this major source of plastic. Now, the sun napkin, as I said, that the number is very huge. That is one problem. But if you allow this sanitary napkin just to be disposed or added into a landfill, it is going to take at least 500 to 700 years to degrade that sanitary napkin. So imagine the millions of sanitary napkins which are added into the landfill, and it is going to stay here for 500 years. That is kind of the scale of pollution what we are looking at. As I said, the sanitary napkins are almost 90% plastic. Now, where does this plastic come from? There are different layers in the sanitary napkin. The top layer, the bottom layer, there are adhesives, the packaging material. All of this contribute to that 90%. Now, the central pollution guidelines to get rid of the sanitary napkin is to deep burial, that is landfills basically, pit disposals, and incinerations. Now, this waste, what we are talking about, is the millions of sanitary napkins. If we look at the numbers, hardly 15% of them goes into incinerators. 25% of them are disposed by burying into landfills, but a major chunk of it is just thrown into dustbins and they are not properly disposed of. 9% of them are flushed into our sewages, thrown into open spaces, which end up into water bodies, or they are just lying around in our dump yards. Now, this is a problem because, as you know, the soiled sanitary napkins, they have blood, and this is a very good breeding site for different blood-borne pathogens, and it is a very high risk for the people who deal with this waste. So not just the plastic, but you are creating different or multiple grounds where pathogens can easily flourish. And the solutions which we have, that is the burning part or the incineration part, it releases carcinogenic pollutants such as dioxins. So if you inhale these kind of pollutants, it is another health risk. So basically, whatever plastic waste we are generating here is either giving us a pathogenic breeding ground, or it is giving us secondary pollutants. So these are not the ways in which the sanitary napkin waste should be dealt with. And surprisingly, when we started this work, there is not even a single eco-friendly alternative or a single green way of getting rid of this sanitary napkin plastic waste. So then we came up with the idea that why not we engineer a bacteria and then try to manage this waste. So what we did, our approach in MIT School of Bioengineering, we use synthetic biology, basically make bacteria which will be able to sense that there are these sanitary napkins lying around. Then the sensing part should switch on certain genes that can be regulated, that can be controlled, and whatever enzymes are made by the sensing and regulation should respond. They should act upon the sanitary napkin. They should engage with sanitary napkin, and finally they should degrade that plastic. So this was our plan of work. I have simplified it a bit because I don't want to go into the technical part of each and every aspect, but I'll try to give you a simpler glimpse of the work which we do it in our institute. So basically the sensing part, we were not sure how to start with this. So what do we have in our hand? How can we trigger engineered bacteria? First of all, we have plastic. As I said, the sanitary napkin has plastic, absorbing material, and blood. Plastic we cannot use because it is a polymer. It won't easily go inside a bacteria and trigger something. So plastic was out of question. Second is, what about which material which is inside the napkin or a part of napkin? So we have two things. We have blood, but as I said, blood is, if we deal with blood, we are dealing with pathogens. Any bacteria which can feed on blood is basically a pathogen. So we cannot deal with pathogens when we are trying to do a bioremediation kind of innovation. Pathogens are out of question. So naturally, blood cannot be a triggering factor. The last option what we have is the absorbing material. Now, most of the sanitary napkin have cellulose as their absorbing material, which is a neutral or not that difficult to use kind of material. So we selected cellulose as our triggering material. So for sensing part, cellulose was our target. Now, once we have cellulose in place, it should switch on certain genes. As I said, we need to regulate what is being expressed in the bacteria. So for regulation, in synthetic biology, we use logic gates. So the logic gate which we used was a OR gate. OR gate, basically, those who are not aware of this logic gates, OR gates are circuits or genetic circuits which can be switched on using two different inputs. So if I have input X and input Y, then I can, if I have input X and input Y, then I can get an output when one of it is present or both of them are present. So I wanted to have a genetic circuit which should switch on irrespective of whether X is absent or Y is absent. So that is why we selected the OR gates for our synthetic engineering of the bacteria. Now, this was how we wanted it to respond. So we have the sanitary napkin, we have cellulose, it should go inside the cell. It should switch on a promoter, a gene basically, and then synthesize enzyme 1, synthesize enzyme 2, which should respond and regulate. That is, it should go outside the bacteria, coat the sanitary napkins, uh, not just the cellulose part, but the whole plastic part also, and synthesize enzymes which will finally deteriorate or degrade the plastic. So that was the whole idea behind synthetic engineering of the organism. Now, as I said, we need enzyme 1 and enzyme 2. What are the candidates I have? As I said, sanitary napkin is basically PE kind of plastic. So we have lacase, we have peroxidase, dehydrogenase, monoxygenase. So right now we are working with lacase and peroxidase. So these two are our candidates which we are screening. So basically what we have done or successfully we were able to do is take cellulose as a triggering material. This cellulose converts into cellulobiose which enter inside the bacteria. It switch on a promoter which is cellulose inducible promoter. This we have taken from a fungi which is trichoderma. The cellulobiose then switches on cellulase enzyme gene and a lacase enzyme gene. So this cellulase, when it goes outside, it stops more of cellulose and generate more of cellulobiose. So basically this is a positive feedback loop. The positive feedback loop makes sure that all the circuits are operating at their full capacity, and the end enzyme what we get is a lacase. Lacase is the one which is going to target are PE backbone, the carbon backbone. Now, as I said, we have an OR gate. So not only cellulose, but we also have one more circuit, which is IPTG, which can switch on this gene. So if the cellulose part is not working, I can add IPTG, which is a inducible chemical, wherein the IPTG will again switch on the circuits, and the cellulase which is made will chop more of cellulose, and we will have a positive feedback loop. So this is my X and Y input, which I was talking about in the OR gate. So I can have cellulose as one input and IPTG as one input. In both the cases, even if only cellulose or only IPTG is present, or when both of them are present, I can switch on my circuits. I can have a better control over the circuits. So this was done in our lab. I won't go into the details of this experiment, but this is the schematic of how we took the promoters, the lactase ORF, how we combined them. These are few gel pictures showing the experimentary details and the fragment sizes of each. And finally, this is what we could get. As if you remember the earlier slides, as I showed, the plastic, if it is the sanitary napkin plastic, if it is lying around in the landfills, it would natural degradation will take around 500 to 700 years. But here you can see that beautifully within a few months we were able to digest the plastic. This is the plastic which we started with, and within 6 months, 8 months, we could get certain abrasions, certain roughness in the plastic. So we are trying to reduce that 500 years to at least a few years, a couple of years. That would still be a good contribution towards managing the plastic waste. In the second panel, you can see that when I use a combination, the earlier when I just showed you lactase, but when I put lactase, dehydrogenase, and multiple enzymes together, you can see there are cracks and there are pores which are formed in the plastic. So this is very promising results. Of course, we have not reduced the time to months, but we have at least brought from 500 years to a couple of years. We are still trying to optimize this process and trying to bring it down to under a year kind of a period. We are looking at a few months of incubations to get rid of this plastic. This is another technical data which shows that the degradation is actually changing the bonds which are present in the plastic, and it is basically breaking the chain, the backbone. Okay. This work has been patented, and we got yeah, in last year we got this patent file, which not only gives you this genetically engineered bacteria, but it also gives the patent also has a design of reactor wherein you can carry out this reaction. As I said, the soil sanitary pads have blood. We cannot just perform this experiment or this degradation in open landfills, but they have to be collected because of the pathogens present in the blood. We cannot do it in open fields. So we have designed a reactor wherein we can add the sanitary napkins, add the bacteria, trigger them, the cellulose will switch on the circuits, and that is how we can degrade it within a few months. So basically, the take-home message for this presentation would be our approach for decontamination or the plastic degradation includes synthetic biology-based engineered bacteria, which was capable of making PE degrading enzymes, and we could degrade or deteriorate the PE-based sanitary napkins. Not exactly within few months, but at least over 1 year. We have brought down that period to around 1 year. So, summary of this topic, or how I would like to conclude this presentation. While we are dealing with plastic, we cannot just use one approach. We have to have multiple or multifaceted approach to deal with this kind of plastic waste. So the easiest way, or the current approach what we are using is the circular economy, basically reuse, recycle, and reduce the plastic waste. Recycling part has to still boost up a bit, because as I showed you the figures, we have already crossed the overshoot point. We have already crossed the point where the production and the management has been imbalanced. So the recycling and reducing part has to be really taken into strong consideration. The second is have plastic-free initiatives, have a strong advocacy for businesses who are committed to reduce or completely eliminate the plastic. Now, when I say completely eliminate the plastic, but if you look around, just right now also, if you look around your table or wherever you're sitting, you're surrounded with plastic. I cannot have a completely plastic-free kind of environment or a day of my life which is plastic-free. Of course, there are several businesses which are cropping up and giving us a plastic-free kind of product. But when I say plastic-free, I also have to promote alternatives. When I remove plastic, what do I replace it with? So, not only just eliminating plastic, but providing alternatives for that plastic, which are biodegradable, which are compostable, which are easy to get rid of, also has to be taken into consideration as a parallel dimension. I cannot just remove plastic completely from the system. I have to replace it with something. That something has to be biocompatible. So parallelly, alternatives have to be searched, which are biomaterials or polymers which are eco-friendly. Of course, to put this into place, that is boosting plastic-free initiatives or promoting alternatives, you should have stringent regulation and policy. There have to be strict rules regarding plastic usage or alternatives that are given to the system. Research and innovation, a small glimpse of which I showed you today. So research and innovation are going to play an important role, not just dealing with the plastic, but also finding novel ways, novel enzymes, for example, the synthetic biology approach, or not just dealing with the plastic which you can see, the plastic or the PET plastic bags, but also the sanitary plastic is one of the major components in the major contributors to this pollution. So such sources also have to be tapped, also has to be dealt with. We cannot just ignore them uh, because of several different reasons. Awareness, of course, spread awareness about the pollution, plastic pollution, how big the problem is, and lastly, educate about the proper disposal and recycling. I guess this conference deals with the awareness and the education part. We are trying to bring in so many different people from different aspects, different subject areas, so that we can raise awareness and educate people about different aspects, not only the plastic pollution, but conferences, talks, symposia, workshops are the ones who would greatly contribute to the awareness and educating the people about the problems which are surrounding us and the possible solutions. So these are few references which I have used for my work and the presentation. I would really like to thank International Conference on Clinical and Applied Microbiology organizers, BioLEAGUES, for giving me this opportunity, EDT University for providing the support, MIT School of Engineering for allowing me to carry out this research and giving me the opportunity to be a part of the conference, and all the participants for patiently listening to me. Thank you so much for the time. This is our MIT University. I would like to invite all the participants as and when possible, or when you are in and around Pune, do visit us. We have beautiful labs. I'm sure we will have a lot to discuss in person. Thank you so much. You can contact me on my mail ID if you have any further queries.
Thank you, ma'am. Thank you so much for the wonderful keynote session. I think participants added questions in chat box. I will read the questions, ma'am, now.
Yes.
Does enzymatic degradation completely degrades the polymer, or it just loses its chemical properties and becomes biodegradable?
Enzymes are not completely going to dissolve the plastic, but they are going to make it a bit less hazardous or less complicated. So there are different steps of degrading plastic. You cannot put an enzyme and plastic in one container and it is going to vanish. Of course, there are steps. from one step to another. It has to be sequentially degraded. So as I showed you the PET example, so PETase is going to first break the backbone, then METase is going to break the secondary products. Finally, they are going to be assimilated into the organism. The bacteria is going to use up as a carbon source. So this is going to be a stepwise process. I hope I was able to answer the question.
The next question is,
Yeah.
how do you plan to use the engineered bacteria to degrade the sanitary napkins in the garbage dump in environment?
Yeah. So when I showed you the patent, I mentioned that this sanitary napkins which are lying around in the landfills cannot be the targets. We have to collect this waste. As I said, you cannot dispose the sanitary napkins just in the garbage yards, because people are going to deal with the plastic. People are going to handle that waste, and because it is soiled, that is used sanitary napkins have blood, and these are going to be the breeding ground for pathogens. So first of all, there has to be strict rules of disposing this waste. You cannot just put it into a dustbin and that dustbin goes into a dump yard and you are done. No, there has to be a segregation of this menstrual waste into a separate system, and that has to finally end up into a reactor. That is what we patented. It has to be done into a reactor in a closed system, wherein you take the sanitary napkins. I didn't show you one more step, which is UV irradiation. So when you put it into the reactor, we UV irradiate it. Irradiation does two things. It gets rid of the pathogen, basically sterilize the pathogens, and secondly, it also makes plastic prone to enzyme degradation. So plastic chains become fragile when you expose them to UV, and then we put the engineered bacteria, which would then step by step it would degrade the plastic. So no, landfills are not our targets. Engineered bacteria on their own also has different problems when you release them in environment. So engineered bacteria are not that great when you talk about spreading them in an environment, but a reactor would serve the purpose.
Thank you, ma'am. The next one. Would such engineered bacteria be effective against microplastics?
Microplastics is the target area which we are dealing with right now. So I have a few students who are working on microplastics. Basically, the problem with microplastic is they are small in size. They have a larger surface area. So
Sorry, ma'am. One second.
yeah.
You can continue now, ma'am.
Yeah.
Sorry.
Microplastic, we will have to use a different kind of approach for that. Right now, I don't have the data for you to show, but maybe within a few months, I will have an answer for this. Right now, I also have a big question mark regarding microplastic.
The next question, ma'am. What are the reasons which underline for ignoring sanitary plastics? Sorry. What are the reasons which underlie for ignoring sanitary plastics, ma'am?
As I said, first of all, sanitary napkins in the waste management itself is not segregated as a different waste. It is just considered as a solid waste. So the rules which are applicable for sanitary waste is solid waste management rules. That is, you just collect them and dump it into the landfills, or you burn it. But as I showed you, the problems with dumping is it would take 500 years to degrade one sanitary napkin, and the incineration or the burning part will get just convert a bigger mass into a smaller mass. It is just going to reduce its size, but it is not going to get rid of it. And when you burn a sanitary napkin, basically the absorbing material is bleached with certain chemicals. It is white in color because it is bleached, and that bleaching agent, when you burn it, it is going to release dioxins, which is a carcinogenic compound. So burning that is going to add to a secondary pollutant. It is going to release a secondary pollutant which was not there in the first place. So you are trying to pollute it more. So that rules have to be very clear, that sanitary waste or the menstrual waste is a completely different story. That is why the policies and the regulations have to come into picture. And the second is, people are not willing to deal with this waste, mostly because they are uncomfortable talking about it, or there are certain taboo associated with this. So my experience when I was working on this project for the last 3 years, we are working on this project. Several people they they are not that keen on listening to this or trying to explore this. That is one major reason. That is the taboo behind it. But it was good to see that when our students they presented this work of synthetic biology in Boston, they got the bronze medal. So out of 300 plus countries participating in a competition of synthetic biology, which is ICCAM synthetic biology competition, our students could get a bronze medal. That is something which is a key problem at a global level. It has to be paid attention to. More has to be invested into these kind of problems.
Yes, ma'am. Thank you, ma'am. I think all the questions are answered.
Thank you, ma'am. Thank you
Okay.
for your
Thank you,
time
ma'am.
and the
Thank you.
coordination.
Yes, ma'am. Thank you so much for your time with us, ma'am. Thank you so much. We'll move on to the next keynote presentation of the day. Now, I request Dr. Rajashekar Reddy Ponnuru to render the lecture. Sir.
Yes, ma'am.
Yes. Thank you, sir.
Is the screen shared, ma'am?
Yes, sir. We can see a slide.
Initially, thank you everyone for providing this platform. And I'm sure that this session will be related to microbiology mostly because I'm a formulation scientist, but I'll try to make sure that it is connected in a microbiological way. So we have developed griseofulvin nanolipid particles, and we have generated it as a topical gel for the treatment of dermatophytosis dermatophytosis. And here we can see the dermatophytosis is a pathogenic disease where it attacks the skin, dermis and epidermis layers, where the pH of the skin will actually get in, going to show an impact on the uh, growth of the microbe. So we can observe that through this pathogenesis in this cycle, what we have shown, we have the negative and positive inducers and inhibition of dermatophytosis. The microbe initially attacks the skin, and it will make sure that it is converting all that atmosphere or the environment suitable for itself. So it will generate certain kind of enzymes by its own and make sure that the skin will be like having soluble protein keratins converted into an alkaline state, and also it will make sure that it is infecting the skin. So coming to the class of the antimicrobial agent, griseofulvin is a heterocyclic benzofuran. So the basic mechanism of action is it prevents the microtubule spindling, and it will not allow the mitotic spindle formation at the metaphase itself, and thereby it will prevent the microbes, fungus to have the cell division happen. So it's going to block the propagation of the cells. So it is a fungistatic agent. So coming to the justification, so why we have selected this present investigation is the climatic conditions in our country are very much suitable for the growth of pathogen griseofulvin, and pathogen which causes dermatophytosis like Tinea pedis and Mycosporum, so which I'll be dealing in the further slides. So here griseofulvin is the agent which we have taken as a fungistatic antibiotic. It is a good antibiotic and already marketed formulations are available. But the major drawback in these formulations available is like poor penetration, and also it shows poor solubility. So we try to work upon this aspect where we are trying to improvise the penetrability, and that was the basic aim and goal of our project. So to overcome the above limitations mentioned, what we have done is we have developed a topical delivery and making sure that maximum localization is addressed, and the affected area is like treated well. So we have generated nanoparticulate lipid carriers, which showed a higher loading capacity. They also show lower water content of the particle suspension. Thereby, they avoid the potential expulsion of the active compounds while in storage. So also we have made sure that solubility is enhanced, thereby the bioavailability is increased. And this was due to the reduction in the particle size, which has enhanced the effective surface area, thereby improvising the bioavailability as well as the therapeutic. So coming to the materials and methods, I'll keep it short as the formulation aspect is to be like confined to be like confidential, but still I'll try to make sure that the major ingredients are explained here. So through the factorial design, we have three major components of the formulation: the tween 80, stearic acid, and other ingredient is griseofulvin. And the basic processing parameter which we were like kept seriously into consideration is the sonication time or the mixing time, you can say. And various ingredients here are between 80, the percentage used is like varying, as well as the stearic acid component is also varied. Keeping the PEG 6000 as a polymer for making it more soluble. And we have done several ratios: 1:2, 1:4, and 1:6. And 1:4 was like was shown to be the best results. And thereby this formulation was like further dealt with the characterization as well. So coming to the basic idea of how we have done this preparation. So the drug, the griseofulvin, the active is like mixed with PEG 6000 as a polymer, and where we have used three different ratios by weight, where 1:2, 1:4, and 1:6. And then we mix it properly, and we have passed it through sieve number 80, and we have placed it in a microwave oven where we have already initially kept it at 900 watts for a period of 10 minutes. And then in the once the molten mass is like obtained, the stearic acid, known amount of predetermined amount of stearic acid is mixed with it, and then it is poured into aqueous phase containing tween 80, and we have stirred it using magnetic stirrer, maintaining the temperature. And next is like the resulting mixture was sonicated at speed of 8,000 rotations per minute for about 40 minutes, and then we have poured it into ice cold water. And then the sonicated dispersion was then centrifuged and then filtered and dried. And this lyophilized product is like further stored for other characterization process. A simple like basic idea of how we have generated. So coming to the gel, once we have got the dry form, we wanted that penetration to be like happening more into the deeper levels of the skin. Thereby we generated a liponanate particles containing gel, where we have used 1.5% carbapol as a gelling agent. Coming to the pharmacological activity, we have used two microbes here. We studied it on Taenia carporis as well as Taenia pedis for a comparative study. And here in pharmacological study in vivo, we have used healthy visitor rats weighing about 180 to 250 grams. And the animals were divided into 4 groups. And the groups, group 1 is like a control, where the blank gel was given without any active. And group 2 standard, where marketed antifungal cream was utilized for the comparison purpose. And then group 3, where test formulation, what we have generated is like given. And that is for the Taenia corporis. And then group 4, where we have used Taenia pedis as the microbe. So I'm skipping this part, like I'll be explaining the results. Right. Coming to the solubility study of griseofulvin was done where we have used various ratios, like I was mentioning earlier, and we have found that 1:4 has given the best solubility. Like we can see, it is like a 0.301 plus or minus 0.014. So compared to 1:2 and 1:6, 1:4 was more promising. So we have further developed all other. We have further went to other investigations taking this ratio as the concept. So coming to the scanning electron microscopy, once we have done the scanning electron microscopy, we have seen that the crystalline nature of the substance has been like very much reduced. That might be the reason for the increased effective surface area, thereby the solubility has been improved. And we can see that particles here like are less crystalline compared to the pure drug. So coming to the basic and important characterizations here, we have taken this lipid nanoparticles where we have conducted the particle size estimation, polydispersibility index, zeta potential, and entrapment efficiency. We have prepared 6 batches, of which batch 4 was more promising because we can see that the polydispersibility index is low for this, where it is like 0.252 plus or minus 0.01. And also we have observed that batch 2 was also very much similar, but the major drawback here was the we have seen that the polydispersibility index was not that good when compared to batch 4. And also the entrapment efficiency for batch 4 was very much better when compared to batch 2. So coming to the in vitro drug release, where we have used France diffusion cell, and the batch 4 was like promising the highest percentage of drug release compared to the other formulations. Like earlier said, that might be due to the improvised solubility and permeability or penetration due to the reduced size of the particles. And then we have compared this with a marketed product where we have taken a cream here. We have compared a product, marketed product with our formulation, and we have seen that our formulation was very much promising when compared to the marketed product. And coming to the release kinetics, we have adopted various drug release kinetic systems, and we have matched with zero order, first order, Higuchi model, and Course-Meyer Pepper's plot. And we have seen that the R-square value was much more for in case of Course-Meyer pass, indicating that diffusion was like an anomalous type, where most polymer kind of really high, and thereby non-frequent diffusion was confirmed. Coming to the stability aspect, whatever formulation we generate, we always try to make sure that the marketability will be high with respect to acceptability, performance, and stability. So keeping that in point, we have exposed that material to various temperatures where we had the room temperature and refrigerated conditions. So we have observed that. Hello, am I audible, ma'am?
Yes, sir, you are audible, but we can't see your slides, sir.
Yeah. I've lost internet connection. I'm just connecting.
Okay, sir,
Pardon me
sure.
for that.
No problem. Okay, sir. Yes, sir, we can see that.
Now we can see that.
Yes.
Right. So when we have went with the dissolution compared with the marketed formulation, also we went for the release profile, and then we gone through the stability aspects. And here in the stability aspect, you can observe that we have exposed that to two different temperatures, one being 25 degrees centigrade designated as room temperature, and one is designated as refrigeration, where we have exposed it to 6 degrees centigrade. And we have visually observed that there was no change, no significant or no change at all when it compared with various temperatures. When the comparison was done at various temperatures, where the milky dispersion was like intact. And the particle size, there was a slight, or we can say like an insignificant change in the particle size when we have exposed it to room temperature after 1 month. A slight increase was frankly was observed there. And when we have the polydispersibility index, whenever we have stored it for 1 month, the polydispersibility index was like raised, and also the zeta potential got low. Sorry. The entrapment efficiency, we have found that there was no much difference in the entrapment efficiency. So coming to the pH, extrudability and spreadability, the pH was observed by a digital pH meter, where it was like found to be 6.52 plus or minus 0.12, making sure we have used triethanolamine as a buffering agent, making sure that the pH is not altered. Thereby, it is like suitable for the proper absorption of the drug into the deeper layers of the skin. So coming to the extrudability, as it was a gel, it was like more promising compared to a cream. The extrudability was good. We have used the agent carbopond here, and that made the preparation go smoothly through the tubes what we have used. Next, coming to the ex vivo permeation release, here we have compared our preparation with the marketed product, and also we have done a conventional gel, where we have not converted it into lipid nanoparticles. Normal gel was utilized. And you've seen that our preparation was showing a better value. We can see that it is 53% when compared to that of the marketed product, which has shown just 31%, 31.2% of like X5O drug permeation. And we can observe that there was like enhanced permeability, and also the penetration was high. And this might be the because of the major reason. This might be because of the major factor that we have reduced the particle size, where we have even shown it through SEM, where crystalline nature of the drug was like transited into an amorphous state, and the reduced particle size might be the reason behind all this penetration enhancement. And coming to the Thermal analysis, like we were like claiming that there was like a decrease in the particle size, and also there is a decrease in the crystallinity. So obviously we have to prove that, and that was through this thermal analysis, where we have exposed the product to DSC, differential scanning calorimetry, where we can observe that the sharp peaks in the topmost picture. We can see that the peak was like starting at 216 degrees and was ending at 221 degrees centigrade. And you can see that the following, when we have it as a formulation as a solid dispersion in a gel form, we can see that the peaks spreads at 89.83 degrees centigrade itself. You can see that there was a downfall in the temperature where that like a sharp peak was observed, proving it to be like more soluble and thereby more increased effective surface area, which might be the reason behind the increase in penetrability of the product. Coming to the rheological aspects, where the flow properties were like studied, the viscosity was studied here, where the nanogel, which was like prepared, was exposed to a shear, and there was an inverse relationship where whenever we like trying to increase the shear, the viscosity was getting. So there was a shear thinning aspect. When the shear rate was increased, the viscosity was like getting reduced. It's a thinning process. So we can say that it is a pseudoplastic nature. And this was due to loss of compact packing. Initially, it is like compact packing, but upon the rigorous shear approach, the disruption of the interdroplet network will be happening, and thereby what happens is the material tends to get together. Coming to the major test, we have ICCAM test, where hens egg was taken and Chorea lantonic membrane was like used for the study, where we have divided the test into three, where the negative control group, where we have taken 0.1 normal sodium chloride, and positive control group, where we have taken 0.1 molar sodium hydroxide, and the test group, where we have taken our product itself. So we have seen before and after exposure. You can see the topmost pictures are prior to the exposure with that agent, and later we can see after exposure. You can see, so in the first set, you can see that the irritation score is very low. No signs of hemorrhage or bleeding or lysis of veins or vasculature is not observed there, and coagulation were also not observed, as in case when you have used sodium chloride. But in case of, in contrast, if you see that in 0.1 molar sodium hydroxide, bleeding, lysis, and coagulation was very much observed. So thereby the irritation score also, you can see that it is 15.23. And when compared to the negative and positive controls, the test group was like as good as the negative control, similar to sodium chloride, where we can see that the irritation score is very low. It's like 0.069. So where you can see that in the test formulation, there was no lysis or hemorrhage happening. Next, coming to the antimicrobial activity, here we can see that the antimicrobial activity was like compared for in case of conventional gel, where nanoparticles were not utilized, and then we have a marketed cream to compare, and we have our best lipid nanoparticle gel. You can see that the lipid nanoparticle gel has shown more zone of inhibition. So we can straight away compare them, and you can observe that the lipid nanogel was like almost double of the conventional gel. So this might be because of the nanonization of the particles, and thereby the size reduction has improvised that effectiveness of that product. And similarly, we have done it on Tinosporum rubrum as well. Here also the same thing has happened. We can observe the similar kind of results here. Only thing we have adopted 2 microbes is to confirm that through cup plate method, we are like successful in generating a product which is like more useful when compared to the marketed cream. So we just took for reference that 2 microbes were taken. No specific other reason was there. So coming to the histology, here you can observe that histopathological images where you have a control, standard, and test compared here. So here E denotes the epidermis, D denotes the dermis, and the arrow is indicating the collagenolysis. And the red color indicates the blood vessels there, and blue arrow indicates the hair follicles on the epidermis, and yellow arrow indicates the fibroblast cells. The black arrow indicates the mild infiltration of mononuclear cells. Whatever like pictures are here, we can see that in comparison, we can see that in the dermatitis, whenever the inflammation of the skin is happening, so it was reflecting with the presence of inflammatory cells like neutrophils and lymphocytes. And also we had epidermal hyperplasia, and the sebaceous glands also were found to be destructed. So here you can see that the normal epidermis as well as mild fibroplastic proliferation in dermis was observed when the test population was seen, like which is indicated in red arrows. So you can see that the tinea corporis infection. So the change was compared with the control population, control product. So coming to similar here, you can see the same was done in case of tinea pedis infection as well. The same kind of results we can observe, but what we have seen is that mild epidermal hyperplasia was observed here. And also for tinea pedis, it was more effective when compared to tinea rubrum. So coming to the conclusion of the work, the main objective of work is to develop a solid dispersion and lipid nanoparticulate system of griseofulvin and convert it into a gel using carbopol, and making sure that the nanogel is helpful in the treatment of dermatophytosis. So the preparation was done by microwave irradiation method, where the tedious process is like brought down to simpler and single step of involving all the material and then melting down them and then mixing with other ingredients and straight away getting it converted into a gel. So the morphology study showed a uniform spherical and smooth surface of griseofulvin within the nanoscale region. And this made it suitable for effective topical treatment of dermatophytosis. And there was no significant difference in physical appearance, particle size, zeta potential, polydispersibility index, and drug content under the influence of stability studies, where we have kept the product for a period of 1 month and the product was found to be stable and no significant kind of differences were observed physically as well as chemically. And in vivo studies, it demonstrated the rapid antifungal potential with the better proliferation of fibrous tissue, as the pictures earlier were also showing that the deeper layers were being taking the drug into the deeper layers was possible by this technique. And in addition, the product was found safe and non-toxic, as observed in HEADCAM as well as cytotoxicity studies using MTTSA. We have even studied on lipoperoxidase and glutathione effect as well. It was not completed yet, so we couldn't conclude and put it in the slides. But we have seen positive results when we have seen that glutathione reduction is. So that the developed griseofulvin lipid nanoparticle gel could be a potential candidate as a topical delivery for the treatment of dermatophytosis. So we have applied for a patent and we are waiting for it to be approved. Hope I have generated a kind of microbiological way of putting forward my research, and hope you can hope you have understood. And if you have any doubts or any questions, I'll be more than happy to answer. Thank you, ma'am. Thank you all for providing this particular platform. And thank you all who have organized this event and have taken my session into keynote session as well. So thank you everyone for making it possible. Thank you.
Thank you so much, sir, for taking the time out of your busy schedule to deliver your keynote lecture. Participants, if you have any questions, you can drop it in the chat box, or you can raise your hand so we can promote you to speak and get your question clarified. So we will give them 2 minutes to see if anybody has any questions in the chat box.
Would be thanking Pauline ma'am for coming in contact and also making sure that my presentation is up to this level. So she was continuously in communication. Thank you, ma'am. Thank you for your support.
Thank you so much, sir. It's definitely my pleasure that you have joined this conference. Thank you so much, sir. And, and I see we don't have any further questions, so we will wrap up the keynote presentations. Thank you so much, Dr. Rajshekar Reddy. Sir, thank you so much for your presentation. And some information for the participants. So in about 5 minutes time, we will be starting the technical sessions where you'll be presenting your papers. So please make note, the oral presentations will go on for 10 minutes in total. That is 8 minutes for your presentation and 2 minutes for any questions that the session chair might want to ask you. So for oral presentation, it's 8 minutes for your presentation and 2 minutes for the question and answers. For poster presentation, you'll be given 4 minutes for your presentation and 1 minute for your question and answer discussion. And if you have any questions, you can reach me. I'm sure everybody has my contact number at this point. So you can just call or message me for any queries that you have. And one important notice, you have been allotted particular halls. So if your presentation is in the main hall, please stay here. This is the main hall. And if your presentation is on hall A, it is the same link. We will be making breakout rooms, and you have to just you will be added in that room so that you can just accept the invitation to join that room, and you will be directly moved to that particular room. So please open the agenda, make sure that you are in the right hall. If your presentation says main hall, please stay here, you will not be moved. If your presentation says main hall, I'm sorry, hall A, we will move you directly there. Okay, so in about 5 minutes time, we will start the session. All right. So all the participants whose presentation is in Hall A, we have sent you the request to join that particular room. So if you find any option on your screen that says join this particular breakout room, please accept that. Almost a lot of participants are yet to accept. So please accept the request and join the particular hall. And and for participants who have registered to be as a listener, you are open to visit any hall. We have two halls going on. So in both the halls, presentations will be taking place. So if you feel any particular topic is of interest to you, please you can choose in the breakout room option. You can choose hall A and go there. And if you prefer to listen to the presentations in the main hall, you can be here. Katyani ma'am, can we begin the technical session?
Yes, ma'am.
Okay, thank you, ma'am. Thank you, participants, again for your interest to participate in this conference. We'll begin the technical sessions now. And before that, I would just like to, uh, repeat a couple of things. So when somebody is presenting, please make sure the non-presenters are on mute, and all the participants and presenters, please keep the presentation time in mind. So you will be stopped even if you keep dragging, you will be stopped at 8 minutes for oral presentation and 4 minutes at for the poster presentation presenters. So please make sure that you deliver all the important aspects of your presentation so that you will have enough time to wrap up the presentation. So we'll go according to the order of the agenda. And on that note, I would like to welcome the session chair, Dr. Y. Katyayani ma'am, Assistant Professor, Department of Microbiology, Guntur Medical College, India. Ma'am, thank you for accepting to be the session chair, ma'am. And I welcome all the participants. On that note, I would like to invite the first presenter, Dr. Akhila, for her oral presentation. Dr. Akhila?
Hello.
Okay.
Hello. Am I audible?
Yes, you're audible now.
Okay. Can you see my slides?
I cannot see your slides. I can see the Zoom meeting.
Okay. Wait for a second.
Okay. While you fix it, we will just go to the next presenter and we can come back in favor of time. Muthukumar Vel sir, are you available for your presentation now? Okay, yours is back. Okay. Great. We can see a slide, Dr. Akila.
Okay.
Please begin the presentation.
Okay. Good afternoon. These slides are odd. Hello.
There is a really bad echo, Dr. Akila. I think if you've joined in two devices, please mute the other device other than the one in which you're speaking in. Try to mute the other one, please.
Hello.
Yeah, it's good now. No echo. Please go. Dr. Akila, are you there? Muthukumar Vel sir, are you there?
Yeah, I am available.
Hello.
Yes, sir.
Hello.
Dr. Akila, I think there is some kind of a technical issue and it's getting really delayed. We'll go to the next presenter and can we come back to you?
Okay. No
While we
issues.
fix it and then you can also take
Yeah.
a beat.
Okay.
Yeah?
Okay.
Okay, great. Thank you. Muthukumar Vel sir, you can present your screen, sir.
Shall I share my screen?
Yes, sir. Yes, please.
Yeah. So already screens are shared, I think, Dr. Akila.
Just give me a second. I'll close it. Yes, done, sir. You can present.
Now,
You can share your screen.
is it visible?
Yes, sir.
Good morning to all. So my thrust area is bioremediation. So that basic aim of that work is we have to characterize and evaluate the mosquitocidal bacterium. So in that bacterium, we have to investigate the potentiality of organophosphorus pesticide degradation. So with that bacteria, it has a twin role. We can use it for larvicidal and also pesticide biodegradation. So this is a small introduction regarding that insecticide and biodegradation. As we know that mosquito-borne diseases, malaria, dengue, JE, chikungunya, these are vector-borne diseases, mainly managed by vector control methods. So in that vector control, we are using enormous amount of insecticide, ornochlorine, and ornophosphorus synthetic pyrethroid. So it will harm to the soil, air, water, and the ecosystem. It will also affect the homeostasis ecosystem. So the biological manifestation of that insecticide through the food chain, it will adverse effect which affect non-target organism also. So far, 28 bacterial genera was documented for biodegradation, including Pseudomonas, Bacillus, Alphigens, Flavibacterium, and Methylococcus. apart from that, Pseudomonas serratia have a extensive role in degradation, especially in synthetic peptides. So that we are focusing on Bacillus and Pseudomonas. So this is the way that bioremediation is the more effective and environmental safe cleanup technology for conversion of organic pollutant in the ecosystem. So the main objective of that study is we have to isolate, identify, and evaluate the potential mosquiticidal activity of that bacteria and also degradation activity of the insecticide. So these are the two role which is available in the single bacterium, which is very useful. The second objective is to carry out the molecular characterization of biodegradation activity and the large-cell activity of the particular isolate. So that the hypothesis of that research project is so we can have an isolate which have a twin role of mosquiticidal parallelly biodegradation activity. If we are applying in the field, so it will kill the larvae, mosquito larvae, and also it will degrade the residual insecticide in the field. So this is a scheme of that sample collection. We collected the soil sample nearer to Villupuram district, and also we collected water samples from delta methylene manufacturing industry. So with that basic microbiological technique, plating technique, we isolated pure cultures. Randomly, we selected the colonies. This is a scheme of methodology. We have to isolate the different type of colonies. So with that, using the replica plate method, we are assessing the larvicidal activity by the WHO method. So with that, we are using nutritive medium. We can assess the larvicidal and pubicidal activity. If it is there, then we can parallelly assess the activity of the biodegradation using that M9 minimal medium. We are incorporating with that insecticide, specific insecticide. Now we are using that organophosphorus insecticide. So there is a procedure for degradation efficacy. Determine the efficacy. That is hydrolysis or enzymatic hydrolysis. So that the enzymatic hydrolysis is determined by spectrophotometric assay. So with that assay and the WHO method, we can assess both larvicidal and biodegradation activity. So this is a very basic slide. Will explain how that bacteria metabolize the compound methyl parathion. So this is the main compound which is used in agriculture sector. So it will degrade by the bacterium which cleaves it will yield paranitrophenol. So the paranitrophenol normally in yellow in color. After some days of incubation, it shows the yellow color. So coming to that larvicidal activity, we are using three different strain of mosquito larva: Culex, Anopheles, and Aedes. These are the three different kinds. Culex mainly is a vector for LF, and Anopheles for malaria. Aedes for dengue, JE, chikungunya, vector-borne disease. So this is a very basic thing which will explain how it will use for larvicidal and also in biodegradation. So we come to a conclusion. We arrived at results. There are two different strains which has a different activity. SM6 it has a biodegradation activity. Yes, as a larvicidal activity. And coming to that determination of the organophosphorus degradation, there is a specific enzyme, organophosphorus hydrolase. There is a hydrolase enzyme activity was measured in a different time interval, so that it shows that at 96 hours culture in that minimal medium, it gives maximum degradation of organophosphorus. So this is the kinetic activity assay. So paranitrophenol is used as a standard to calibrate the activity. Enzyme activity, organophosphorus hydrolase activity. So it yield the determination of that enzyme. So this graph shows that there is methyl parathion degradation and paranitrophenol production in different time interval. So that a yellow line, it shows that paranitrophenol concentration, that the rest of the line shows that concentration of methyl parathion. So that the initial stage, that the methyl parathion concentration is slowly decreased and it reaches 120 hours, it shows 1.5 something microgram per ml concentration. Parallelly, the nitrophenol concentration has increased. It reaches 96 hours, its peak, 2.02 microgram per ml per nitrophenol concentration. When coming to the gene, which is essential for the degradation, there are three genes are well documented in Bacillus pumilus: OPD gene and OPD-A and MPD. These are the three main genes. So with that published sequence, we took that pumilus primer, we amplified that specific concerned sequence. So we got a 400 base per amplicon. So with that amplicon, we find out that there is some enzyme, that is DNA translocase. It has a hydrolase activity which play a major role in organophosphorus hydrolase. When coming to that protein profile of that organophosphorus degradation, so these are the different lane. It shows the fourth lane. It shows a minimal salt medium incorporated with methyl parathenium. So along with that, we give 0.1% glucose with that. So that the 0.1% glucose is the induction for that bacterial growth. And automatically, once it grows, it will depends on that insecticide. It leaves the insecticide. It will utilize for its growth. So that the protein profile it clearly indicates that there is a protein will be lying in between 70 to 85 kilodalton. Already there is a report which was confirmed in 2013. And coming to the characterization of that bacterial strain, these are the very basic microbiological techniques: gram staining and spore staining. So it shows that gram-positive rods. And 16S RNA research is closely related to that it's bacillus. When coming to that larvicidal activity, it yields two different proteins, that is 51 kilodalton and 42 kilodalton. So these are the two major binary toxic protein which kill the mosquito larvae. So that binary toxin profile, it shows that there is a strain is Lysine bacillus. There are two strains, that is Bacillus cereus and Lysine. Bacillus cereus, it has a biodegradation activity, and the Lysine bacillus, it has its own toxic against mosquito larvae. So our this is the conclusion. We have a two isolate. One is Bacillus cereus and one is Lysine bacillus. So we have to clone that binary toxin gene in that Bacillus cereus. With that, we can achieve our target. There is a one single isolate which has a twin role. There is a larvicidal activity and also degradation activity. So this is our major key. So right now we are isolating the strain. We characterize everything. We need to clone the things into that Bacillus cereus. So completely that the genetically engineering strain, it has a twin role for that larvicidal and biodegradation activity. So these are the references I come across. Thank you. This is the discussion part. Any question, ma'am?
Nothing, ma'am.
Thank you, ma'am. Thank you, sir, for your presentation. Since there are no more questions, we can go ahead to the next presenter. Dr. Akhila, are you available now?
Yes, ma'am.
Yes. Share your screen, ma'am. Sorry. Dr. Akhila, I think you can share your screen now.
Okay.
Yeah, actually.
Can you see my slide?
Not yet. It says Akhila has started screen sharing. We can see your screen now.
Can see my screen?
Yes.
Start the presentation?
Yes, please do.
Yeah. So, good afternoon, one and all. I'm going to present my research study on gene silencing strategy to combat antibiotic resistance in multi-drug resistant Klebsiella pneumoniae. So the Klebsiella is considered to be the one among the escape pathogen, and which is a major pathogen responsible for hospital-acquired as well as community-acquired infections. And it is one of the pathogen which is commonly seen in the urinary tract infection and which able to cause pneumonia and soft tissue infections. And these are called to be as a primary reservoir of the gas GI tract and which is also in the hands of the hospital person. So the hospital outbreak of this multi-drug resistant Klebsiella, which is majorly caused by extended spectrum beta-lactamase producer. This is considered to be the one of the emerging concern because most of the antibiotics, which from the beginning, which we call it as a magic bullets, which are unable to treat the infection because of its efficacy is got reduced due to the development of antibiotic resistance by this notorious pathogen. So most of the 60 to 80% of the Klebsiella related infections are due to the expression of the biofilm associated gene. So combating this multi-drug Klebsiella pneumoniae as well as the highly virulent Klebsiella pneumoniae is considered to be the major challenge for the clinicians. So now that because of the development of the antibiotic resistance, it leads to the limitation on the therapeutic option, which leads to a new measure for the management of hospital-acquired Klebsiella infection. As we all know, this RNA interference, that is gene silencing technology, which is commonly used to reduce the or to inhibit the gene expression at the mRNA level. So in eukaryotes, this gene silencing strategy, which we call it as a RNA interference, but in prokaryotes, we call it as a cis-trans mechanism, where the cis non-regulatory RNA, which is going to bind at the 5' end of the target mRNA, and because of the base pairing of the mRNA with the cis RNA as well as the trans RNA leads to the inhibition of the gene expression. Finally, the protein expression is going to get inhibited. So we are going to take the advantages of gene silencing technology to inhibit or to reduce the or to induce the sensitivity in drug-resistant Klebsiella species. Based on this literature, the objective of our study was set as to screen the antibiotic-resistant Klebsiella species and its phenotypic expression of the genetic traits, as well as to induce antibiotic sensitivity in drug-resistant Klebsiella species by sequence-specific gene silencing using antisense RNA. We have collected around 70 clinical Klebsiella pneumoniae from the Madras Clinical Machine, and all the isolates were subjected for the phenotypic as well as genotypic characterization. Once the isolates are confirmed as a Klebsiella pneumoniae, further the Klebsiella pneumoniae were used for the screening of antibiotic resistance by minimum inhibitory concentration assay as well as a disk diffusion assay. Then the biofilm forming genes were checked, that the expression were checked by the real-time PCR. And for the objective 2, the work plan is followed like this. We screen the isolate for the by using around 10 antibiotics, and among the based on the antibiotic sensitive assay, we selected the isolate which shoots the resistant to more than 7 antibiotics that were selected for the gene knockdown studies. Then that particular isolate were used for screening of the efflux gene, that is ACR. That ACRA is one of the important gene which is present in the ACRAB efflux system. As we all know, the efflux system is one of the intrinsic resistance mechanism acquired by the bacteria to combat the antibiotics. So we targeted the ACRA gene, which is commonly present in the Enterobacteriaceae family. Then we designed the antisense RNA against the ACRA gene of the efflux system. Then we transformed into the multi-drug resistant isolates of Klebsiella pneumoniae. Further, the efficacy of the antisense RNA was checked by using antibiotic susceptibility testing as well as MIC assay, followed by growth curve and real-time PCR analysis. And these are the primers which we used for synthesizing the siRNA, and these are the primers which we already published and patented to. And this, the designed antisense RNA were outsourced and followed by that gene is amplified by the PCR. Then we use the restriction digestion as well as RD cloning. Then we clone the ACRA gene inside the antisense vector, which is specifically meant for the expression of the antisense RNA, which is tagged with the GFP as well as the minomycin as an antibiotic screening marker. So these are the results of the antibiotic screening. The isolates which are obtained from the urine sample, which shows the maximum resistance to almost all antibiotics, and this then the isolates were used for screening of the ESBL production. We use the CX as well as the CTX biotics for a screening of the ESBL. Then after that, whatever the isolate which shows the resistance by the antibiotic sensitivity assay were used for screening of the antibiotic resistance gene at molecular level. So most of the isolate which shows the resistant and as well as it shows the resistant gene expression in that is like the BLA-SHP, BLA-TEM and the BLA-CTXM. They are the major beta-lactamase targeting genes, and followed by quinolone resistant genes, that is a plasmid codiated reads as well as the chlorophall resistant gene. And this is the representative image of the uh, cell where it shows the amplification of the ACRA gene. And finally, this PCR product is used for the cloning as well as for gene silencing study. And whatever the isolates which show the resistant were subjected for the MIC assay. And since we targeted the ACRA gene, that is one of the gene which is coding for the efflux system of the Enterobacteriaceae. So to study the effect of the and to compare the effect of the antisense RNA with the efflux pump inhibitor, we divided the treatment into 4 groups, where we treated the one group with the wild type Klebsiella pneumoniae, another type with the Klebsiella pneumoniae along with the efflux pump inhibitor, and the third group which consists of antisense treated Klebsiella pneumoniae alone, and the fourth group with the antisense treated Klebsiella pneumoniae plus the inhibitor. And obviously with the efflux pump inhibitor combination with the antisense RNA, which shows the maximum sensitivity compared to the other treatment groups. And these are the representative image of the MIC assay. And this is the image which shows the wild type Klebsiella pneumoniae, which is almost resistant. And one more thing, we use the ciprofloxacin and cotrimoxazole for a further gene silencing study and to see the effect of the ACRA gene of the efflux system, because this ciprofloxacin and cotrimoxazole is one of the major antibiotics where our wild type which is showing the resistance. So to induce the sensitivity, we targeted these two antibiotics. So this is the antisense treated Klebsiella pneumoniae. We can see like around, see in the wild type it is a bacteria which is showing around 6 microgram per ml of in the MIC strips, where in case of the antisense treated Klebsiella pneumoniae, the MIC shows around 0.064, almost around 90% of the sensitivity, inducing the sensitivity in the antisense treated groups. And this is in case of the cotrimoxazole, where the antisense treated Klebsiella pneumoniae, which shows the MIC of 0.125 microgram per ml, where wild type which is almost resistant to this particular antibiotics. Then further to prove this antisense efficacy, the divided groups, these are which already I have explained. These treatments were subjected for the incubation at 37 degree. Then after every 2 hours, we check the OD of the bacterial growth of each group. Then we determine the growth assay, and this is the antisense RNA which is treated with the ciprofloxacin. So here we can see that is almost like around 20 hours of the growth inhibition in case of the bacteria which is treated with the antisense as well as the ciprofloxacin when we compare with the control, and it is when we apply the statistic, it is proved statistically significant too. And in case of the cotrimoxazole, the antisense treated along with the cotrimoxazole antibiotic, which reduced the doubling time till 20 hours of the incubation, and it induced the sensitivity in the treatment group compared to the controls. As well as here one more thing, the scrambled antisense RNA is nothing but it is a random sequence which is not present even in the FLEX system or even in the prokaryotic systems. So this is the gene expression. So we extracted the RNA and converted into a cDNA by reverse transcriptase PCR. Then that cDNA is subjected for real-time PCR to study the ACR gene expression at the molecular level. And this is here although we got the expression of the inhibition of the expression of the ACRA gene in the antisense treated along with the antibiotic, but it was not statistically significant. But in case of the cotrimoxazole, whatever the antisense treated with the antibiotic, which shows the statistical significance compared to the ciprofloxacin treatment groups. So from this, I can conclude that the antisense RNA technology is one of the expedient approach in suppressing the function of the gene, which could be exploited in the control of MDR pathogen, not only in the Klebsiella pneumoniae. And one of the advantages of the targeting the ACRA gene in the ACRA gene of the FLEX system in the Klebsiella pneumoniae is because this ACRA gene, it is highly conserved in the Enterobacteriaceae, and when we worked along with the MDR strain of Escherichia coli, this ACRA gene antisense can be used even to induce the sensitivity in Escherichia coli. So the targeting the gene, that is targeting the gene for antisense RNA, which is conserved to the family level, we can reduce or we can induce the sensitivity of the MDR pathogen against the antibiotic, which is already showing the resistance. So regardless of the current study being an in vitro demonstration, it suggests the use of lower dose of the drug to inhibit the drug-resistant Klebsiella pneumoniae, which is leading to the minimal side effect due to the antibiotic. So the targeting the reflexes-mediated gene with the antisense RNA is considered to be the feasible and novel approach to induce the antibiotic sensitivity in drug-resistant Klebsiella pneumoniae. By targeting this gene, which encodes the efflux system with the antisense RNA, it is possible to restore the antibiotic sensitivity in drug-resistant Klebsiella pneumoniae. So authors are very grateful to the Day1 University for intramural funding, and these are the references which we use for the study. Thank you.
Ma'am, are there any questions for the
How
presenter?
did you check for biofilm production, ma? How did you check for biofilm production?
Dr. Akila, can you hear us? Dr. Akila, can you hear us?
Yes, ma'am.
How did you check for biofilm production?
Sorry, ma'am.
Biofilm production, ma. How did you check?
Yeah, we use the crystal violet testing.
Okay.
Then after that, expression of the biofilm gene by real-time PCR.
Okay. Are there any results that are for Escherichia coli?
Are there any results for?
Escherichia coli. You said you have tried, no?
No, I didn't put in the slide because we didn't publish the result yet.
You didn't? A lot of disturbances. The next participant, ma.
Okay, ma'am. Thank you. Thank you, Dr. Akila. So the next presenter would be Ms. Jyoti Mukherjee. Ms. Jyoti Mukherjee.
Hello. Yeah.
Hi. Yes, please share your screen. After Ms. Jyoti, it will be Ms. Pallavi Rawat. So I request the upcoming participants to have the presentation ready. I invite Ms. Jyoti Mukherjee.
Just a minute, please. Good afternoon, everyone. My name is Jyoti Mukherjee. The title of my study is Functional Characterization of Caginous Cagin, Pigeon pea seeds, acid-base hydrolysis derived peptides. Basically, I'm going to derive a protein hydrolysate from pigeon pea, commonly known as tur dal. Pigeon pea is basically a multi-purpose plant. It can be consumed in the form of dal, green pods as vegetable, and husk as green manure and fodder. If we see, pigeon pea is dominant in terms of protein. Pigeon pea has around 21 to 28% of protein content in it. So for that reason, I'm going to choose pigeon pea for my hydrolysis and make a functionally active and make the active sites of the peptide available for digestion and utilization. Pigeon pea is also referred as poor man's meat supplement. These days, protein hydrolysates from fish collagen, whey protein, pea protein are extensively used in the market in the terms of bodybuilding and various uses. My aim is to make a meal replacement therapy and vegan protein. Pigeon pea is also a repository hub of antioxidant, anti-diabetic, anti-hypertensive, and anti-neurodegenerative properties. So the present study is aiming to characterize a acid-base derived protein hydrolysate from pigeon pea. These are my objectives. The first objective would be the extraction of pigeon pea protein hydrolysate, in which I would go for the oxalate extraction method. Further, I will go for the acid-base hydrolysis, and finally, I will do membrane ultrafiltration. The second objective would be the evaluation of the protein hydrolysate peptides for their physicochemical properties, in which I'll go for proximate analysis, the protein estimation by gel-dal method. Further, we will go for degree of hydrolysis by OPA method. Finally, we'll go for quantitative physicochemical analysis, then FTIR, forming properties, emulsifying properties, water and oil holding capacities. My third objective would be the screening of the peptides for functional properties and potential applications, in which I am focusing on the antioxidant properties by three different methods: DPPH, hydroxyl radical scavenging activity, and metal chelating activity. One minute, please. One minute, my presentation is stuck. Sorry. This is the materials and methodology which I will be following in the study. First, I have done the literature survey and have taken a good quality pigeon pea from Gulbarga, Karnataka, India. Further, I have went for the pre-treatment from crude fat extraction by Soxhlet method. Then I have went for the hydrolysis method. Then I have went for the quantitative estimation of anti-nutritional factors like phenols, tannins, phytic acid, and trypsin. Then I have went for the protein estimation through gel-dal method, then proximate analysis and degree of hydrolysis, as I have told before. Then I have went for membrane filtration and drying of the hydrolysates. After doing the drying, we have went for the determination of the functional properties, in which water holding capacity, oil holding capacity, emulsifying and foaming properties were determined to check the functional use of the protein hydrolysate. Finally, I have went for the screening of bioactive peptides to antioxidant properties, DPPH, HRSA, and MCA.
results and discussion. The first table determines that in the untreated one sample, that is UH, the protein content is 20%, while if I compare the untreated one, the acid hydrolysate, which I have derived, in that the protein content has increased significantly, that is 24%. And if I see the fat content, it is inversely decreasing compared to the untreated one. In acid hydrolysate, the fat content is 2%, while in the untreated one, it is 4%. So it is actually a potent vegan applicative use can be done from the speech and pea protein hydrolysate, as the protein is increasing after the hydrolysis because the bonds are being broken down and the peptides which were in dormant stage are getting active after the hydrolysis, and the fat is getting reduced because of the extraction method. If I talk about the yield, when I have taken the yield from the untreated one, it was 2.38 gram per 100 ml. And if I talk about the acid hydrolysate, it has given less yield because the supernatant we are using for the hydrolysate, we are not using the precipitate one. So the supernatant quantity is actually comparatively bit low than the precipitate. If I talk about the degree of hydrolysis, the acid hydrolysis is giving highest amount of DH value because after the hydrolysis, usually proteins will undergo hydrolysis. Even if in the natural process, the hydrolysis will happen. But if we make it hydrolyzed through acid, more hydrolysis degree increases, for which the active sites of the peptides starts increasing, and there is change in the amino acid sequence as well. So for which the protein content is ultimately increasing and visible in the protein content in gel dal method. If I talk about the FTIR, that is Fourier transform infrared spectroscopy, there are bonds, there are the peaks near 3,500 to 3,000 centimeter inverse of amide 1 and amide 2, which is indicating that the lysis or the hydrolysis of the hydrogen bonds are being done. While in the untreated one, we cannot see any sharp peaks. While in the acid hydrolysis, there are quite sharp peaks in this range, indicating that hydrolysis has been done and the peptides are available. If I talk about the anti-nutritional factors, we can see there is quite decrease in the phenolic content, and also there is decrease in the tannin content. And why I have taken iron? Because iron is a precursor for phytic acid. So if the iron content is determined, from which only we can determine the phytic acid. So we can see these anti-nutritional factors are being decreased. Anti-nutritional factors are essentially important to get decreased to make the proteins more viable, active, as these anti-nutritional factors are binding with the proteins and forming a complex which is ultimately decreasing the bioavailability of the proteins and making the mineral absorption, vitamins absorption less and in the body as a utilization purpose. So for which the hydrolysate is actually setting a better ability to for
Ms.
utilization.
Jyoti, sorry to interrupt you. You have under 2 minutes to wrap your presentation.
Okay, it's okay. Next is the foaming properties. In foaming properties, I would say the foaming properties are good at pH 6 because of the alkaline range. Similarly, for the emulsifying the properties, in which pH 6 is the most suitable one. If I talk about the water and oil holding capacity, the water holding capacity is increasing similarly for the oil. And if I talk about the antioxidant properties, most important, the DRSA, that is DPPH radical scavenging activity, is giving the more better results than HRS and MCA, determining the fact that antioxidant properties are present in PHNP. Ultimately, the summarized thing for my study is that I am putting forward a vegan protein hydrolysate from pigeon pea, which is not only suitable in protein content, it is also having a decreased fat content. It is suitable in terms of foaming, emulsifying properties, and also it is decreasing the anti-nutritional factors, which can be a better alternative to the soya protein commonly available in the market. So my aim is to give a cheaper source of protein supplement in the market. This is my acknowledgement. I would like to thank my supervisor, Dr. Nagvani ma'am, and these are my references. Thank you.
Your presentation is nice, ma. Actually, your topic is nice, but you need to present in a, what are the things that need to be presented in a right way? You have gone very fast in the end.
Yeah,
Yes?
actually, because of the time constraint and my presentation was stucking in between, so I had to go a bit
Initially, you were slow and rapidly you became very fast. But of course, it's nice. It's a good study. Thank you.
Thank you so much, ma'am. Have a nice day.
Thank you, ma'am. Thank you, Ms. Jyoti. For the next presentation, I invite Ms. Pallavi. Ms. Pallavi Rawat.
Am I audible, ma'am?
Yes, ma'am, you're audible. We can see your screen. Please begin.
Yes, ma'am. Good afternoon, all. Myself, Pallavi Rawat, research scholar from Shri Guru Ram Rai University, Dehradun, Uttarakhand. And my topic is synergistic effect of different Piper longum fruit extracts and electroactivated sodium caprylic solutions on various MDR salmonella strains. So first of all, we will see what is electroactivation. So based on the electrochemistry of water and Faraday's experiments, the electroactivation of aqueous solution has been practiced for more than a century. In this regard, Russian engineer Bakir discovered in 1972 that when mildly salted water solutions are exposed to an electric current, they might exhibit certain physiochemical characteristics that produce either alkaline or acidic media. So the electroactivation solutions are prepared by electrolyzing a diluted aqueous salt solution in an electroactivation reactor with the exteriors of electrodes. So the interaction involving the surface of the anode generates an anolyte, which is a highly acidic electroactivated solution. Simultaneously, the interaction of the diluted solution of salt with the cathodic surface of the electrochemically activated reactor leads to a basic solution that is highly electroactivated. So electrochemical reaction on both the electrodes causes variations in their pH and oxidation reduction potential and contribute to the creation of extremely oxidative species. From a pragmatic standpoint, a food business that employs organic acids may utilize the electrochemical activation process in transforming organic acid salts to their respective acidic form at various concentrations for substantial uses. So now about the Piper longum, which is my study plant. It is widely recognized as a long pepper or peepli. Piper longum and Piper nigrum are closely related species. Both possesses the alkaloid piperine compound, which gives both plant their pungent and spicy flavors. In the past, several indigenous groups have used the fruit stems and roots of Piper longum to make paste, juices, or concoctions that were used to cure bronchitis, coughs, colds, asthma, and they were used to treat scorpion stings, snake bites also. In a separate investigation, the plant extract showed noteworthy antibacterial action against various bacterial species like Staphylococcus albus, Bacillus megaterium, Pseudomonas aeruginosa, E. coli, and Salmonella typhi. So my aims and objectives are: aim is to investigate the medicinal potency of Piper longum fruits and electroactivated solution of sodium caprylic to combat the infections caused by MDR salmonella strains. And the objectives are determination of the antimicrobial activities of a combination of electroactivated solution of sodium caprylic and various extracts of Piper longum against different MDR salmonella strains, and assessment of their MIC MBC values. So material and method, this is the rough diagram of my reactor which I composed, and its design is: the reactor comprised 3 plexiglass compartments to prevent any disturbance between the current flow and the reactor. These are the plexiglass compartments. Anodic and cathodic portions were partitioned with the middle compartment region by anionic exchange membrane. This is the anion exchange membrane, and this is a cationic exchange membrane. And the rudenamuridium coated electrode with a 40 centimeter square active surface area were inserted into the reactor, and they were placed on both the sides of the reactor. The positive terminal of the electric current source was linked to the anodic compartment, and the negative was attached to the cathodic compartment. And the procedure was followed was a 300 ml of investigated solution was placed in reactors each compartments at time 0. After being loaded with the specified solution, the electroactivation reactor was attached to a DC electric generator here, which was then observed to provide currents like 100, 150, 200 and 250 milliampere current to the reactor. Electroactivation was carried out for a maximum of 45 minutes under each current value. Meanwhile, 10 ml of sample solutions were taken every 5 minutes from all the 3 compartments for total of 10 samples. Duration of 5 minutes gaps. All observations were obtained just after the solution were prepared. The whole process was done in 4 configurations. I had take 4 configurations. First was all compartment was filled with NaCl solution. Second configuration in which anodic and central were filled with sodium caprylate and cathode was filled with NaCl. Third, in anode, sodium caprylate salt was used, and central and cathode was in which NaCl was used. And in fourth configuration, all compartments were filled with sodium caprylate salt. My tested microorganisms were 4 non-typhoidal salmonella strains, which I procured from MTech Chandigarh, and these 4 were MTCC 660, 3225, 733, and 735. Then I evaluated the antibacterial properties of the mixture of plant extract and electroactivated sodium caprylate solution. And the analysis was done on the same day and in the several concentrations like 1%, 3%, and 5%. And to achieve the uniform bacterial colonies, a culture of chosen strains was spread throughout the MHA plates. And then the wells were puzzed into the culture to hold a 100 microliter mixture of solutions. The acidified NaCl and non-electroactivated NaCl were taken as positive and negative controls. And the estimation of MIC, MBC, it was done by microtitration plate technique. In this process, 5% of mixture solution under examination were serially diluted to obtain various concentrations like 2.5, 1.25, 0.625, 0.312, and 0.156% before the addition of 50 microliter of bacterial suspensions to the well, except for the negative control, which contained 175 microliter of broth. And the medium that was suspended without any sample solutions being studied were the controls, which were considered positive. After being inoculated, examined specimens were stored at 37 degrees Celsius for about 24 hours, and during which the bacterial growth was observed. Now the results and discussion. In this, the bacterial qualities of the various solutions varied, and in 1% solution shows the least antibacterial efficiency, and the 5% solution shows the highest antibacterial efficiency. So the table 1 showed the antibacterial efficiency of mixture of electroactivated aqueous solution of sodium caprylate and aqueous extract. The first solvent was aqueous extract of Piper longum, and these were the readings at different current 100, 150, 200, and 250. And as you can see in bold, the 250 milliampere current shows the highest inhibition against the 4 bacterial strains. And the table 2 showed the mixture with the hexane extract, and table 3 showed the mixture combination with the petroleum ether extract. And here same 250 milliampere current showed the maximum inhibition. And this is of chloroform extract, table 4, and table 5 is with methanol extract. So I had taken 5 extracts of plant. Now the evaluation of MIC of the mixture, and the best MIC was observed in the methanol extract with the combination of methanol extract, and the least was seen against the aqueous extract. So according to the MBC desired outcomes, a combination of plant extract and electroactivated sodium capryliate solution considerably decrease the bacterial effect of these 4 non-typhoidal salmonella strains. So at last, the current study which focused on inactivating non-typhoidal salmonella strains demonstrated that in the circumstances of the test carried out, treatment with a combination of food-grade sodium capryliate salt and plant extract is very efficacious. Furthermore, the outcome demonstrated that compared to traditional obstacle methods employed to guarantee microbiological food safety, electroactivation of these solutions produces particular synergistic circumstances with a greater impact. So the novelty of my research work is that, to our knowledge, no earlier publication has ever been employed a 45-minute electroactivation method with these ranges of current intensities against these strains. As far as we are aware, no study has ever been done on this sodium capryliate electroactivation procedure. In addition, extreme caution should be exercised when using acids in excessive concentrations, since they may have adverse impact on the food product's sensory quality. Utilizing low concentration organic acid solutions throughout the combination appears to be the preferred approach. The analytes of organic salt solutions undergo significant changes in characteristics throughout the electroactivation process that has the potential to upset the state of hemostasis of bacteria and work in a concert with heat or other obstacles. So globally, we anticipate that their high reactivity qualities may render them extremely efficient antibacterial agents, diminishing the need for various chemicals in the processing of food and safeguarding. In light of this, it is crucial to carry out more studies to have a better understanding of how this combination affects pathogenic and spoilage bacteria in various food originating from plants and animals. To better comprehend how these solutions might harm cell components, more research on their mode of action is necessary. So the addition of plant extract with electroactivation solution can enhance their antimicrobial activity, and their combination can prove to be a highly effective approach in many industries. In conclusion, these combination solutions may serve as a reliable stand-in for food safety by avoiding spoiling brought on by MDR non-typhoidal salmonella strains and by effectively regulating their pH range. Ultimately, this investigation creates novel and assuring opportunities for the development of low heat, nutritious and safe manufactured food items. If a combination of plant extracts and electroactivated solutions is to supplant conventional techniques of antisepsis and decontaminations and become widely employed in healthcare settings, more application-focused studies and developments are needed. Moreover, the pharmaceutical sector may use these combinations to manufacture drugs. So I acknowledge to my upper officials of the universities as well as the School of Basic and Applied Sciences, Research and Development Section, Sri Guru Ram Rai University, Dehradun, Uttarakhand, and these are my references. Thank you, ma'am.
Is there any message to the healthcare sector, ma?
Yes, ma'am.
Is there any message to the healthcare sector?
Yes, ma'am. In healthcare sectors, we can use them as a disinfectants. Means there are various problems related to nosocomial infections and
Yes.
contaminations. So with the help of this combination, plant and this electroactivated salt, we can cure this problem up to much extent.
How?
Then by making with the help of plant extract and this salt, we can make disinfectants.
So you want to prepare a disinfectant with the help of this plant extract? Yes?
Yeah, combination.
Yeah.
My study is the synergistic combination.
Okay,
So this
combination
study
of?
is plant extract and electroactivated sodium capryliate solutions.
So you can prepare any gels or sanitizers like that you are telling. So
Yeah,
that will be useful in healthcare sectors. Okay.
Yeah, in
one more
healthcare also.
advice is, one more advice is while preparing the slides now, it should not be in a paragraphic form. It should be in a bullets so that the slides will be easily visible.
Okay, ma'am. Okay.
Got me? Thank you.
Okay, ma'am. Thank you.
Thank you, Ms. Pallavi. I invite the next presenter, Ms. Vidya Chauhan. Ms. Vidya Chauhan, I invite you for a presentation. Following Ms. Vidya would be the presentation of Ms. Rajeshwari.
Am I audible?
Yes, ma'am, you're audible.
Okay, ma'am. I'm just sharing my window first.
Okay.
Okay. Hello?
Yeah.
Yes, ma'am. So my screen
is visible. Yeah.
Okay. Okay. So, very good afternoon to all, and myself Vidya Chauhan, and I'm from SGRI University, Dehradun. I'm a PhD scholar there. So, I'm very grateful that you have given me this opportunity to present my research topic in front of all of you. So, the topic of my research is FTIR analysis and in silico evaluation of selected secondary metabolite of medicinal plant Virginia ciliata's root and rhizome for the inhibition of some carbapinamase enzyme. Okay. So, firstly, I will start with the introduction. So, as we all are very much aware of the phenomena antibiotic resistance, and it is a very much concerning phenomena of today's life, and the resistance occurs between the microorganisms and mostly among the bacteria because of the indiscriminate and continuous uses of antimicrobial drugs. And because of that, some bacteria had become resistant to more than one drug, and this phenomena is referred to as multi-drug resistant. So the increasing incidence of carbapinamase resistance is particularly of our worrying concern, because only few antibiotic has been remained which are effective for such multi-drug resistant pathogens. Hence, my study basically focused on a certain alternative to combat certain antibiotic resistant pathogens. And since it has been noticed that from ancient times plant had been used as a substituent medicine for many for treating many diseases and for treating many microbial infections. Therefore, I have selected a medicinal plant named Virginia ciliata, which is an Himalayan plant, and it has been noticed that this plant has been used by the ancient people, also by the modern people too, for their therapeutic activity, and it has some bioactive compounds present in it, because of which it has its antimicrobial, antioxidant, and other beneficial activities present in it. So my study majorly focuses on the possible antimicrobial activity of this plant's rhizome and root extract, which has been prepared by using different solvents of increasing polarity. And after that, I have gone for certain techniques like GCMS and FTIR technique, and then I have gone for molecular docking certain secondary phytochemicals which I had obtained from my previous techniques like GCMS and FTIR. Okay, so these are the aims and objectives. So the aim of my study is to access the therapeutic potential of Virginia ciliata plants for the formulation of herbal drugs, so as we can cure more and more respiratory afflictions which are caused by certain MDR pathogens. And these are my objectives. So first objective was collection and identification of the plant. Then after collecting the plant and identifying it, it has been gone through Soxhlet extraction procedure for preparing different extracts according to their increasing polarity. And then I have gone for certain techniques like FTIR and GCMS, through which I can analyze certain phytochemicals which are present in my plant. And then after that, I have gone for molecular docking of these phytochemicals, which I have analyzed through FTIR and GCMS. And then I have docked these phytochemicals with certain proteins which I had taken from my carbapinamase resistant organisms. Okay, so GCMS technique, as we can see, GCMS technique has been used since many times for its beneficial use, as GCMS has given us very much information about the phytochemicals and secondary metabolites which are present in plants. Also, GCMS has been used for different samples for the analysis of certain substances which are present in a mixture. So in my plant, it has been shown that there are number of phytochemicals present in it. For example, likewise, there are gallic acid, tannin, flavonoids, glycosides, sterols, terpenoids, acetic acid, etc. And all of these secondary metabolites are somehow related to its therapeutic activity. After that, I have gone for FTIR. So in order to identify the type of chemical bonds that are present in these compounds, or you can say in my secondary metabolite, the FTIR was done. So the method by which the chemical bond is described by the wavelength of light observed is seen in the annotate spectrum, and the result I have shown in further slides. After this, molecular docking was done, and molecular docking is one of the tool which is used for computer aided drug designing, and it is a very much known tool for nowadays, and this technique has been used for developing and designing drugs. So these are the list of the proteins and the ligands which I have taken. So the proteins, all the 6 proteins was selected from the microorganisms which I was using for my study. All were carbapinamase resistant microorganisms, and in them these carbapinamase enzymes were evaluated. So these are the 6 one: OXA, KPC, NDM, IMP, WIMP, and NDM2. And ligands were selected from my GCMS library, which I was conducted previously. And these were the 35 phytochemicals or secondary metabolites which I have selected for my study. So in results, I have shown the plant authentication, which was done from BSI, and its extraction procedure, which was a Soxhlet extraction procedure, was done for the extraction purpose. 3 different solvents were made. 3 different extracts were made in different solvents, and these are the GCMS results. I haven't shown all of the results because it was taking too much of slides, so I had restricted my slides to certain numbers. So one of them is the list of phytochemicals in the extracts of Virginia ciliata root sample, and the solvent was ethyl acetate, and there was its chromatogram. Then another is FTI finding result. Again, this is the result for rhizome extract in methanol, and the another one is its chromatogram. Then finally, this is my docking result. So as I have told you earlier, that I have selected 35 phytochemicals, or 35 phytochemicals has been taken as an ligand, and 6 were my proteins. But as you can see here, only 32 of them has been selected for the further docking process, and most of them have cleared the Lipinski rule, in which they have cleared the Lipinski rule, in which they can be used for docking purpose, and they can be used for drug designing as well. Finally, this is the summary of my whole study. So as we all know, due to the limited treatment options for infections which are caused by multi-drug resistant microorganisms, there is a continuous search for certain novel compounds with which will have a broad spectrum of activity. And we have seen medicinal plants are one such alternative for which we are looking at. So plant-based antimicrobial have enormous therapeutic potential, as they can serve the purpose without any side effects, and they are also reachable to the people, more and more people. Also, there is a need that more and more studies can be made on certain plant, so that their efficiency can be exploited, and they can be further studied for better treatment also. So medicinal plants can also be used as source of lead compounds for drug design and development. And by this research, I was looking forward to design a better solution, or to design a better drug for treating certain multi-drug resistant pathogens, which cannot be treated with the antibiotic which we are having in nowadays world or scenario. Thank you.
What is the final message you are giving against antimicrobial resistance?
Ma'am, I'm just trying to say that apart from our synthetic drug, we can also use plant as an antimicrobial agent, because they have a plenty of therapeutic activities present in them. Also, they had a negligible amount of side effects present in them, and they are accessible to most of the people around. So that will be grateful for all the humankind.
Yeah, did you show that in the study?
Yes, ma'am, to a certain level.
Yeah, where? Did you mention that in the study?
Not in, ma'am, actually I was not able to compile all my study here, so I have just mentioned a limited part of it.
You have mentioned what you have done, yes, no?
Yes, ma'am.
Yeah, but you didn't compile it.
Yes, ma'am.
You
Yes,
didn't compile your result finally.
yes, ma'am.
Okay, you continue that, ma. It's a nice study. Okay.
Yes, thank you, ma'am.
Thank you.
Thank you, Ms. Vidya.
Thank you, ma'am.
So on that note, I'd like to welcome the next participant, invite Ms. Rajeshwari for her presentation.
Hello, ma'am. Am I audible?
Yes. Hi.
Okay, can I share the slides then?
Yes, please.
Yeah. Okay. Is my slides visible?
Yes, ma'am, it's visible. Yeah.
Okay, 1 minute. Okay. Good afternoon, everyone. I'm Dr. Rajeshwari Vittal, working at NITE University Center for Science, Research and Education. The topic for today is a molecular-based method which detects the presence of Legionella species from respiratory therapy equipment of tertiary care hospital, which can be a threat to patient care. So now let us see the history of the organism. So first, this Legionella species, it was reported from a soldier's blood culture in the year 1950. Even if it was discovered in the year 1950, the importance of Legionella as a human pathogen was recognized only in the year 1976, when there was an unexpected outbreak of fever with pneumonia, which was reported among the participants of American Legion Conference in Philadelphia, United States. Because of this incident, the pathogen was named Legionella because of the association with the American veterans. So this, and according to CDC, a study which was estimated that the true number of Legionella cases may have maybe more than 1.8 to 2.7 times higher than what it was reported. Okay, now let us see the salient features. So this Legionella, it comprises of around more than 50 species, and among these, around 30 species, it can infect human, especially the lower respiratory tract. So Legionella species are aerobic, gram-negative, non-spore forming proteobacteria, which has strict growth requirement. So these are ubiquitous in fresh water, especially it will thrive in rivers, lakes, streams, fountains, and hot springs, where there will be association with complex biofilm communities, and it can replicate inside various protozoan hosts as well. Legionella species, it survive in aquatic environment with the temperature ranging from 25 to 37 degrees Celsius, but it can also withstand the temperature more than 50 degrees Celsius for several hours, and it can also propagate at low temperature of less than 20 degrees Celsius as well. So the causative agent of Legionella is Legionellosis, and it has two distinct forms, that is Pontiac fever and Legionnaires disease. So Pontiac fever, it is seen in majority of our population which is exposed to contaminated aerosols. So it occurs in an acute outbreak mode and it is not related to pneumonia. Whereas the Legionnaires disease, it occurs from the inhalation of Legionella contaminated aerosol, but Pontiac fever appears in even the healthy individuals as well. So where do we find? As I said, from the natural environment, it can thrive to the artificial environment. So the sources of Legionella, it can be cooling towers, fountains, drinking water coolers, respiratory therapy equipment, feeding and dialysis tube, dentistry units, shower head, humidifier, birth pools, swimming pools, water parks, spa, and sauna. So in our work, we have targeted on, or we have looked at respiratory therapy equipment. So with this background, the scope of the study is, as we know, Legionella species are ubiquitous in aquatic environment, but are a potential hazard out of 40%, with 24 out of 60 species can cause infection in human. So often infection due to this pathogen go unreported because of the lack of awareness, and this organism is difficult to isolate as well. So this is considered as a disease of developed countries, but underreported in other countries of the world, including India. So in this study, an attempt was made to determine the prevalence of Legionella, Pneumophila, and related species in tertiary care hospital by both culture as well as culture independent method, recognizing the hurdles in culture dependent method. So with this, the hypothesis of our study is this Legionella, the respiratory therapy equipment in tertiary care hospital may harbor Legionella species, posing a potential threat to patient care. So utilizing a molecular based detection method will reveal the presence of Legionella DNA, allowing for a comprehensive assessment of the risk associated with these medical devices and highlighting the need for enhanced infection control measures in order to safeguard patient health and well-being. So the aim and objective of the study is as follows. This study aims to determine the prevalence of Legionella, Pneumophila, and related species in RTE in a tertiary care hospital. So by investigating the presence of these pathogens, the research pursues to assess the potential threat to patient care in healthcare setting and to contribute valuable insight that can help that can form strategies for mitigating these threats and safeguarding patient well-being. So objective of our study is to determine the prevalence of Legionella and related species from respiratory therapy equipment in Mangaluru, India, by both conventional and molecular technique. So our methodology or the isolation technique is as follows. First, a total of 30 swab samples were collected from the respiratory therapy equipment by using a sterile polyester sterile swab. Then the sites for sampling was identified as follows. We selected the compressor air outlet of nebulizer, the port of attaching mouthpiece of spirometer, the insertion tube surface of bronchoscope, and also the air inlet and outlet vents of ICU ventilators. So the collected samples were immediately immersed in sterile water and transported.
Ma'am, I'm sorry to interrupt you. You have 2 more minutes to wrap the presentation.
Sure. Okay, thank you. Within 4 hours, and the detection of Legionella was done by both culture and culture independent method. In case of culture dependent method, we used CDC method, that is disease control and prevention guidelines were used, where the buffer charcoal yeast extract agar was used as a plate. In that, a volume of 0.1 ml of sample was spread, and we have to, and the antibiotics were used so that the other microorganism will not, to inhibit the growth of other microorganism like glycine, vancomycin were the antibiotics which were used. Once the plate was spread, it was incubated at 2.5% carbon dioxide, and both the swab samples along with these isolates was subjected to DNA extraction, and then later it was subjected to a PCR, that is by molecular based techniques, where the primers which were used were conserved region of 16 RNA and virulence gene like dot A gene, and macrophage infectivity potentiator were also used. So after this, the isolates which showed positive was subjected to antibiotic susceptibility test by using McFarland standard, by using a QB-Bure method, where positive samples were subjected. So the results is as follows. We got the 50 number of samples were selected. These are the phenotypic characteristics which we can see. This first image, that is A, it shows the micro isolates which has been colonies which has been grown in BCYE buffered, GVPC. That those are the antibiotic name: glycine, vancomycin, polymyxin, and BCYE plate. Which these are the selective plates. Then from there, as I said, from the 50 samples, growth was seen. GVPC showed, that is phenotypical growth was shown in 33, where confirmed Legionella by PCR was only 2. So as I said, the two methods were used, conventional and culture independent method. So on GVPC, out of 37, only 2 showed molecule positive, whereas in culture independent method, we found 2 positive for 16 srDNA and 2.11 isolates showed positive for the virulence gene, which can also be used for for telling that it is a Legionella. After that, the isolates were subjected to, were sent for gene sequencing, and we have right now, about 16 srDNA gene sequences are present, and there are about 9 to 10 of .A gene sequences which are present. So in the phylogenetic tree, we can see that the sequence obtained, they didn't show any clonity. There was diversity present. As we know, the .A, that is .A gene, type 4 secretion, these are even close to the other bacteria also have, for example, Coxella and all, they also carry this. So it may be because of the horizontal gene transfer as well. So in the antibiotic susceptibility test, all the isolates showed sensitive, and the key findings which we found in our work is, this study documented the prevalence of Legionella in the coastal city of Mangalore, and it reveals the occurrence of Legionella pneumophila and associated species in respiratory therapy equipment. And this is the first documentation of Legionella species in respiratory therapy equipment in tertiary care hospital from Southwest Coast region, and it defines an important risk at a time when coronavirus has been resulting in severe pneumonia. And genetic diversity observed in the isolate from respiratory.
I'm sorry to interrupt you again. The 10-minute slot itself is over, so I would really request you to wrap it up in under 1 minute, because everybody's schedule will be
disturbed. Was observed, and this is important because there may be a horizontal gene transfer as well. So this is the future studies which we can think of because of the. Okay, thank you.
What is the most sensitive antibiotic in your study?
Ma'am, we used azithromycin. We found it to be very sensitive, and levofloxacin as well.
So azithromycin and levofloxacin are more sensitive in your study.
Yes. All,
so your sample size is only 50, no?
Which one, ma'am?
Your sample size.
No, 50.
Why that much low?
No, I showed only one part of the work. Actually, we took from different also, like cooling towers, artificial water body were also involved, but when
Yeah.
it comes to this, only 50 weight.
Okay, it might have been more, na?
Yeah,
So it's a
yeah.
good study. It might
Yes,
have
yes.
been more so that it will form a base.
Correct,
And one
correct.
more thing is only 2 are positive by culture
Yes.
out of those
Yes.
50.
Yes, but
That was
in
confirmed by
PCR,
PCR.
yes, PCR we got more positive. So it
Here
may have
also
gone to
2 are
VBNC
positive
state.
for Legionella and remaining 11 are positive for virulence gene. That's what you are
Yes,
telling.
yes, yes. And because in the culture independent also we got positive of for 16 srDNA, it may because it must have gone for that VBNC stage. So yeah, the conventional method we can't completely rely. That's what I
Yes.
experienced.
Okay. Your title may be a bit specific.
Okay, okay, okay.
Thank you.
Yeah, thank you. My thanks to Pauline as well for helping me. Thank you so much.
Happy to help you. Thank you so much.
Yeah, bye.
On that note, let me invite bye. See you. I'll invite the next presenter, Ms. Neetu Babu. Am I audible?
One thing I want to tell everyone, don't focus much on the introduction part. Just present results, your discussion neatly. Focusing much on the introduction part and going very fast for the results and discussion. Okay?
Okay, ma'am. Shall I start sharing the screen? Yes, thank you, ma'am, for the insights. Yes, Ms. Neetu Babu, please share the screen. Good afternoon. The topic of my presentation is comparative evaluation of extracellular enzymatic activity of pathogenic Candida species and its correlation with mucositis in head and neck cancer patients on radiotherapy. So coming to the introduction, Candida albicans is the most important species that is isolated from oropharyngeal infections in patients who are undergoing radiation therapy for head and neck cancer. And oropharyngeal candidiasis, mucositis is a major dose-limiting toxicity that occurs in these patients. Recently, there is an increase in the incidence of non-albicans Candida species causing infections in these patients. The difference in the enzymatic activity expressed by the Candida species as well the different Candida species, that is the Candida albicans as well as the non-albicans Candida species, will determine the virulence, and thus it plays a pivotal role in understanding the pathogenesis of candidiasis. And also it will help to introduce new anti-candidal drugs which can be targeted against these enzymes. So the virulence of Candida species is attributed to a variety of mechanisms which includes biofilm formation, its adherence capacity and production of extracellular enzymes like proteinases, phospholipases, esterases and also hemolysins. So my study objective was to determine the extracellular enzymatic activities like phospholipase production, proteinase production, esterase activity and hemolysin production of the Candida species that was isolated from head and neck cancer patients who were undergoing radiation therapy. And the second objective was to determine the correlation between the severity of mucositis and the virulence factor production by this Candida species. Materials and methods. This was a cross-sectional study which extended over a period of 4 years, and 276 patients who are undergoing radiation therapy for head and neck cancer in our tertiary care cancer hospital was included in the study. Inclusion criteria was all the patients were histologically proven head and neck cancer with stages 3 and 4, and exclusion criteria was that patients who received antifungals 15 days prior to radiation therapy or patients who had Candida infection at the start of treatment. Swabs were collected from the patients who developed candidiasis in between, and these swabs were processed in the microbiology laboratory as per standard protocol. Species identification was done based on color and high chrome agar, and also I did a Vitek automated system identification, and other tests like germ tube test and chlamydospore test were also done. The conventional method was also done along with the automated procedure.
Then extracellular enzymatic activities like phospholipase, hemolysin, esterase, and proteinase were used by plate method. In vitro method was done by methods described earlier by the references are given in the end of the presentation. Mucositis was graded by radiation oncologists according to the radiation therapy oncology group criteria, that is 1, 2, 3, and 4. The grades were grade 1, that is mild, grade 2 moderate, grade 3 severe, and grade 4 life-threatening. And Spearman's rank correlation was done to analyze the strength of association between mucositis and this extracellular enzymatic activity. So the determination of phospholipase activity, proteinase activity, hemolytic activity, and esterase activity were followed by methods already explained in the literature. Coming to the results, this is the demography of head and neck cancer patients who was selected in, who was included in my study. That is, men were more than women. Men comprised of 83.7% of the study population, whereas women were only very less, that is 16.3%, because head and neck cancers are mostly seen in men. That is why this difference. And the mean age of the patients were 65 plus or minus 11. And the primary site of the cancer, malignancy, most of the patients had the malignancy. Majority of the patients had the pharyngeal malignancy, which was followed by malignancy of the oral cavity, then larynx, and finally nasal cavity and paranasal sinuses. Only 5.4% of the patients were there. Then mucositis grading. Grade 1 mucositis was not developed by any of the patients. Grade 2 mucositis was seen in 36 patients out of a total of 76 patients who developed mucositis, and grade 3 mucositis was developed in 39 patients, and 1 patient developed the life-threatening grade 4 mucositis. Coming to the results, phospholipase activity was shown by very few strains of the Candida albicans as well as non-albicans Candida strains. A total of 18 strains only showed phospholipase activity, whereas proteinase, hemolytic, and esterase activities were shown by majority of the strains, that is 88, 84, and 77 number of strains. A total of 97 Candida strains were isolated in the study. Then coming to the correlation between mucositis and the enzymes, a Spearman's rank correlation revealed that there is a weak positive correlation between the enzyme, the enzymatic production of mucositis and phospholipase and proteinase activity, whereas hemolytic activity showed a negative correlation. And these are the photographs of the extracellular enzymatic activities as seen on the plates. First is phospholipase, then proteinase, hemolysin activity, and esterase activity. Coming to the discussion, Candida albicans was isolated from 56 Candida albicans strains were isolated, and non-albicans Candida strains were 41, and which is in par with other studies which reports an increase in the incidence of non-albicans Candida species recently, causing infections in these radiation patients undergoing radiation therapy for the head and neck cancers. And proteinase activity, as I showed in the previous slide, was shown by 88 number of 88 strains, whereas hemolytic activity by 84 and esterase by 67. Phospholipase activities were shown by very few isolates, that is only 18 isolates showed phospholipase activity. And in our study, Candida albicans was the most frequently isolated Candida species, and it showed a significant activity of all the 4 enzymes, which accounts for its virulence. High virulence was shown by Candida albicans strain. A substantial portion of non-albicans Candida species demonstrated moderate enzymatic activity. Even though the mean proteinase activity as well as the phospholipase activity was slightly higher for the Candida, non-albicans Candida strains, but high degree of activity was shown by Candida albicans strains. Non-albicans Candida strain showed mean activity was slightly higher, but the high degree of activity was shown by Candida albicans. The pronounced phospholipase and proteinase activity in Candida albicans displayed a strong association with the occurrence of mucositis, as it is shown by the Spearman's rank correlation. So the overall prevalence, coming to the conclusion, overall prevalence of Candida infection among head and neck cancer patients is 35.1%, and the most prevalent species is Candida albicans in patients who are undergoing head and neck radiation therapy for the head and neck cancer. And even though all the strains included in this study are pathogenic, the enzymatic activity shown by them were variable. And the Candida albicans strains isolated in the study demonstrated strong performance of all 4 enzymes. That is, high activity was shown by Candida albicans strains for all the enzymes. It suggests its potential for virulence. And majority of the Candida albicans strains were proteinase producers, which indicates their role in increasing the severity of mucositis in head and neck cancer patients, which is a cause for concern. And phospholipase activity, which has a potential in the emergence of drug resistance, even though it was very detected in 18 strains only, very few strains only, it has to be closely monitored. And these are my references. Thank you.
How will you take your study further forward?
I would like to proceed with the molecular part of the study, ma'am, because all these virulence factors, genes has to be studied in detail.
Okay.
But
you
I
got
have...
phospholipase is having a less compared to the other enzymic activities.
Yes, ma'am. Only 18 strains showed phospholipase activity.
How will you take this forward?
Phospholipase activity, ma'am?
Yes.
I have to see what is the gene that is present in these. That is, the Candida albicans and non-Candida albicans strains, is there any difference? And I'll have to compare it with the normal population. And in this study, I have not compared it with normal population. I'll have to compare it with the normal population also to see the activity.
So what is the non-Candida albicans strain that is more common?
Majority is Candida tropicalis, ma'am. And then I got Candida krusei, and the other 2 strains are Candida glabrata and Candida parapsilosis. Totally, I got 5 strains. Candida albicans.
That in the presentation, it will be nice if you have included.
Yes, ma'am. Only 10 slides were allowed, so I focused on the topic. That is
okay. Nice.
Thank you.
Thank you, ma'am. For the next presentation, I invite Ms. Pavni.
Yes, ma'am.
Please share your screen.
One minute, ma'am. Yes, ma'am. 1 minute. Ma'am, can I present later? Some problem with the network.
You want to present later, is it?
Yeah.
Okay.
If possible. Or else give me 1 minute, I'll just connect.
Okay. I think you can just go ahead.
Yes.
Next 2 presenters are not available. Ayan Ghosh, are you available for your presentation? Mr. Ayan?
Yes, ma'am. I'm available.
Yes. Okay, then Pavni, we will finish Mr. Ayan's presentation and we'll come back to you.
Yes, ma'am.
Okay, great. Ayan,
Can
please share. Yeah, please
okay,
go ahead.
ma'am. Just a So it's visible?
Yes, it's visible. Yeah.
So, very good afternoon, everyone. My name is John Bush, and I'm a student of Humanity Institute at first semester. And my point of discussion will focus on stress and infection. When we are trying to find out the missing link between several communicable and non-communicable diseases, we found oxidative stress, which matching the all sort of diseases irrespective of their nature. There may be acute or may be acute exacerbation of their chronic diseases. Today, in our study, we have trying to figure out what could be the main factors which can result into the increasing rate of infections. In the COVID-19 era, we have already witnessed that there is a close relationship between oxidative stress, and lung infection. Not only the lung infections, in this time there are a lot of infections and lot of non-infective diseases are there, which are non-communicable diseases. They are also caused by these also are caused by the oxidative stress. In this work, we are trying to figure out biomarkers that is the level of oxidative stress and relationship with the infections, and we are trying to figure out how oxidative stress is creating infections. So our main aims and objective is to find out the several communicable and non-communicable diseases, to find out the basic cause for infection, to find out the complication for the infections. And for this study, we have made a hypothesis, that is H0, that is completely opposite to our study, that says the oxidative stress is not the reason for infections. And another is H1, that is working hypothesis, that says the oxidative stress is the reason for infection. And for this study, sorry, to make this study, the mode of our study is mainly case control study. There, where the case are established against a good number of case control who have shown incidents of infection, but no oxidative stress will be seen. A total number of 658 cases were observed initially who have visited us with any sort of infections. Among of them, 847 cases were selected as they have matched with our inclusion criteria. That is, who have matched with any stress-provoking factors. That is, having addictions, whimsical interests of drugs, comorbidities, etc. These cases are organized under the groups of having mild, moderate, and severe oxidative stress as per the level of biomarkers. And this is the mainly we have studied only two biomarkers, that is glutathione and that is and another is SOD, that is superoxide dismutase. We can see that the in case of patient without oxidative stress, the amount of glutathione is much higher. And in case of the patient with oxidative stress, the amount of glutathione, that is antioxidant, which is lesser than the previous. In the similar way, this is applicable for the SOD, that is superoxide dismutase. And this is the bar diagram. And from this bar diagram, we can say that there are three classes are present. One is mild, moderate, another is severe. In the class, 4 bars are present. The blue is denoting common cold, orange is denoting URTI, that is upper respiratory tract infection, gray for LRTI, that is lower respiratory tract infection, and UTI for yellow. In case of mild oxidative stress, the chances of common cold is higher than URTI. Then LRTI, then minimum chances is there in UTI. Whereas in case of moderate, we can see that the LRTI is playing significant role. It is uprising after the URTI, after that common cold, and after that UTI is there. And finally, when the severe comes, LRTI playing the most significant role. Then UTI, then URTI, and finally it comes. Finally, common cold is seen to be decreasing comparing to the others. And what is the reason behind that? Because it is the severe oxidative stress, the lung infection or alveoli is going to be damaged as much in virulent manner. And this is the pie chart where you can see the main cause behind the oxidative stress is the comorbidities, that is 33%, and then comes psychological stress, that is 26%, addiction 18%, and drugs and others are 23%. And this is the chart where you can see how oxidative stress is creating our infections. And finally, coming to the conclusion, the lower p-value is higher than significance of the data could be derived. In this case, group C has p-value of 0.001, that is highly significant with the H1, disproving the H0 that is null hypothesis, and establishing the direct correlations with the oxidative stress and infections. These are the reference, and thank you.
So what is your final conclusion?
Final conclusion is that the oxidative stress has a direct relations with the oxidative stress. Oxidative stress has a direct relations with the infections.
With the infections. Okay, what is your inclusion criteria?
Inclusion criteria was those who have matching with the inclusion criteria, right? Who have the similar or in infectious disease, like who have respiratory symptoms, who have breathing distress. These types of patients we have included in
So
our
all
studies.
infections are taken into control.
Yes.
All infections
we have included in our criteria.
Pardon?
All type of infections we have
All
included.
type of infections. Okay. Any age group?
Any age group? Mainly we have included 17 to 18, 17 to 25 years. After this was the second, and the majority of the patients was the more than 25 to 36 years.
Okay. What is the method you have used?
Case control study.
What is the method?
Case control study.
No, ma, not. You have detected two now, SOD and glutathione.
What one, sorry?
So what is the method?
What is the method? That means
You did not include your inclusion criteria, exclusion criteria, and what is the method you have followed?
Yeah, ma'am. The mode was the mainly case control study, ma'am. It's in case of inclusion
Go to
criteria,
the next slide. Next slide. How this one?
Yes, ma'am.
How did you detect this one?
We have studied with the level of blood. We have collected blood sample from the patients, and from the blood we studied the glutathione and SOD.
Yeah, how did you study?
I have measured the data. I have collected the data from the blood reports.
Pardon?
I have collected data from the blood reports of patients.
Patients.
Yes. Those
No,
who have the oxidative
you have
stress.
you have evaluated total glutathione and SOD now values.
Yes.
How did you come to the conclusion of those values? What is the method you have used? You didn't get me. It's okay. Next presenter.
Yes, okay.
Thank you, Mr. Ayan. Ms. Pavni, are you ready?
Yes,
with your
ma'am.
presentation?
Yes, ma'am.
Okay, please share your screen.
Is my slides are visible, ma'am?
Slides are not visible. Your file explorer is open. I think it's loading.
Is it visible now, ma'am?
Sorry, not yet. Still loading. We can see your screen.
I could be able to see here, madam.
Okay. Or maybe is it only for me? Am I not able to see the slides? Is everybody else able to see
We
the
are
slides?
also not able to see.
Have you shared the correct screen?
Yes.
Like you have to share you have to share screen the correct tab. Can you just check that once?
Is visible now, ma'am?
Yes, it's visible.
I'm sorry for the interruption.
No problem. Please go ahead.
Yeah. Very good afternoon, everybody. This is Pavani. I'm working under Dr. Nagavani Shivshetty ma'am in Geetham University. My research topic is identification and interpretation of Staphylococcus aureus resistant to conventional antibiotics in a hospital environment. The introduction, MRSA is a strain which was firstly isolated in 1960. It is a gram-positive bacteria that are small, round, and occurs like a clusters in a bunch of, it looks like a bunch of grapes under the microscope. It colonizes and inhibits the mucosal surfaces. Sorry. MRSA is one of the most common and virulent strain of antibiotic-resistant Staphylococcus aureus. In medical facilities, MRSA causes life-threatening bloodstream infections like pneumonia and surgical site infections. High-level resistance to methicillin is caused by mecagin, which encodes by PBP2A. Resistant strains cause many hospital-acquired infections. Aims and objectives of this study to isolate Staphylococcus aureus from clinical samples, inclusive of blood, urine, pus, sputum, and other body fluids, to identify MRSA by phenotypic method according to the CLSI guidelines, and also to check antimicrobial resistant pattern by Kibbe-Boyer method, as well as MIC by broth dilution method. So the materials and method. To differentiate MRSA and MSSA, cefoxtin disc diffusion method was used. If the zone of inhibition was less than 21 mm, it was considered as a cefoxtin. The first picture shows it is a MRSA, and the second picture, which is a clear zone of inhibition around the cefoxtin disc, so it is considered as MSSA. So the materials and methods. All the clinical samples except urine were cultured on blood agar, Mekong agar, and nutrient agar. For urine samples, clade agar was used. So the virulence factors like coagulase, hemolysis, and DNase, AMR studies to different antibiotics also captured in this study. Determination of MIC of amikacin, azithromycin, levofloxacin, and cephalexin by broth dilution method was checked. So these are the different types of media in which MRSA was showing. In the first picture, golden yellow color colonies and blood agar, and nutrient agar. The second picture it was showing, and yellow color colonies were produced on mannitol salt agar. So in DNase agar, when we have streaked on MRSA sample, there was a clear lysis when we added 1% HCl around the colony in the picture. So we have found around 11 colonies among 84 MRSA samples. So 5 samples were from pus sample and 4 were blood sample. The prevalence was more in CCU followed by orthopedic ward. So the total number of samples, 185 gram-positive cocci samples were collected. Among 84 were MRSA, 68 were MSSA, and 32 samples were coagulase-negative Staphylococcus. So the sample prevalence was more in blood samples, followed by pus samples. And ward-wise data, CCU was showing highest prevalence, followed by PICU and NICU. Highest antimicrobial resistant was found in quinolones, followed by macrolides, and highest resistant was shown in cephalosporins, followed by macrolides and quinolines by Kibbe-Boyer method. So the highest antibiotic resistant antibiotics also cross-checked with MIC by broth dilution method. 4 antibiotics were checked in MIC method like amikacin, azithromycin, cephalexin, and levofloxacin. So amikacin was showing 13 sensitive by broth dilution method, and 39 samples were intermediate sensitive. Whereas azithromycin, 12 samples were sensitive, 14 were intermediate sensitive. Levofloxacin was 5 samples were sensitive by MIC and 12 were intermediate sensitive. And we couldn't see any sensitive for cephalexin by broth dilution method as per the CLSI guidelines. So all the samples were extracted by automated DNA extractor, which is a QIAcube, left side, the first picture, which is showing. And we have used QIAGEN method, QIAGEN kit for the DNA extractions. So the summary and conclusion, the total number of MRSA isolated were 84. Out of 2,500 samples were screened. Prevalence was more in male patients compared to female group. Most effective age group showed was 46 to 60 years, followed by 31 to 45 years. Prevalence of MRSA found more in blood samples, followed by pus. In case of ward-wise screening, MRSA were high in CCU, followed by PICU. High AMR pattern was observed in cephalosporins, followed by macrolides and quinolones. Among 84 samples, 45 found to be multidrug resistant. More than 3 group of antibiotics resistant, we consider it as a multidrug resistant. In our study, we have also seen more than 6 group of antibiotic resistant patient samples also. The MIC results indicated most of the organisms were in intermediate range of susceptibility. This study will be helpful in defining guidelines for control and treatment of MRSA infections. This opens the door to the understanding the prevalence of drug resistance to the high-end antibiotics commonly used in oral forms for treatment of infections. These are some of the references which I have taken for my study. And I would like to thank my guide, Dr. Nagaveni Shivshetty ma'am from Geetham University. Special thanks to OIC Hospital and Research Center, Hyderabad. I would also want to thank BioLEAGUES for giving me this opportunity. Thank you.
So did you check for vancomycin?
Yes, ma'am. Vancomycin also were checked.
Yeah, what is your result?
Only 2 samples were resistant, madam, for vancomycin. Remaining everything is sensitive.
But you didn't show that one, no?
Actually, percentage-wise, we have checked, madam. That in AMR pattern, which I have shown. I'm so sorry for that. I'm sorry, something is wrong with my screen share.
It's okay, ma.
I'm in the, yeah, I have checked for the linezolid, we got 8 sample resistant, madam. For vancomycin, we got only 2 sample resistant. That's why we have not compared with the MIC method. For highest resistant groups which were showing by Kibi-Boyer method, we have checked the MIC methods.
Okay, what did you do with the PCR?
PCR further we will be doing, madam, for gene identification, sequencing PCR we will be doing. So far we have done DNA extractions and procured the DNA extraction. Then I will be doing the
shown the culture results only. So you didn't do any PCR results?
Molecular characterization not yet done, madam.
You have not done. Okay,
not yet done. Thank you, ma'am.
Thank you very much. I would like to invite the next presenter, Mr. Samik Banerjee. I just want to mention Dr. Chandralekha and Ms. Vandana Rani are not reachable. So we are moving ahead. Yes, thank you, Samik. Please share your screen and start presentation.
Ma'am, is visible?
Yes, sir, it's visible.
Good afternoon, ma'am. I am Shomik Banerjee, student of Humanity Institute of Advanced Medical and Cultural Research. Today I am going to discuss about impact of antibiotic resistance in environment and public health. In current scenario in US, more than 2.8 million antimicrobial resistance occurs each and every year as per the CDC report. With a further delay, I am going to discuss what is antibiotics and what is antibiotic resistance. Antibiotic, that means it can kill the bacteria. Not only it can kill the bacteria, it can remove the bacterial infection. So antibiotic are the class of drugs that used to treat bacterial infection. They work by either killing bacteria, that is bactericidal, or inhibiting their growth, that is bacteriostatic, allowing the body's immune system to eliminate the bacteria, not only the bacteria, rather than the bacterial end products also, with the degraded cell and other tissue. And whenever we are passing through the era of bacterial infection, not only the bacterial infection, there might be some kind of other infection, and at that time people are dying for this infection, and at that time the world facing the fields, and what is the main reason, and discover something to kill the bacteria. And we know the first antibiotics known as the penicillin, discovered by the Alexander Fleming in 1928. And the 1940 to 1960, the golden age of antibiotics. At that time, there are several antibiotics are discovered, like tetracycline, streptomycin, erythromycin. And over the time, the overuse and misuse of antibiotics that can resist the antibiotic, and also it will creating the major global health concern. and what is antibiotic resistance, and what is the terminology are appears in a current situation where the new infection are occurs as well as the new bacterial gene modification are occurs, and that's why the antibiotic resistance are occurs. And what are the reason behind the antibiotic resistance? There might be the overuse and misuse of antibiotics uh, without any proper guideline. There might be the use of antibiotics without proper identification. There might be the use of antibiotics in a whimsical manner. In this image, you can see the classification of antibiotics. What is the aims and objectives? Aims to research this research to aims to elevate the cause of antibiotic resistance as well as plan an effective management policy differentiating constitutional and generalized approach. Objectives to find out the root cause of antibiotic resistance, to rule out the exact cause, how antibiotic resistance impact in environment, to rule out the exact cause, how antibiotics resistance impact in public health. And this is the materials and methods. The mixed method, we have one part is literature review and another part is clinical survey. We have taken 100 cases, among which 30 cases are showing remarkable antibiotic resistance, and not only the antibiotic resistance, there are remarkable amount of oxidative stress also. In case of antibiotic resistance, 10% are those who have high sensitivity to azithromycin in past, but in the long run, after 6 to 5 years, 5 to 6 years, the azithromycin sensitivity is not in those patients, so they are become resistant to azithromycin. And during the discovery of antibiotics, we have witnessed there are lots of advancement occurred in the antibiotics. What are the reason behind that? The reason is that to minimize the adverse reaction, to secure more efficacy when the microorganisms are not too much sensitive. Till now, they are modifying. And these are the resistance result and discussion. There are several theories: the jam theory and epidemiological triad. Jam theory, these are two perspectives. One is the Koch postulate, another is the Pastu theory. And jam theory clearly states that germs can be found in the diseased individual. And the father of pathology, Rudolf Virchow, he explained the epidemiological triad, and he also stated that germs are the result, not the cause of the pathological condition that have occurred previously. And the epidemiological triad have three basic pillars. One is the pathogen, host, and environment. Whenever we are not focusing in the lower part of the iceberg, rather than we are focusing in the upper part of the iceberg, and we are cutting the upper part of the iceberg in a virulent manner, then the lower part of the iceberg are becoming more deep seated. As a result of that, the diseases are becoming more deep seated. And the bacteria which are targeted, I must say, bacteria are not a pollutant agent, rather than bacteria are the scavenging agent. When the bacteria enters your body have some materials to eat by the bacteria, rather than otherwise your body have clean, then the bacteria are not going to be survive in your body. So the first and foremost things is not the bacteria. First and foremost things is the environment or atmosphere provide your body by the bacteria. And the next is reductionism and holism. Reductionism is a concept state that uh, disease can caused by the specific microorganism. So whenever we are kill the specific causative microorganism, and then disease will be treated. And the holism and reductionism ignore the environment, and holism don't ignore the environment. And the holism concept state that not only kill the specific causative microorganism, rather than we kill the host microbiome, and also negative effect to the host internal and external environment. And this is the future genetic mutation as a cause of antibiotic resistance. According to WHO, health is a state of physical, mental, social well-being, not merely the absence of disease or infirmity. So the health become affected in the by the means of exciting cause, maintaining cause, and fundamental, and then disease will be found. And disease have some manifestation: clinical manifestation, subclinical manifestation, and structural alternation. And the combination of three manifestation is known as the symptomatology. And each and every and one manifestation is known as the symptoms. When the drugs or the antibiotic are administered and to treat the symptom, then it is known as the symptomatic treatment as per the reductionistic perspective. And when the drugs or antibiotic are administered and it treat the whole disease, that is known as the symptomatological treatment as per the holistic perspective. When the antibiotics are kill the microorganism, not only they kill the microorganism and also kill the host microbiome, and it will impact and occurs to the oxidative stress and other also metabolic dysfunction, and also impact to the fundamental cause remain and the maintaining cause remain. When the fundamental cause remain and maintaining cause remain, and more antibiotics are administered, then it will occur the epigenetic mutation of the bacteria to fulfill the Darwin's theory, survival of the fittest, and occurs antibiotic resistance. And the primary action, that is the action by the drugs, and the secondary action, that is the reaction the body against the drugs. When the secondary counter action occurs, and the diseases are becoming much more deep-seated, and also it occurs the non-homologous environment, and it will produce the antibiotic resistance in a greater extent. And how antibiotic resistance can left impact in the environment and public health. Impact in public health: reduced treatment efficacy, increased healthcare cost, limited treatment option, and impact in environment, environmental pollution, and impact in ecosystem. And this and I conclude that the antibiotic resistance is an anthropogenic crisis because of the bias of knowledge and incomplete understanding of the nature. So it is a high time to implement the holistical and multisectoral approach to reduce the antibiotic resistance infection. These are the reference, and thank you.
What is your final message?
Final message, ma'am, to implement the holistical perspective.
How?
Bacteria not only kill the specific causative microorganism, antibiotic not kill the specific causative microorganism, they have impact in other environment. So we have focus on the other environment, and then we treat the infection.
What is that other environment?
Sorry, ma'am.
What is that other environment?
Other environment, whole environment, ma'am. Whole environment is the
okay. Next.
Thank you, Sami. For the next presentation, I would like to invite Ms. Manita. Manita, are you available?
Yes, I am available.
Hi.
Good afternoon. Good afternoon, Dr. Katyayani. Good afternoon, Pauline, all the participants.
Hi.
Hi. I'm just sharing my screen. I hope it is visible.
Yes, it's visible.
So here I am presenting a part of my work. It's not my whole work. The title of today's presentation is Burden of Carbapenem-resistant Acinetobacter baumannii involved in ventilator-associated pneumonia at a tertiary care hospital, Bathinda, Punjab. So, ventilator-associated pneumonia is defined as infection of the pulmonary parenchyma in patients exposed in invasive mechanical ventilations for at least 48 hours and is part of ICU-acquired pneumonia. Ventilator-associated pneumonia affects 9 to 20% of mechanical ventilated patients, and its global prevalence is 15.6%. But this rate is differ from hospital to hospital, from state, or even within the country. And even if in the same hospital, different department may have different rate of ventilator-associated pneumonia. We can divide ventilator-associated pneumonia in two parts, early onset versus late onset. Early onset is caused by Streptococcus pneumoniae, Staphylococcus pneumoniae, Haemophilus influenzae, and Moraxella catarrhalis, whereas late onset is caused by Klebsiella pneumoniae, Pseudomonas aeruginosa, Acinetobacter species, and Enterobacter species. This is a CDC criteria for diagnosis for WAPE, where at least two from the clinical criteria, if fever is more than 38 degrees Celsius, whether it's a condition of leukopenia, whether WBC is lower than 4,000 or more than 12,000, and onset of purulent sputum, worsening of gas sections. Microbial criteria, we have to see if we are seeing bowel samples, then it's 10 raised to 4 colony forming units. And in case of ET, endotracheal aspirate, 10 raised to 5 colony forming unit. And further, clinicians see CT extra. So here are non-fermenters who can be associated with this ventilator-associated pneumonia. But WHO highlighted Acinetobacter baumannii and Pseudomonas aeruginosa as a priority of concern. So actually, my original work is based on non-fermenting gram-negative bacilli. As I said earlier, I am here presenting only a part of my work that is based on Acinetobacter baumannii. Acinetobacter baumannii is a notorious pathogen as identified researchers because it can survive in harsh condition. Even it can survive on desiccation form. It's by producing carbapenemase enzyme. Carbapenemase enzyme, generally beta-lactam antibiotics are divided in 4 classes, and beta-lactamases are divided in 4 classes, class A, B, C, and D. So carbapenemase cover class A, B, and D. Just give me 1 minute. I am okay. Lesser pointer. So this carbapenemase cover class A, B, and D. These are the enzymes belong to these classes. I have highlighted OXA in a red mark because OXA has a weak hydrolysis activity, and due to it, actually this Acinetobacter baumannii is a non-fermenters, and non-fermenters shows weak hydrolysis activity. They are slow growth. They show slow growth. So early identification is not possible. So whenever a patient is suffered from Acinetobacter baumannii or any other non-fermenters at initial stage, it is very difficult to identify them, and it takes almost 4 or 5 days for the identification. So my actual research work is based on this OXA because I have proposed this work on 2021. As we all know that COVID-19 was going on, so research work was focused on COVID-19. So this class of enzymes was neglected by all the researchers. So that time I proposed this work. Due to time limitation, I have actually there are intrinsic and extrinsic resistance mechanisms that is adopted by this Acinetobacter baumannii. So I have published few papers where I have given much detail about it. So it is Acinetobacter baumannii. It is considered that it carries a OXA51 gene, and it is specific for Acinetobacter baumannii. But I have done a genomic analysis, systematic review on genomic analysis, and what I have found that mainly 92.62% A baumannii, they carry BLA OXA23, that is plasmid mediated. So there are different kind of mode that bacteria can adopt for resistance. So like porine mediated, porine are proteinaceous channels through which substance can enter into the environment. So by loss of these porine channels, they can acquired resistant to antibiotics. There are, as you can see here is a efflux pumps, and by overexpressing this efflux pumps, most of the antibiotics, it pumps out from the external environment. Same as if our house is flooded with water, what we will do, we will get rid of that water by applying a mortar. So same strategy applied by this bacterium. There are plasmid, they harbor, and this plasmid may carry a different resistance genes. So by taking up this plasmid, they can acquire this resistance mutation. There are other enzymes, mutations can be possible. Antibiotic, they can either modify the antibiotics or they can hydrolyze those antibiotics for acquiring this resistance. So there are so many ways from they can confer resistance. Aim of this investigation was to identify and ascertain the prevalence of carbapenem resistance A. baumannii from ET and BAL samples, as well as the antibiotic susceptibility profile of these pathogens by using Vitek-2 system. So material and methods, we have carried this study at Center for Interdisciplinary Biomedical Research, Adesh University, Bhatinda, and we have collected 200 suspected ET and BAL samples from the patient at the bacteriology laboratory of Adesh Hospital, Bhatinda, and identification in AST was done by using Vitek-2 system. Inclusion criteria, we have selected only those patients for study who were on mechanical ventilation for more than 48 hours. And in our exclusion criteria, if pneumonia prior to mechanical ventilation or less than 48 hours, we have excluded those patients. So this is our method. We have received samples ET and BAL both from bacteriology lab of Adesh University. We have divided sample in two parts. One part we have preserved in 50% glycerol at -20 degrees Celsius, and the second part we inoculated on blood and McConkey agar, and we have performed gram staining for identification of gram negative isolates. Gram positive isolate we have excluded. Then gram negative isolates were identified for non-fermenters by Vitek-2 system. Total 200 samples we have collected. And after that, isolate and samples other than non-fermenters we have excluded. Then AST was done. And finally, by identification was done by Vitek-2. Few antimicrobials we have done that was not in that Vitek system card by Kirby-Board diffusion method, and by this Vitek report, if the non-fermented showed carbapenem resistant, we have preserved those samples for our further molecular study. So these are the results. This is a gram staining of A. baumannii.
Ma'am, I'm sorry to interrupt you. You just have about 1 minute. So please
Okay.
wrap the results and discussion in
Okay,
1 minute.
okay, thank you. So this is A. baumannii on blood and McConkey agar. Total out of 200 samples, 16 were positive for A. baumannii, and as you can see in bi diagram, that one is in male or female ratio, one is one female. This is a pie chart where you can see 69% elderly patients were developed ventilator associated pneumonia, because A. baumannii is an opportunistic pathogen and only infected immunocompromised patients. This is the AST profile. As you can see, this Acinetobacter is resistant to almost all antimicrobials. Only it is susceptible, intermediately susceptible to colistin, and 50% were susceptible to minocycline. So that's a worrisome situation. In our institute, infection control measures was taken very carefully. This antimicrobial stewardship awareness program is going on, surveillance is going on, and we have separate biohazard waste management awareness program is going on for the students, for the patients, for the relatives. And this is my take-home message, that prevention strategy could be, and we have to follow antimicrobial stewardship, new innovative research work like nanopore delivery, and other vaccines therapy, fast therapy could be done. Screening and molecular surveillance is must. And most importantly, patient transfer protocol should be done. If a patient is transferring from one hospital to another, then we must inform the other hospital that what kind of, if he is carrying any resistance genes. International collaboration is must. And we all know about the glass, which was introduced by WHO in 2015. India is also a part of it. And most importantly, our keynote speaker, Dr. Bhardwaj and Dr. Renu, Dr. Jugraj has told us about One Health approach. So we must follow it. This is my conclusion. So we have found a low prevalence, 8% in our hospital. Few years ago, it was very high, because our hospital infection control committee has a strict taking a strict measure to control this resistant. But as you have seen, that almost all strains are resistant to all antimicrobials. So it is therefore recommended that critically ill patients should be treated after screening for drug resistance to minimize the epidemiological outbreak, and this patient should be isolated from other patients. I would like to acknowledge my husband, Dr. Prashant Sevta, and my son, Gurupreet, for their continuous support. I would like to acknowledge Professor R.G. Sani and all the other staff of CIBR faculty. Thank you so much. These are references I have used for it.
How many samples you have collected, ETA and BAL?
Ma'am, total we have collected 200 samples. Total
So you
we
didn't
have
divide. Total 200. You didn't divide how many are TA and how many are
no,
BAL?
total we have collected total 200. We didn't divide that half we will do that or half we will do that. And
Okay.
yes, and after ma'am, let me out of this I am not able to see.
No, you didn't mention there. That's why I am asking you.
Okay.
And another
No,
thing
ma'am.
is
No,
it
we
a
have
conventional method you have followed or automated method?
Okay, ma'am. Actually, we have seen previous study. Okay, as I have told earlier, that VAP rate is defined from hospital to hospital. So previously, it was a 15 non-fermenters was 15%. So what we have done that our target was to we have followed Daniel formula, and by following Daniel formula, we have to collect 200 samples that may collect that may be this non-fermenter. So we didn't divide that half we will done that or half we will collect bulk. We have totally collected 200 samples.
So did you follow conventional method or automated method?
Okay, we have conventionally we have inoculated like blood. We have inoculated on a blood agar and McConkey agar, but automated method we have used Vitek 2 system. Few antimicrobials like polymyxin that was not included in that card, so we have done that by Kirby-Ball diffusion method.
So you have done antibiotic susceptibility testing with the Vitek cards.
Vitek card, and if few antimicrobials that were not present in that card, we have done by Kirby-Ball diffusion method.
Okay, majority of with the Vitek and a few antibiotics which are not tested with the Vitek, you have done with the Kirby-Bar disc diffusion. That's it?
हाँ जी मैम जी हाँ जी
Okay.
मैम
Okay, thank you.
Thank you मैम.
Thank you. I invite the next presenter, Ms. Tanushri Sharma. Yes, ma'am. Please share your screen. Am I? Yes, ma'am. Is the presentation visible, ma'am? Yes, it's visible. Is the presentation visible? Yes, it's visible. Please go ahead. Okay. Good afternoon, everyone. I am Tanushri Sharma, and from Bhavansi Vivekananda College, Hyderabad, and I am here to present my master's project on microencapsulation of secondary metabolites isolated from leaves of Osmum sanctum, its synthesis and characterization. So we know that natural bioactive compounds offer a potential therapeutic source with their influence on oxidative stress, which is caused by high concentration of free radicals in our body. Plants like Osmum sanctum or Tulsi, as it's known, has shown pharmacological activities in anti-cancer, anti-inflammatory, and more. That was the reason for the selection of this plant. Studies have also shown that microorganisms like fungi and bacteria have a very close relationship with medicinal plants.
Endophytic microorganisms that reside within the tissues of various plant parts can act as an important source of these bioactive compounds, such as alkaloids, phenols, and more in the form of secondary metabolites produced by them. Most of these metabolites, however, are sensitive to light, heat, and degrade at acidic pH, which is usually found in such as the stomach pH. To counter this drawback, microencapsulation can be used to protect and stabilize these polyphenols obtained from natural extracts, creating a possible delivery system. So based on the data so far, our hypothesis was as follows. First, endophytic bacteria isolated from Osmum sanctum leaves should have the same capacity to produce bioactive compounds as the whole plant leaves. The second was that the compounds can be immobilized through microencapsulation. So to achieve our project...
Slides are not moving, Tanushree.
Sorry, ma'am?
Slides are not moving. It's in first slide.
Now?
Yes.
Okay. So our project aim was to find an alternate source of bioactive compounds, and the other aim was to create a delivery system for them. To achieve our aims, our objectives were first to isolate the endophytic bacteria from the leaves and identify the organisms. The second was to estimate the antioxidant potential of these secondary metabolites obtained from the isolates. And the third was to immobilize these metabolites using microencapsulation. This is a project overview. First, the endophytic bacteria were isolated from the leaves. We use gram staining and various biochemical tests for basic classification of the bacteria. And after the secondary metabolites were obtained, we conducted antioxidant tests and synthesized the microcapsules, which were later analyzed through SEM and FTIR analysis. We also determined the encapsulation efficiency through biochemical tests methods. So to obtain the so we isolated the endophytic bacteria by surface sterilizing domestic white tulsi leaves with 99% ethanol. And after plating, serial dilution plating, we obtained 4 distinct colonies, which were then subcultured. We maintained control plates throughout this to eliminate the possibility of contamination there. For the classification, we used gram staining, catalase test, and Voges-Prosker test. We also conducted the indole test and citrate agar test. We checked for the ammonia and hydrogen sulfide production as well. For the ammonia utilization test, 0.3% arginine and 0.2% sodium citrate was used instead of the conventional ammonium citrate. We also did the starch hydrolysis test by iodine flooding of the. For all these biochemical tests, we use 24-hour grown cultures. For our second objective, we centrifuged a week-old culture and used the supernatant for the antioxidant tests, out of which the first was the total phenol concentration test using the phenol reagent. The second was the DPPH test we used, and the last was the reducing power assay. For the second 2 tests, we used ascorbic acid as the standard. For our final objective to synthesize microcapsules, we mixed our sample supernatant with 1.5% sodium alginate. And then we created a water oil emulsion using commercially refined soybean oil. And upon slowly adding 2 microliter of the 1.5% calcium chloride to the emulsion, we saw the formation of white beads immediately. These beads were collected, washed, and then coated with 1% chitosan. Using SEM, scanning electron microscopy, we analyze the size and morphology of the microcapsules, and FTIR spectrophotometer showed us the chemical composition and interactions in the capsules.
I'm sorry to interrupt you, Ms. Tanushree. We just
Yeah.
have about a minute, so please wrap the results
Okay.
and discussion.
Yeah, I'll quickly. Okay, so this shows the biochemical tests and classification tests that we did. Based on this, we see that TI3 showed significant catalase activity, but TI1 showed the better ammonia production and arginine degradation. But none of these isolates showed the indole, a positive test for indole or nitrate reduction tests. For total phenol concentration, we use catechol as the standard, and the concentration of phenol of the isolates is expressed in mg per ml. Here, osmium sanctum crude leaf extract showed the highest concentration, followed by the TI2 isolate. For determination for the results of determination of free radical scavenging activity, we saw that the crude extract and TI2 showed an increase gradual increase in activity up to 100 microgram per ml of the concentration, but it declined after that. And the others only showed high activity at such smaller concentrations. This could possibly be because of a threshold concentration, which is seen here. In the reducing power, a reducing power assay showed that there's a gradual increase in the reducing power activity with increase in concentration of the bioactive compounds. Our SEM analysis of the microcapsules showed an average size of 2.4 microgram for the 2 microliter of the sample inoculated, encapsulated. And the FTIR...
Tanushree, I'm sorry to interrupt you again, but I'm going to have to ask you to wrap it up. So please jump to the conclusion.
Okay. So our FTIR analysis basically showed that the chemical interactions that could be caused due to the formation of the microcapsule, and previous work by D'Souza et al. Also showed that phenols are present based on the wave number reported there. The microcapsule encapsulation efficiency without the microcapsules without the chitosan coat showed a lower efficiency at 14.1%, but with the coating, it showed 53% efficiency. In conclusion, these endophytes that were isolated could be potentially exploited as a source of bioactive compounds. And whereas it comes to microencapsulating or immobilizing these phenols or bioactive compounds, chitosan offers more stability to the capsules than plain sodium alginate capsules. I would like to thank the Department of Biochemistry and Nutrition at Bhavans Vivekananda College for the opportunity to conduct this research, and my guide, Vanita S., for her guidance throughout. A special thank you to the University College Technology, Usmania University, for the SEM and FTIR results. These are the references I've used. Thank you.
Your slides preparation is nice, but you should confine to the time.
Yes, ma'am.
Yes.
I thought I would be able to finish, but it took longer than I expected.
Okay, next part.
Thank you.
Okay, thank you, ma'am. And also just to reiterate, like Katyani ma'am suggested earlier, please, I know you have already prepared the slides and it took a lot of effort. I completely respect that. But you have already been informed of the time limit. So for 8 minutes, I would also suggest that you plan it wisely, and so that you wouldn't miss out on the important results and discussion part. And as you know, everybody's presentation is evaluated for the awards that we are providing. So make sure you stick to the time, make sure you, as much as possible you stick to the template that we have provided, because everything will play a role in that final score points. So on that note, I want to invite the next presenter, Ms. Mayuri. Hi.
Yes, ma'am.
Ms. Mayuri, are you there? Yeah.
Can you see my screen, ma'am?
Yes, I can see. Please go ahead.
What's oh my okay. Good afternoon, everyone. Myself Mary. I'm a research scholar from Shri Guru Ram Rai University, Dehradun, Uttarakhand. My today's topic is review of a cancer multi-drug resistant molecular mechanisms and novel therapeutic strategies. It is based on a review paper. So firstly, we come to the introduction. Firstly, we should know about the cancer. Cancer is the second leading cause of death globally, with 10 million in 2020. Factors that are associated with the elevated risk of cancers are tobacco use, lack of physical activity, alcohol use, low vegetable and fruit intake, and a high body mass index. Breast, cervical, lung, thyroid, and colorectal cancers are the most common type of cancer in women, while prostate, lung, colorectal, liver, and stomach cancer are the most common cancer in a man. There are several different methods of cancer treatment, including radiation therapy, surgery, immunotherapy, endocrine therapy, and a gene therapy. But the chemotherapy is the type of treatment that uses drugs to kill cancer cells. Several chemotherapy drugs used in a clinics today, such as DOX, DNR, EPI, DOC, and DDP. These all are the example of drugs used to treat cancer, but sometimes they can be ineffective due to drug resistance. So as the term here comes, a drug resistance, so we should know what is a chemo resistance. Chemo resistance which can lead to the rapid reoccurrence of cancers and ultimately patient's death. Depending upon the mechanism of actions commonly used, chemotherapeutic agents can be divided into several classes, like as shown in the pictures: antimetabolites, alkylytic agents, mitotic spindle inhibitors, topoisomerase inhibitors, and others like enzyme antibiotics, protease inhibitors, tyrosine kinase inhibitors. Multi-drug resistant is when cancer cells become resistant to many different types of chemotherapeutic drugs. This means that the drugs are no longer effective in killing the cancer cells, which can be very harmful for patients because it makes treatment less successful. MDR of cancer cells during chemotherapy can be associated with a variety of mechanisms, including enhanced effects of drugs, genetic factors, example, gene mutations, amplifications, epigenetic alterations, and growth factors, increased repair capacity, and elevated metabolism of xenobiotics. Now we come to the aims and objective. Here in this review, I am aiming to provide a comprehensive overview of the complex phenomena of the cancer multi-drug resistance and explore potential solution to overcome this major obstacle in cancer treatment. Firstly, I will talk about the elucidating the molecular mechanism underlying MDR. Second, identifying and evaluating novel therapeutic strategies to overcome MDR. Third, highlighting the importance of personalized medicines in optimizing cancer treatment. Fourth, addressing the unmet needs and a future direction in MDR research. Here in this diagram, it is clearly showing that the targeting multi-drug resistance cancer, where the P glycoprotein actively extrudes many types of drugs from the cancer cells, keeping their intracellular level below a cell-killing threshold. Strategies that circumvent PGP-mediated multi-drug resistance include the common co-administration of pump inhibitors and a cytotoxic agents, which is shown as an engage, and the use of cytotoxic agents that bypass PGP-mediated effects, as shown as evade. And the third approach takes advantage of the collateral sensitivity of MDR cells, which is shown as exploit. Now we come to the neoplastic resistance. Cancer cells also have the ability to become a resistance to multiple different drugs, which become because of increased effects of drugs. As by P glycoprotein, as I have mentioned in earlier slide, multi-drug resistant associated protein, breast cancer resistance protein, and a reproductive cancer resistance protein. Now we'll talk about the mechanisms involved are increased effects of drug, enzymatic deactivations, decreased permeability, alteration in binding sites, development of alternative metabolic pathways. Here in these diagrams, we have shown the mechanism of chemotherapeutic drug resistance in a cancer cells. As I have talked right now. Now we will come to this figure where it is clearly showing the drug resistant mechanism in a cancer, where the cancer cell develop resistance to anti-cancer agents through various mechanisms such as diminished drug uptake, as we have shown with the right hand side of the arrow, and then the enhanced drug enhanced drug effects, improved DNA damage repair, resistant to cellular alteration of drug metabolisms, alteration of drug target, epigenetic changes, and the target gene amplification. These mechanisms act either individually or in a combination, leading to the development of single or multiple drug resistance in a cancer cells. So the cancer patients can be treated by the two methods, local and a systematic. Radiation, surgery are considered local treatments, whereas a chemotherapy, hormone therapy, and a targeted therapy are considered a systematic treatment. Systematic treatment are especially effective against metastatic or a late phase cancers. Growing evidence suggests that MDR is mediated by the increased effects of chemotherapeutic drug, which reduces the drug absorptions by cancer cells. The mechanism of MDR may also be mediated by the release of drugs outside the cell, as it is showing in the left side of the arrow. And MDR may develop due to the oncogene mutations, changes in the tumor microenvironment, tumor heterogeneity, target site mutations, or epigenetic changes. Now here it is showing the resistant cell and a sensitive cell, and this is a after that there is a mixture of sensitive and a resistant cell, and after the chemotherapy there is a surviving resistant cells which regrow, and there is a resistance cell regrow to produce a resistance tumor. And in this second figure, it is showing that PGP mediated MDR donor cell, and where there is a PGPA as a receptor and an MPA release.
Sorry to interrupt, ma'am. Your 8-minute slot is actually over. Please try to wrap the presentation in 1 minute.
Okay, ma'am. Okay. And here it is showing the multi-drug resistant in a cancer mechanism overview. By a different methods like increase of drug effects, DNA damage repair, inactivation of anti-cancer drugs, inhibition of cell death, cancer stem cells, and other different methods. And here it is shown that I have given this one example, like the nanomedicines to overcome a multi-drug resistance mechanism in a colon and a pancreatic cancers. I will tell you in a short that this reviewed the main resistant mechanism in colon and pancreatic cancers, along with the most relevant strategies offered a nano drugs to overcome this obstacle. Now here's a summary. Cancer multi-drug resistance is a major obstacle in a cancer treatment, significantly reducing the effectiveness of the MDR involved diverse molecular mechanisms, leading to a reduced drug accumulation, increased drug effects, and enhanced DNA repair. I will, in short, I will tell due to the complicated nature of the MDR mechanisms and the heterogeneity of tumor disease, there may never be an individual drug that can be used for every type of cancer. Therefore, further efforts are needed to investigate the mechanism of cancer drug resistance and identify there are currently unknown vulnerabilities to design novel potential chemotherapies. In acknowledgement, I would like to thank my guide, Dr. Neha, and BioLEAGUE for giving me this opportunity. These are my references. Thank you.
Did you do any study regarding this?
Yes, ma'am. Yes, ma'am. I'm doing right now. I'm doing the research on this cancer only, MDR cancer.
Okay. You just presented a presentation, now. It's not a
Yes.
study, I think.
No,
Just
ma'am. This presentation is based on totally on a review paper.
yeah,
But
review
right,
paper. Yes,
yes,
this is
ma'am. Right now I'm
taken from a
working on.
review article. Yes.
Yeah, this is based on a review, but right now I'm working too on this.
Yeah, what is your work? That's what I'm asking you.
Ma'am, basically my work is on a multi-drug resistance bacteria, and
you didn't focus that one, huh? You have presented a few things from the review. Yes. What is your work?
Here I have
Professional
talked.
work.
Here I have talked about the MDR in a cancer because in my research, in my PhD research, I am studying on that how multi-drug resistance can cure is just not helpful for the sorry, how to overcome cancer drug resistance with the help of medicinal plants.
Okay. What is your sample size?
Ma'am, right now 200.
200. How many you got?
Ma'am, approx.
So you didn't start yet, I think. Yes.
No, ma'am, I've just initially started.
Uh-huh.
I've just started. It's been 6 months only.
Okay. Next presenter.
Thank you, ma'am. Thank you so much.
Thank you, Ms.
Next
Margaret.
presenter.
I would like to invite the next presenter, Ms. Hemlata JK, for your presentation. Yes, ma'am. Am I audible, ma'am?
Yes, you are audible.
Good afternoon, all. Good afternoon, Katyani, ma'am.
Good afternoon.
Myself, Hemlata from Guntur Medical College, final year PG. And my topic today is a comparative study of diagnostic procedures for dengue in tertiary care center, Guntur. My co-author is Dr. K. Parmeshwari ma'am, professor and head of the Department of Microbiology, Guntur Medical College, Guntur. And coming to the introduction, dengue is the most common, rapidly spreading mosquito-borne arboviral disease in the world. It is also called breakbone fever, saddleback fever, and global prevalence of dengue has grown dramatically in the recent decades as a public health emergency. According to WHO, dengue fever or dengue hemorrhage fever is considered as the second most important tropical disease next to malaria. Dengue viremia is the most important cause of acute febrile illness, causing serious morbidity and mortality. And dengue virus has 4 different serotypes, dengue 1 to 4, and recently the serotype was discovered in 2013 in Bangkok. Dengue classified into dengue fever, dengue hemorrhagic fever, and dengue shock syndrome. Secondary dengue causes more severe disease than the primary one. Hence, early diagnosis of dengue is essential to prevent the complications. My aims and objectives are to detect the NS1 antigen and
Yes,
antigen.
நடக்குது மீட்டிங் உள்ளே போட்டிருந்தானே என்ன 14 and 15.
Ma'am, please go ahead. There was some person who did not put them. Yeah, please go ahead. Hello, ma'am.
Continue.
Okay, ma'am. My aims and objectives are to detect the NS1 antigen and IgM antibody by ELISA in patients with fever, and comparison of NS1 antigen and IgM antibody by ELISA. My materials and methods are: my study place is Guntur Government Hospital, Guntur. A study group is sample size with 1478 sera samples of suspected dengue cases, and study period is July 2022 to January 2023. And here, the blood samples from the IB patients were collected in 2 tubes under aseptic conditions. The serum was separated by centrifugation, and one serum sample is tested for dengue NS1 antigen by ELISA, Biorad Platelia TM
dengue NS1 antigen kit, and another serum sample is tested for IgM antibody detection by ELISA, NIV dengue IgM capture ELISA kit. My inclusion criteria include the clinical basis for the inclusion criteria is febrile illness, patients with presenting with febrile illness of more than 9 days with features like headache, myalgia, arthralgia, rash, hemorrhagic manifestations, and leukopenia, and also thrombocytopenia. Exclusion criteria include febrile cases without the above symptoms and the patients with clinical evidence of urinary tract infection, pneumonia, abscess, or any other apparent cause of fever were excluded in this study. The results include a total of 1,478 samples were included in the study, and among them, 360 were tested as positive and accounted for 24.4%, and with only NS1 antigen positives are 110, accounting for 30.4%, and only anti-IgM antibody positives are 63 with 17.4%, and both NS1 antigen and IgM antibody positives are 190, accounting for 52.2%. And here I have depicted the pie chart with 52.2% as 52.2% as both NS1 and IgM antibody positives, and 30.4%. And this is the gender-wise distribution now. Male preponderance is higher with 65% and females with 35%. And coming to the sensitivity and specificity of the tests, NS1 antigen is more sensitive. NS1 antigen ELISA is more sensitive with 87.5% and specificity is 82.5%. And IgM antibody sensitivity is 80% and specificity is 79.7%. And the combination of the NS1 antigen and IgM antibody is sensitivity is 92.3% and 87.6% is the specificity. Coming to the discussion, the present study was carried out in 1,478 samples. In the present study, the percentage of positivity of NS1 antigen or NS1 antigen, IgM antibody, and NS1 antigen and IgM antibody was found to be 30.4%, 17.4%, and 52.2% respectively, which was similar to the study conducted by Dharitri et al. Incidence of dengue infection was most common in males, which was similar to the study conducted by Kinalsha et al. And the sensitivity and specificity of NS1 antigen is 87.5% and 82.5%. And for IgM is 80% and 79.7%. NS1 plus IgM antibody is sensitivity is 92.3% and specificity is 87.6%. Respectively, in our study, Deepak et al. Observed sensitivity and specificity of NS1 antigen is 83.58% and 94.82%, and for NS1 antigen plus IgM antibody is 95.55% and 79.31%. The sensitivity and specificity of IgM antibody is similar to the study of Dharitri et al. The conclusion is dengue NS1 antigen ELISA is an effective method for diagnosis in early stage. Early detection of dengue fever will help in treatment at appropriate time actually to prevent the patient going into the dengue hemorrhagic shock syndrome. And NS1 antigen ELISA has highest sensitivity than IgM antibody. The study provides strong evidence of value combination of both antigen and antibody based tests for diagnosis of dengue fever. And this is the take home message to stop dengue. It's block means break up the hardened soil in the pots and all, lift and empty the flower pot plates, overturn the pots and wipe their rims so that the water will not be clogged, change the water in the pipes, vases, and keep the roof gutters clean and place any insecticide to prevent the water clogging. So that is one of the prevention control for dengue. And these are my bibliography, ma'am. Thank you.
Nice, you have confined to the time. One thing is you can do dengue NS1 antigen from day 1 of the fever.
Okay.
Next presenter.
Thank you. Thank you, Ms. Hemlata. For the next presentation, I would like to welcome Dr. Edla Sujata from Kakatiya University. Sujata ma'am, are you available?
Hello, ma'am. Am I audible?
Yes, ma'am. You're audible.
Okay, ma'am. Good afternoon, one and all. My research area is isolation and identification of nanoparticles from endophytic fungi. Then, as my topic is characterization of silver nanoparticles from endophytic fungi. The resume occurs as enlighten, and then a study of its antimicrobial and anti-cancer effects. Okay. In recent times, then nanoparticle application is tremendous. Then it is gaining much importance because of its wide range of applications in different areas. Then those are being used as signal enhancers in electron devices, optical sensors, biomarkers, then many more. Then because of its high penetrative power, then we are using these particles as best bacterial and an antifungal agent. Hence, research studies on nanoparticles progressed steadily nowadays. That too, silver nanoparticles have much applications. Then here, the experimental organism is Mucorsis nowadays. We have synthesized nanoparticles from this fungi, actually, which has been identified, isolated and identified from plant that is noni plant, that is Indian mulberry, that is Morinda citrifolia. Then here, after a synthesis of these nanoparticles, we have gone for characterization of these particles by using SEM, TEM, and UV spectroscopy. Then after those were subjected to characterize for their antibacterial, antifungal activity, and then cytotoxic activities are also we have studied. And here, regarding materials and methods, actually, we have collected many medicinal plants. Then we have isolated different fungi. Here, we are presenting the endophytic fungi, which is isolated from that Morinda citrifolia. Then isolated in pure culture by using serial subculture mechanisms. Then morphological identification we have done based on their sporangiospore arrangement, and then branching of sporangiophores and all those. Then we have gone for molecular identification by extracting its DNA, then PCR method. Then we have gone for biosynthesis of these silver nanoparticles. Then those synthesized particles were subjected to SEM analysis, STEM analysis, and FTIR analysis. Then EDX also. Then those particles were used to study their antimicrobial activity, and then their cytotoxic studies were conducted on 2 cell lines, that is MCF 7 cell lines and HCT 116 cell lines. Then here these results, regarding the results and discussion, here those nanoparticles were subjected to the this UV-based spectroscopic analysis. Here we got a typical peak at 440 nanometers. Then the change of color here we can see after incubation of 48 hours. Those culture filtrate may totally turned as dark brown colored, indicates for the reduction of sulfur by addition of silver nitrate. Then here we are showing the results of UV spectroscopy. Then here, those nanoparticles were subjected to FTIR analysis. Then here, FTIR has been performed to know their functional groups which are present in those components. Here, we have obtained the bands at different wave numbers. These actually indicates the presence of different functional groups. Then these nanoparticles were also subjected to EDAX analysis. With it, we can detect the silver percentage in nanoparticles, that is 66.64. In addition to the silver element, we have seen oxygen, phosphorus, sulfur, chlorine, calcium, and other elements also. Here, those particles were subjected to those antimicrobial effect. In those, actually, this agar well diffusion plate technique we have selected. Then we have used to control this gentamicin. Then here we have repaired the wells, and then we have loaded the wells with this nanoparticle suspension of this mucorcecinolloides. Actually, this nanoparticle suspension has shown moderate inhibitory zone on pathogenic microorganisms. Here we can observe silver nanoparticles have shown 3 mm zone of inhibition against Staphylococcus aureus and 6 mm zone of inhibition against gram-negative bacteria, that is Proteus vulgaris, and it has shown 5 mm zone of inhibition on Micrococcus luteus. Like that, we have selected some gram-positive and some gram-negative bacterial strains for a study. Then antifungal activity was also tested on human pathogens, that too on Candida species. Here we have selected 2 Candida species. Those are Candida albicans and Candida glabrata. Then those have actually produced 3 and 4 nanometers respectively. Again, as albicans, it has shown 3 mm of zone of diameter, zone of inhibition, and 4 mm of zone of inhibition on glabrata. Then in the same way, here the suspension nanoparticle extracts were subjected to study their cytotoxic activity against 2 cell lines, MCF7, then HCT116. Then here these 2 are different cell lines. Then cytotoxic activity was shown. Very good activity has been shown. Cell viability of here the cells 55.36 was recorded with 6.25 mg per ml concentration, whereas the cell viability has drastically reduced to 2.36 percent at the concentration of these nanoparticles has been increased per 100 mg per ml. Then with that, we have calculated IC50 values. It was recorded as 5.47 mg per ml. Then here you can observe these diagrams, whatever we have conducted in our laboratory. Then these cell killing capacity at 24 hours, 48 hours, and then 72 hours. Then here we have compared this nanoparticle cytotoxic effect with this general suspension, nanoparticle suspension. Here at 24 hours, we have seen this much. At 48 hours, at 72 hours, we have shown considerable cytotoxic activity. Okay, then here it is the fungal extract. Then this is actually silver nanoparticle suspension. Then we have actually done with crude extract and then silver nanoparticle extracts. Then here in the same way, actually we have gone for study of these silver nanoparticles which are extracted from Micrococcus mucorcecinolloides. Then here cell lines HCT116 cell lines we have used. Here the cell viability was decreased to 3.68 at 100 mg per ml of sample. IC50 value was recorded as 14.62 mg per ml. Then here we have presented these IC50 values for both strains. At 72 hours, the silver nanoparticle concentration has shown, the silver nanoparticles has shown recommendable then cytotoxic activity. Here also we can observe the considerable cytotoxic activity of silver nanoparticle suspension on HCT116 cell lines. Then here the overall summary of my research work is the silver nanoparticles which are synthesized by endophytic fungi have been characterized well. Much importance is there for these nanoparticles. The TEM and SEM have revealed here the SEM-TEM analysis. Here you can observe these diagrams. Then these Symptom analysis have shown the shape and size of the silver nanoparticles. These have shown approximately the range is 10 to 20 nanometers, but all the particles have not shown the same size. Then here it is very clear. Then surface structure of these nanoparticles was also revealed here very clearly in SEM and TEM analysis. Then here the results of SEM and TEM have revealed the nanoparticle size and shape, that is average size is 10 to 20 nanometers. The results of XRD, EDAX, and FTIR confirm the crystalline nature of these biomolecules. Then here particularly EDAX analysis shows the elemental composition of these silver nanoparticles. Then these silver nanoparticles have shown considerable antimicrobial activity against the pathogenic bacteria, especially on gram-negative and gram-positive bacteria. Then here anti-cancer activity is
Ma'am, I'm sorry to interrupt you, but the time limit is over.
Over? Okay.
Yeah. Thank you.
Okay, thank you. Is there any query, madam?
Nothing. Very nice.
Okay, ma'am. Thank you.
Thank you, ma'am. I invite the next presenter, Mr. Purna Chandra Rao, for the presentation. Sir, you're on mute, sir. Can you please unmute yourself and share your screen?
Okay, ma'am. Now it is okay? Is it okay, ma'am?
No, sir, I cannot see the screen.
Cannot see the screen. 1 minute, ma'am. I'm coming. 1 minute. Am I appearing to you?
I can hear you, sir. I'm not able to see your PPT, sir. Can you please share the screen?
Wait. I am going to present. Now, am I appearing?
You are appearing. Share your screen.
Okay, ma'am. Okay, ma'am. My screen. Is it, ma'am? Ma'am, is it appearing? My screen?
No, your screen is not visible.
Screen is not visible. 1 minute.
You can go for the next presenter, Pauline. So that he
Ma'am,
will.
1 minute, ma'am. Is it
Yes, sir.
appearing?
It's not appearing, sir. I'll just give you 5 minutes to find out what the issue is. In the meantime, we can finish the other presentation. Right after that, we'll give you your turn, sir. I think Dr. Meghna is there. So we can just go ahead with her presentation and we can come back to yours, sir.
Okay, ma'am. Sorry.
Dr. Meghna?
Yes, good afternoon, ma'am.
Good afternoon, ma'am. Please begin the presentation.
Yes. Is it visible?
Yeah.
Okay. Good afternoon. So, I Dr. Meghna Diasa and I'm working at Charusat University. And my topic is antibiogram of Enterococcus isolated from clinical samples in tertiary care hospital, India. Enterococcus is a group of gram-positive, non-motile, non-performing aerobic and facultative anaerobic bacteria. It is, although it is a normal flora of human intestine, but it become an opportunistic nosocomial pathogen due to the antibiotic resistance. And the Enterococcus is mainly responsible for UTI, bacteremia, endocarditis, meningitis, and rare intra-abdominal infections. More than 38 species have been described, and among these species, Enterococcus faecalis and faecium is mostly pathogenic and infectious species. So nowadays, as enterococcus is resistant against wide range of antibiotic, especially beta-lactams, aminoglycosides, and glycopeptides, so it become more difficult for medical as well as scientific research to discover alternative therapy or the new medicine for the elimination of enterococcus infection. In order to logical support patient therapy, this study is being done to ascertain the distribution of enterococcus species from diverse clinical samples in tertiary care hospital, Karamsad, India. This study will also add in the understanding the necessity of control measure that must be put in place to address potential therapeutic difficulties brought on by multi-drug resistant enterococci. As enterococci is multi-drug resistant pathogen, so continuous surveillance is an essential tool for understanding for magnitude and future trend of AMR infection, as this information can be incorporated and developed into future strategic plans for antibiotic usage and infection control procedure. Next is my material method. So in our study was conducted at microbiological section of CDL, Sri Krishna Hospital, Karamsad, and the study was approved by Institutional Ethical Committee, Bhakaka University. The clinic in our study we use 5 different clinical samples, that is blood, pus, urine, nasal secretion, and endotracheal. As per the standard condition in under aseptic conditions, all the samples were inoculated on nutrient agar plate, McConkey agar plate, and clay, and incubated for 37 degrees Celsius at 37 degrees Celsius for 24 hours. And the identification of the enterococci was done by gram staining method, colony characteristics, various biochemical tests, and further confirmation was done by the further species confirmation was done by Vitek 2 automated system. We also perform AST antimicrobial susceptibility test by Kirby Body's diffusion method as well as Vitek 2 system also. In Kirby Board method, we use MHA plate, and suspension was inoculated on MHA plate, and plates were covered with different antibiotics and incubated for overnight at 37 degrees Celsius. And the CLSA guideline, Clinical and Laboratory Standard Institute guideline, was used to interpret the results. So in our 7-month study, we got total 82 enterococcal samples, and we selected the patient from age 21 to 90. And here result shows that 30%, that is the enterococcal infection is highly present in the male as compared to the female, and 30% males are, that is which falls under the age group of 61 to 70 are affected from the enterococcal infection. In our study, we use as 5 different clinical samples. So among that 5 clinical samples, maximum, that is 54% enterococcal species isolated only from the urine sample, 32% enterococcal sample species isolated from the blood, followed by pus, and only 1% isolated from the nasal secretion as well as from the endotracheal sample. In our study, we got 5 different species of enterococcus, that is Enterococcus faecium, Enterococcus fecalis, Enterococcus gallinarum, Enterococcus caeciliiflavus, and Enterococcus raffinosus. So among out of these 5 species, maximum, that is 50% Enterococcus faecium was isolated, and 37.8% Enterococcus fecalis we got. And the table is also shows that the Enterococcus faecium maximum 56.10% Enterococcus faecium is obtained from the urine sample. This table shows the resistance pattern of Enterococcus species. So as we can see that the Enterococcus species are highly resistant towards many drugs, but maximum resistance shows Enterococcus faecium maximum resistance, that is 78.8% shows towards the ampicillin, and 60.97% shows against linezolid, and Enterococcus fecalis shows shows 87.9% resistance towards the high level streptomycin, and next 70.96% for the ciprofloxacin. And when we talk about the susceptibility, so Enterococcus faecium and fecalis both shows susceptibility towards high level gentamicin. Our results also we also compare our result with other research finders. So we also find that Suman Gala et al. 2020, they also find the similar result, and we got maximum amount of Enterococcus from the urine sample. This may be due to the proximity of the urethra and anus in the perineum. So our research also conclude that Enterococcus has a great ability to survive under the harsh environment in the hospital. The antibiotic stewardship and measurement of infection control is required to prevent or slow the emergence of multi-drug resistant enterococci in healthcare. The current study presents the prevalence and antimicrogram pattern of the enterococcus isolated from various clinical samples. The overall study concludes that the Enterococcus fecalis is more resistant as compared to the Enterococcus, and this study proved that Enterococcus infection is a nosocomial infection and also show a resistance against the wide range of antibiotic. And this finding create a challenge to reduce the infection of Enterococcus as well as the resistance towards the antibiotics. And these are the few references that we use in our study. Thank you.
In what conditions did you collect nasal secretions? In what conditions did you collect nasal secretions?
Hello.
Yeah, in what conditions, ma? Yes.
Hello.
So you are not audible. How did you speciate Enterococcus?
Sorry, ma'am.
How did you speciate ma Enterococcus?
We use Vitek machine to for the confirmation of species.
So I used automated methods.
Yes, automated method. Yes. And those antibiotic which are not available for the Vitek 2 machine, for that we use Kirby-Board disk diffusion method.
Any special reason you have not used blood agar?
No, ma'am.
Then?
Actually, at that time, my student, this work is with my students, so my dissertation students.
Okay, next part.
Yes, thank you.
Thank you, ma'am. I would like to invite Purna Chandra sir. Is the issue sorted, sir?
Is that my audio is visible, ma'am?
We can hear you, sir. Video is not visible.
Okay, ma'am. This is Purnachandra Rao. I am
Sir,
from
your
Ambedkar
PPT is
Medical.
not visible, sir. Do you
Yeah,
want to go without the PPT?
No, with PPT only, ma'am.
We
Not
cannot see
PPT,
your
it is a
slides.
poster. It is a poster, ma'am. Yes, ma'am. I am coming.
Okay.
I'm doing screen sharing, ma'am.
Palin, what about the others who are missing in the middle?
Ma'am, like two people mentioned that they are not available because of an emergency. I saw Chandralekha ma'am had joined, but I think she had also had to disconnect. Just one more presentation, ma'am. In that case, only the last presenter is pending.
Okay.
After, yes,
You
sir.
can go, no?
I'm sorry, ma'am. Can you please repeat that?
You can go with the last presenter.
Okay.
Ma'am, 1 minute. Ma'am, 1 minute.
Go without presentation also.
Yeah. In that case, okay. So you can just do it without the slides.
Without the slide, ma'am. Ma'am, my work is correlation of carbapenemase production and biofilm production among the clinical isolates of Acinetobacter baumannii. Ma'am, I have done this study in my Kolar Devraj Aras Medical College. It is a part of my thesis. One of the objective I selected for the presentation. So in this presentation, ma'am, I would like to share that my objective carbapenemase production in Acinetobacter baumannii. That test I have done with the phenotypic method. It is a carboacineto-NP method, ma'am. Okay, 190 clinical isolates I have collected from the patients, either IP or OP. Among that 190 isolates, I have, seen 158 strains are able to produce the biofilm production. Among that one, 108 strains are able to produce carbapenemase by carboacineto-NP method, and remaining 16 strains, ma'am, out of 32, 16% of the strains are able to produce the carbapenemase production in non-biofilm producing strains. Means in my study, I have taken two groups. One, it is biofilm producing strain group, and one is biofilm non-producing strain group. In biofilm producing strains, how much percentage of carbapenemase are there, and in non-biofilm producing, how much percentage of carbapenemase producing is there. Instead of taking genotypic methods, I have selected the phenotypic method, that is carboacineto-NP method. Any kind of carbapenemase we can detect in this method. In genotypic, if I want to take means, I have to select so many genes. There are so many different types of carbapenemases are there. OXA51, OXA23, like that, so many are there. So instead of that is little bit expensive for me. So I have selected the phenotypic method to detect this one. In this method, I have used TCP, ma'am, for biofilm production, tissue culture plate method. It is a 96 well plate method, as you know, I feel. And by using that method, I have detected the biofilm producing organisms in Acinetobacter baumannii. And detection of Acinetobacter baumannii I have done by using 10% lactose agar test, and that Acinetobacter baumannii has capability of growing at 42 degrees Celsius. So that criteria also I have taken to identify that one. After streaking, I have kept in the incubator by setting up to 42 degrees Celsius. It has been grown. So like that, by using and OF method one it is, and biochemical reactions, non-fermenter, as usual, regular biochemical reactions I have used. Especially, I use it to detect Acinetobacter baumannii, this 10% lactose agar. It will utilize the lactose and it will turn to yellow color from red color. So if it turned means we can detect it is Acinetobacter baumannii only. Like that, I have detected Acinetobacter baumannii manually, ma'am. And later on, carbapenem resistance I have done by using imipenem and meropenem, by using the CLSI guidelines only. Later on, to detect this one, carbapenemase enzymes, I used carboacineto-NP method. So finally, after doing these two groups of analysis, I found that in biofilm producing organisms only, carbapenemase production is appearing more percentage wise compared to non-biofilm producing organisms. So chi-square test I have used meant to compare the two groups. It is showing a 0.002% only. So I can say by having this analysis, group comparative analysis, that correlation between carbapenemase production and biofilm production, it is more in biofilm producing organisms, not in non-biofilm producing organisms. And statistically, it is significant. By using the chi-square test, I can tell this one. And the reason behind this one, to why it is showing in biofilm producing organisms, that one, I have taken another objective also, efflux pump also. And one gene I have taken, that is ABA-I gene. It is a auto-inducer synthase gene. That is in my further objectives, I can explain, ma'am. In this objective, I have taken this, in this presentation, I have taken this objective. One, it is biofilm production, and second one, it is carbapenemase production. These two I have done, ma'am, and I correlated by using the chi-square test. And in as a result, I got it is significant correlation, 0.002. And reason and all I can explain in the further future studies, ma'am.
Okay. What are all your clinical isolates? What samples you have collected?
Yes, ma'am. It is from respiratory, means sputum and endotracheal tube secretion. Later on, surgical wound. Most of the timings, surgical wound samples only I have got. Secondary respiratory tract infection samples I have got. And later on, VAP, ventilator associated pneumonia, that ET tube, those samples. And very
that in what samples you have got how many?
Very less, 1 or 2 only. Remaining, most probably it is from surgery department and from the medical department, that is from the ICU, ET tubes and sputum samples.
Okay. What is your reference for using 10% lactose agar?
Ma'am, I have seen that one in Coneman I have seen, and some old books reference I have seen.
Okay, next presenter.
Thank you, ma'am.
Thank you, sir. The fun presentation for today will be by Ms. Dugyala Yogita Bala.
Yes, ma'am. Am I audible, ma'am?
Yes, you are audible. Please go ahead with the presentation.
Okay. Is my screen visible, ma'am?
Yes, it's visible.
Good afternoon, everyone. Myself, Yogita Bala. I'm pursuing Masters in Microbiology from Usmania University here in Hyderabad. I'm here to present my project, isolation, chemistry, and cytotoxicity of marine well, which is collected in Rishikonda, Vishakhapatnam. As we all know, 70% of the planet is covered in water that consists of countless microorganisms with potential bioactive compounds which are yet to be discovered. They are given more attention since they are more stable than those products obtained from plants and animals, as they are isolated from extreme conditions such as high pressure, high salinity, etc. Various compounds like proteins, peptides, pigments, carbohydrates, bioactive compounds, probiotics, etc. are derived from marine microorganisms. Some compounds that are bioactive compounds that are derived from marine microorganisms are hemastatin, vancomycin, bonactin, etc. They are used in pharmaceutical industries for their potential applications. The objectives of the study were isolation and characterization of the particular bacterial strain to find out the optimum growth conditions at which the bacteria grows the best, identifying the compounds responsible for antibacterial activity by GCMS analysis, assessing antibacterial, anti-cancer, anti-inflammatory activities, and molecular identification of this bacterial strain by 16S rRNA sequencing. The sample is collected from Rishikanda beach. The water sample is spread on the Zobel marine agar plates procured from high media. We spread 0.1 ml of the collected sample, and these plates were incubated at 30 degrees centigrade for 24 hours. And the different pigmented colonies were obtained. Out of 19 isolates, we have picked up this pale yellow color colony and restrict onto the other Zobel marine agar plates to establish the pure cultures. And we have evaluated the optimum growth conditions, and the results were observed as the bacteria grows best at its 37 degrees centigrade, pH 8, ma'am. And we have studied the colony morphology and gram nature of the strain, and the blood hemolysis activity of the bacteria. It exhibits alpha hemolysis. We have done different biochemical tests, and antibacterial activity of this particular strain is tested against test organisms such as Proteus mirabilis, Bacillus cereus, Bacillus subtilis, MRSA, and Proteus vulgaris. Out of all these bacteria, this Bacillus horikoshi shows 17 mm zone of inhibition against Bacillus subtilis. We have done the antibiotic susceptibility test also using Kirby-Bauer method, in which the bacteria shows resistance towards methicillin. And for microbial identification of the isolate using 16S rRNA gene-based molecular method, we have identified the particular strain to be Bacillus horikoshi, and we used a TLC directed bioautography method for the GCMS analysis, in which there are different major compounds are said to be dodecan, eicosane, henicosane, etc. Out of all these compounds, dodecan showed 32% of peak area, which is the highest peak area exhibited. Dodecan is used as a major compound in kerosene and some other jet fuels. It is also used as a perfect alternative to petroleum derivatives. Eicosane is a natural substance found in Microcystis aeruginosa and other microorganisms. It is used in cosmetics, lubricants, and plasticizers. Henicosane has potential applications in pharmaceutical industries. For the anti-cancer activity, we have used MTT assay. We evaluated the anti-cancer activity of Bacillus horikoshi, the pellet lysate of Bacillus horikoshi, on the HeLa cell lines. It showed 94.58% of cytotoxicity using 12.5 mg per ml pellet lysate. And we compared this with the standard paclitaxel drug. We have also evaluated anti-inflammatory activity of this bacterial isolate using 10% human RBC suspension. We used SDS as the standard. Here, the Bacillus horikoshi exhibited more than 97% of inhibition percentage, and the values are compared with the SDS. As of my knowledge, there are no studies based on the evaluation of antibacterial, anti-cancer, anti-inflammatory potential of this bacteria, that too derived from the marine source. We are planning to explore further antifungal potential, anti-cancer activity of this particular bacteria against other cancer cell lines, and are also planning to evaluate anti-Alzheimer's activity by deriving the bioactive components. In the conclusion, I would like to say that the bacteria possesses certain therapeutic potential against and exploring this potential of this particular bacteria for its therapeutic properties could provide new insights into creating various ailments. Hence, this study is significant and has the potential to contribute to the development of novel therapeutic agents that can combat various diseases. I acknowledge, I'm extremely grateful to my guide, Dr. K. Shanti ma'am, and for guiding me throughout the project, and these are the references that we have used for this work.
Can you
Yeah,
zoom in your references?
References, ma'am.
References. Get it to the center.
Sorry, ma'am, the screen is stuck actually. Give me a second, ma'am.
Yeah.
Is it visible, ma'am?
No, it's not visible. Get them to the
References.
center. Yeah. I want to know the references that substantiate your study.
Now, ma'am.
Still zoom
Is
in,
it
ma.
visible?
Zoom in. Can you read them out?
Yes, ma'am. Karthikeyan et al.
No need of
Promising.
authors. Just read the title.
Okay, ma'am. Promising bioactive compounds from marine environment and their potential effects on various diseases. Published in Journal of Genetic Engineering and Biotechnology, and
Okay.
molecular characterization of marine bacterial isolates of Visakhapatnam coast, efficacy in dye decolorization and bioremediation of cadmium, published in Journal of Genetic Engineering and Biotechnology. Next one is membrane stabilization as a mechanism of anti-inflammatory activity of ethanolic root extract of Piper chaba, published in
Okay.
Clinical Phytoscience, ma'am.
So what are the points that are correlating with your study in these references?
Ma'am, actually, the anti-cancer activity we have done based on my ma'am's work, ma'am. She has done it on Micrococcus luteus, and it is explained in the uh, first references that I have stated, and anti-inflammatory activity was stated in third reference, ma'am.
No, what are the points that are correlating with your bacterium, Bacillus horikoshi?
Ma'am,
You
I
said now
couldn't
it's having antibacterial, anti-inflammatory, and cytotoxic activity even on cancer cells now. So what are the
Yes, ma'am.
points that have been that are correlating with your study in those references? Does they also isolate those, and they also stated the same?
Ma'am, they isolated other marine bacterial isolates, and from them they have extracted these bioactive compounds, and then they have performed the study.
Then how can those be your references? Your references should be your pertaining to your marine bacterium, no?
They have isolated marine bacteria, ma'am, but they have isolated these bioactive compounds which show anti-cancer activity from these marine bacterial isolates.
Yes, I
We
understood.
didn't extract bioactive compounds solely, ma'am. We just used the pellet lysate of the marine bacteria.
No, what I'm asking you, your Bacillus horikoshi is having all these properties. In your reference, whether is this quoted or not, does any study is having the same results correlating with your study?
Results?
Yes.
No, ma'am.
Okay, thank you.
Thank you, ma'am.
Thank you so much, ma'am. I would like to take this time to once again thank our session chair, Dr. Katyani, ma'am, for taking the time out and giving valuable inputs to all the presenters. Thank you so much, ma'am. It was so wonderful to have your acquaintance for this conference. And a special thanks to all the presenters and participants who have stayed here.
Yes, thank you,
I'm
Palin.
sorry. Thank you, ma'am. Thank you so much.
Yeah, I
And
want to
a little,
congratulate all of those who have presented. They have done to their level and have also gained some knowledge from these presentations. And thank you once again, the organizers who have selected me as a session chair.
thank you so much, ma'am. It is very kind of you to acknowledge us in this. And a little note for the participants: the certificates will be provided by the mid of next week to your email ID. And you can all, the session for tomorrow, it will start again at 8:30. So we're looking forward to meeting you all tomorrow again. Thank you so much. Thank you, Katyani, ma'am.
Yeah, okay, thank you.
Thank you.